Gosia, gdzie jest Gosia? Żeby ona może sięgała z tym mikrofonem, biegała. Tam jest Gosia. Tak? To weź go. Tak. Mogę jeden zabrać jeszcze? Okay. Good morning, everybody. I'm really grateful for all those of you who decided to come here early morning, the third day of our conference. So I really appreciate. And the session is a population level session. So yesterday we had opportunity to discuss about life in different aspects of this life, a molecular aspect, a cellular, organismal, but today we transferred to completely different dimension, which is population. In today's session, our keynote speaker was Dr. Liran Shlush from Weizmann Institute, Tel Aviv. And unfortunately, this man is not here with us because of the war, yeah? And it's a difficult situation. We live in such times that we cannot perceive anything. And 1 week ago, he decided to come here, but after a few days, he wrote me, it's impossible, I'm drafted into the army. So that's a pity. He's not here with us. So we start with the second speaker, who is Professor Tomasz Grzybowski from the Department of Forensic Medicine. So a completely different subject. And he's also a specialist of mitochondrial genome analysis and also ancestry research of human population. So please, Professor Grzybowski, the floor is yours.
Good morning. Well, first and foremost, I'd like to thank the organizers of this conference for inviting me and giving me this unique opportunity to present our short message on human biogeographic ancestry within Europe on the very day of the Institute's anniversary. I'm essentially a forensic geneticist, but the truth is that most forensic geneticists are also population geneticists. And even though prediction of human biogeographic ancestry based on the DNA has mostly forensic applications, it might also contribute to better understanding of human demographic history and individuals' genealogy. All in all, I hope you'll enjoy this short presentation, even though your interests lie probably in completely different areas of life science. Okay, what is biogeographic ancestry? We usually use short term BGA. The term ancestry itself is a widely used term to describe genetic inheritance each individual has passed to them from their ancestors in the immediate past from their families, but for a longer period of time from the population members that have occupied the region when the person was born. And biogeographic ancestry refers to the population-specific component of every individual, which can signal its origin to a particular geographic region. There are several possible applications of biogeographic ancestry. In forensic, the principal task is to provide a proxy of eyewitness when it is not available to investigators, or to corroborate the eyewitness accounts when they are conflicting on unreliable. And in these cases, creating an accurate picture of a person's ancestry from the DNA they leave behind can direct an investigation towards a more manageable pool of suspects. In medical genetics, the possible applications may include checking the self-declared ancestry of participants of association studies of complex diseases where inaccurate ancestry assignments may lead to false association of population-specific variation with a particular risk of disease. And in medical genetics, there are some rarer applications like testing medical archive material. And finally, in ancient DNA studies, biogeographic ancestry is a commonly used tool to gain a better knowledge of the demographic history of archaeological remains. Well, there are several requirements which should be met if this forensic biogeographic ancestry will be reliable. First and foremost, we just need polymorphic DNA markers different in frequencies between populations. Now, the long-established study from the long-established finding from the study of human variation is that differences between individuals directly arising from population differences only account for a minority of total variation, of about 10% of total variation. The other 90% is independent of a person geographic origin. So in our studies, we should concentrate on this very minor proportion of human variation which sort of defines human population structure. Then we just need reference data from relevant populations. Is quite obvious requirement. There's no need to explain this requirement. Importantly, we just need a sensitive typing method of selected polymorphisms. Since we work on very challenging and degraded material, we just need sensitive methods suitable for compromised samples. And finally, we just need a reliable and mathematical model for ancestry prediction of an individual. Well, it's quite straightforward to achieve a continental resolution in human biogeographic ancestry testing. Several years ago, we developed a very simple test capable of differentiating between European, East Asian, and African samples. And the test included as little as 14 polymorphisms, and was the typing method was pre-NGS, from pre-NGS era, so to speak. So these polymorphisms were typed in two sensitive mini-sequencing reactions. So it's quite straightforward to achieve this continental resolution. However, if we consider prediction of ancestry within a continent in general, in Europe in particular, the task becomes much more challenging. Europe itself is a continent characterized by a huge cultural and linguistic diversity, but simultaneously is characterized by very low level of genetic structure. Slavs themselves inhabit well over half of Europe, but they are of course differentiated in terms of culture and language. but the existence of structure in the population of Slavs is highly disputable. In the past, there were some attempts to differentiate between Slavs and non-Slavs. The first attempts were made with haploid markers, that is, non-recombining portion of Y chromosome and mitochondrial DNA. And for example, we can see from this multidimensional scaling plot on the left side of this slide that we are able to differentiate between most southern Slavic samples and the Slavs living in Central and Eastern Europe. And the genetic markers used were Y chromosomal microsatellites, and we needed as little as 20 STRs to achieve that resolution. In that regard, complete mitochondrial genome sequences are also quite promising. More recently, we have shown that most of Russians living in the European part of Russia are really separated from Poles in the first dimension of multidimensional scaling. And indeed, there were some earlier studies on Y chromosome that pointed to some specific sublineages of Y chromosome, which are prevalent in Central and Eastern Europe, but very rare in Western Europe. Here we should point to M458 lineages and M558 lineages within a Y chromosomal haplogroup R1A1. M458 exceeds frequencies of 26% in Poland, Slovakia, Western Belarus. It's also quite frequent in Eastern Europe, and 558 has more Eastern distribution, so to speak. It exceeds 26% in Poland and Western Belarus, but also common in some Volga-Uralic populations of Eastern Europe. So we have some examples of Y chromosomal lineages, which are sort of characteristics of Slavs. We have also found some specific combinations of Y chromosomal haplotypes, microsatellite haplotypes occurring in Western Slavs. In that context, mitochondrial haplogroup U4 seems to be specifically informative. It's statistically more frequent in Eastern than in Western Europe. U4 frequencies are about 10% in Poland. Some populations of the Volga-Uralic regions have U4 at frequencies of more than 26%, but its diversity in that eastern regions is rather low, so we don't point to the origin of this particular haplogroup in Eastern Europe. We just think that it appeared in Central Europe, and if we were asked to point to the clade which is the most Slavic specific, we just should point to U4A2A, dated back to about 5,000 years. As you can see, its founding node, its founding node is represented in Western Slavs and occurs in all three language groups of Slavs, Western, Southern, and Eastern. As we can see, there are some haplotypes representing this subhaplogroup, which are present in some Finno-Ugrian populations of the north, and also in Western Europe, in Germany, in Central Western Europe, in Germany, we can find the representatives of this particular haplogroup. Well, to summarize our considerations on the haploid markers, they are really very promising in terms of predicting of paternal and maternal ancestries. However, a special care must be taken when interpreting the results of haploid markers, especially in highly admixed populations where haploid markers can give contradictory results. So it makes sense to use a broader range of genetic markers which can offer complementary characteristics. Unfortunately, the genome-wide studies employing autosomal markers in populations of Central and Eastern Europe are not very common. However, in one such study employed more than 166,000 SNPs, it was found that most Slavic samples are not distinguishable from other European samples. You can just see that Russians, Poles, Germans, Latvians, and even Estonians, all of them are clustered together, showing common ancestry despite ethno-linguistic differences. However, the use of as many of 500,000 SNPs really enables differentiation between most European populations, including Slavs. And moreover, there is a strong continent-wide correlation between geographic and genetic distance. So it sounds promising, right? But unfortunately, it's quite problematic in forensic science. I mean, the use of this modern ancestry arrays, including hundreds of thousands of SNPs, are simply not suitable for forensic casework, where we usually deal with minute amounts of DNA. So we can't just take, for example, Illumina array to type our challenging samples. So the best solution for forensic science is just to find the most differentiated autosomal markers and combine as many markers as possible into manageable assays. And that's exactly what we're trying to do in our current research. Now, let me summarize shortly our working plan from the project we are currently realizing. First, we took more than 400 samples of Slavs representing three major language groups, Poles and Czechs representing Western Slavs, Belarusians, Ukrainians and Russians representing Eastern Slavs, and Serbians belonging to Southern Slavic language family. For comparative purposes, we use all available data from the 1000 Genomes project, and unfortunately these data are not very abundant and representative for entire Europe. Nevertheless, it's better than nothing. At least we have three other language families represented here. I mean, Finno-Ugrian represented by Finns, German represented by Brights, and Romance represented by Spaniards, Italians, and Japanese. Spaniards and Italians. We additionally took Japanese samples to provide sort of background of East Asian ancestry in Europe. At the very end, we also used the data from Poles from the recent 1,000 Polish Genomes project. I'm going to present short description of this data at the very end of my presentation. Well, at the very beginning, we performed whole exome sequencing from populations of Poles, Serbians, and Russians, representing three major language groups I've just mentioned. And based on complete exome data, we selected 300 the most informative SNPs, the most informative in terms of distinguishing between Slavs and non-Slavs. The selection itself was based on machine learning approach and using three different classifiers. It's a matter of separate publication, and I'm not going to talk about this today. Anyway, based on this initial 300 SNPs, we finally selected 54, the most informative SNPs, which we evaluated using three different approaches used commonly in population genetics. I mean, the parameter of delta, that's absolute difference in other frequencies between populations. The FST distances is very, very commonly used in population genetics. And third parameter, so-called informativeness of assignment. And we finally selected 49 SNPs. We developed AmpliSeq panel, including more than 50 amplicons. We think that this AmpliSeq panel is well suited for forensic genetics, since, as you can see, the amplicon size range is from 125 to 275 base pairs, so the amplicons are short, and we think that it will be enough to analyze the graded and challenging samples. And the AmpliSeq panel is just dedicated to S5 Thermo Fisher next generation sequencing platform. Now, this is just a distribution of selected SNPs by chromosomes. Since we have no time to present all the evaluations of all the SNPs using three different population parameters, I've just selected FST distances as an example, just to illustrate the performance of the created panel. And now, this is just a heat plot illustrating pairwise comparisons between Slavic populations. And the bad news is that Slavs within a group are completely undistinguishable. I mean, Slavic samples is absolutely homogeneous in terms of genetic diversity, so we are unable to differentiate between Slavs themselves using this panel. But the good news is that when we analyze all studied populations by FST distances, Slavs are clearly distinguishable from other Europeans, not only from Japanese. As you can see, this distances, pairwise distances between all samples and Japanese is highly significant. But it was predictable since it's East Asian population sample after all. But most distances, most differences between distances in pairwise comparisons between Slavs and non-Slavs are significant. And this is just the multidimensional scaling, another analysis which illustrates the very same point. If we try to present differentiation between Slavs and MDS analysis, we see no structure in these samples. But if we use all European samples from MDS analysis, we can see that Slavs are distinguishable from other populations in both first and second dimension of multidimensional scaling. We also verified this observation using another analysis commonly used in population genetics, I mean principal components analysis, including the 1,000 Polish genomes database. And we can see that second eigenvector of principal component analysis clearly separates Slavs from non-Slavs, and also third component of third eigenvector in PC analysis also enables differentiation between Slavs and non-Slavs, while the first component separates Europeans from Asians represented in Japanese in our sample. And now let me finalize these considerations with the structure analysis. Structure is certainly the most popular method of visualizing population differences. So it's very commonly used in population genetics. Structure is the analysis which differentiate between particular samples based on differences in allele frequencies. In principle, it uses some sort of Bayesian iterative algorithm to place samples into groups whose members share similar patterns of allele frequencies. And structure applies some model to the data of K population or genetic groups, each represented by a subset of allele frequencies identified in the data. So the most important step in structure analysis is to calculate the likelihood of the data for a range of K values or genetic groups to provide sort of posterior probability of K. And for our particular sample, the number of K for which posterior probability is highest is 4. So we have 4 defined groups in structure analysis. And we have just example of structure analysis with the K number of 4, information of ancestry of samples, and so-called admixture model. I mean model which allows for gene flow or admixture between the populations. And to oversimplify it, this picture just shows that we are able to define one single group of Slavic populations clearly distinguishable from other studied population, but simultaneously we are not able to differentiate between particular Slavic samples. So, finally, the new panel, including as little of as 49 SNPs, enables distinguishing between Slavs and non-Slavic populations of Europe. And I believe that it's suitable for forensic applications due to the short amplicons. Two particular SNPs are the most promising in terms of BGA production within Europe. But unfortunately, but that's the fact, the populations of Belarus, Poland, Ukraine, Serbia, and European Russia are really highly homogeneous, and we are unable to differentiate between Slavs using this very restricted and little set of genetic markers. Thank you very much.
Very much, Professor Grzybowski, for your presentation. I saw that there were no uniparental markers in this set, yeah? Only autosomes. There were no sex chromosome, no mitochondrial DNA in the SNPs, yeah? You studied in this.
Since I'm a little bit dead, and after infection, so
okay.
I'm
No.
able to hear.
Okay. I think you should come back to the microphone. But I asked about the set of SNPs you used for the analysis. They were no sex chromosome, no mitochondrial DNA SNPs, yeah?
Yes,
in this set.
Just make one step forward from our usual analysis of haploid markers, and this time we concentrated on autosomal polymorphisms. It makes sense for most populations, but especially for highly admixed populations. This admixture is not very high in Slavic populations, but there are some regions of the world, like Latin America, for example, or in the Middle East, when the level of admixture is very high, so it's very risky to rely on haploid markers alone.
Okay. I just wanted, I thought that maybe it would be useful to include some of them, yeah? Okay. Are there any questions or comments? Piotr Kozłowski.
Thank you very much for interesting talk. You mentioned at the beginning that let's say biogeography take advantage of data generated in forensic genetic. And do you think that it may be in the opposite side, that data or markers identified in genetic geography may be used in forensic science, or maybe you know such an example where such a, let's say, population group identification is used in forensics?
So the question is about the other markers used in different areas, and about the possibility of using them in forensic science.
The question is whether, let's say, identification of population of, let's say, some forensic sample can be used in let's say, forensic investigation.
Okay.
Yeah. So
that's the principal goal for this kind of analysis. It's just an application of ancestry analysis in practice. I mean, in criminology. Unfortunately, these applications are not very common in our country. In some other countries, there are some specific regulations. In some countries just forbid use this kind of analysis for the institutions of law enforcement. But still, it's in most countries it's useful. There are some examples of very famous cases, so to speak, which were resolved using this approach. For example, the Madrid bomb attack in, I don't remember, 2015 or 2000. I mean, it was just demonstrated very, very clearly that the crucial biological traces just came from the person from Northern Africa. And it was a crucial information for the law enforcement agencies.
What about pollen? Is it allowed in Poland to use this?
Yes. In Poland, everything is possible. It's not forbidden. Okay. It's not directly. I don't know. There are no regulations about this in Poland. In Bydgoszcz we had some interesting case, including this kind of analysis. I mean, we analyzed some samples of saliva deposited or beer cans. This was just items from drug crime. Okay. Some sort of illegal lab was established in Bydgoszcz, and the crucial biological traces were the stains of saliva deposited on beer cans, and the police had no idea who may have left these traces, and asked us to provide this sort of biogeographical analysis, and we found that it was deposited by Asians. So it just directed the investigation towards some pool of suspects. Two particular Asians were identified as possible suspects, and then we performed our routine analysis of human identification, since we have reference material. In these cases, we just don't have any reference material, so we are trying to say about something about these samples based on this sort of analysis alone.
Okay. Okay, so I have a question. First of all, thanks, very interesting presentation, but I have a question about samples. To what extent these samples are representative to individual regions? Because, for example, we know that Poland for a long time was represented by samples from Szczecin, yes? And if we know history of this place, it is difficult to say whether the samples are representative for Poland or for eastern part of Europe more, yes? So, because as you presented, usually in this analysis you use something like about 100 samples, yes? So, of course it is very important, and we believe that after, when we obtain more data, maybe we will see a little bit more, but now it looks that there is no differences, but whether number of samples is important.
Well, to my knowledge, at least, there is no structure in Poland at all. The samples from Szczecin and Wrocław and Kraków or Suwałki are not distinguishable.
So what do you think it is, history or biology behind this situation?
Based on DNA analysis, I mean,
yes.
Of course there are cultural and historical differences.
No, but this, we have no structure, but this lack of structure can be from history, that so many times we move from one place to another, and or from biology, that people like migration, which took place about 200 years ago to 1,000 years ago, something like that. So biology or history is deciding about this lack of structure.
Okay. Just, I would say that we have no biological structure, so it's enough for us. We don't deal with history, with culture. We don't mix these areas in our forensic analysis, at least. Okay, for some population historical studies, it would be quite important. But still, as I mentioned, the use as many as 500,000 SNPs really enables to differentiate between Europeans, between Poland and Germany and Russia and so on. So.
In the future, if we will have more samples, you think we will be able to distinguish, or in situations so...
My strong conviction is that we will not be able to distinguish. So.
Okay, the last short question, because we are out of time.
Dzień dobry, dziękuję. Wójcicki Rafał. Mam takie pytanie. Myślał Pan o wykorzystaniu ranżacji strukturalnych, czyli structural variants, zamiast SNP-ów, bo od jakiegoś czasu wiadomo, że SNP-y jednak nie wykazują takiego różnicowania pomiędzy populacjami. Publikacja chyba była w tym roku albo w zeszłym roku. Chińczycy zrobili swój pangenom, żeby rozróżnić różne populacje chińskie w kwestii też wykorzystania w medycynie później. I no, tam używali tych nowych technik już, żeby to nie były SNP-y, tylko żeby było to różnicowanie strukturalne, długich fragmentów. Czy pan myśli w ten sposób? Jeżeli tutaj nie rozróżniamy SNP-ami, no to tak zastanawiam, że tą populację moglibyśmy rozróżnić wariantami strukturalnymi.
Mamy... the question...
Oh, I'm so sorry. I said in Polish. I'm sorry. Shortly, the question was just about if you are thinking to use the structural variants. So because SNPs are well known that they are not distinguishing well the populations. So structural variants, like for example in the recent publication about Chinese population this year or last year, when they've made pangenomics using long reads and distinguished really the population. Sorry about this. I didn't thought that I'm speaking Polish. Yeah.
Well, it makes sense to use a variety of different markers for this kind of analysis. And as far as indels are concerned, for example, we have just tested some sets of 30 or 35 indels for this kind of analysis, and they are useful in terms of continental resolution, but we haven't tried structural variants other than indels now.
Okay, yeah. So I was more asking about long structural variants, so not like indels, but like coming from actually rearrangements of a small fraction of genomes. So I think that would be good direction, because it's the way when the research is going now to actually. Because of the bigger differentiation seen in structural variation than in SNPs. Yeah.
Yes, there's one more consideration on structural variance. I mean, I'm just thinking about the method of analysis in forensic. That's crucial in our area. So it would be probably more difficult to analyze degraded material using this sort of markers. But it's just an impression.
Okay, thank you very much once again. And we go to the next session. From the human population, we transfer to viral population. But in fact, this viral population is very close to our population. I think even closer than we would like to. And our second speaker is Professor Krzysztof Pyrć, I think the most famous virologist in Poland. Probably many of you know him from TV or internet interviews. But in fact, he is a man who studied coronavirus much longer before the pandemic. So I hope he will tell us more about the coronaviruses and the modeling of the viral population. So, Professor Pitt, the floor is yours.
Thank you so much. Thank you so much to all of you for being here. Thank you to the organizers for the kind invitation. I was asked to tell a few words on this conference, and actually, like a week ago, I realized I was included in the session population at the life at the level of population. So those who know my research may be a bit surprised, and I share this feeling. So I speeded up through the panic, then I moved to anger, then to acceptance, and then I thought, well, we are doing things that are at least partially on the population level. So I'm going to tell you actually pretty broadly about the translation we're trying to do between the single cell, single tissue, and the population and humans. First, I know that most of you probably know me from different places, but just very shortly, I'm a molecular biologist by education. I graduated from Jagiellonian University. Then I moved to the Netherlands, where I work at the university hospital, studying viruses, actually, studying emerging viruses, trying to look for new viruses, trying to understand them, why they are actually emerging in humans, and what makes them so dangerous. And after I got my PhD, I decided to come back to Poland, where I set up my research team, actually continuing the work on emerging viruses with special focus on coronaviruses and flaviviruses. We also do some other stuff, but that's the main focus of what we do. And I was lucky actually to work with great young people who helped me to build the team, to build the research, and I was also lucky to work with experienced people who helped me to build the laboratories, because virology is unfortunately a thing that may be dangerous to the researchers and to the population, so for some reason they don't allow to work with the things that are interesting in a normal lab. So it took me like 10 years to build the high containment labs, but that allows us to work on guys like SARS-CoV-2 or MERS or avian influenza. So this is actually a great thing that this happened. And of course, everything changed in 2020 because we thought we had this nice quiet niche called coronaviruses. Well, it didn't work out that well. So all we did actually was redirected in one day to SARS-CoV-2, which was in line with what we do, but we had to actually leave a lot of science for this 3-4 years, and unfortunately focus also on some boring stuff like diagnostics, like sequencing, like monitoring of the SARS in Poland, like supporting the public services with research, but also with advice at different levels. But fortunately, we managed to survive, and we managed to keep our core research in-house. And actually, as I said, I'm going to tell you today on how we translate our research to what is happening, or how we try to translate our research to what is happening in real life. So I will refer a lot to the pandemic because it was actually a great lesson for us also in terms of science, because at the beginning, we thought, well, we know so much about viruses, we know so much about coronaviruses, we should be able to cope with that. But unfortunately, the problem was that the information that we had from the lab was not so really useful in real life. I remember the situation when a guy from Dr. Gakowski, he was heading the team that was modeling the pandemic in Poland at these days. He called me asking, well, okay, we have this new variant, you know the parameters of the virus, you have it in your lab. How this will translate to what we do, how this will translate to our models. And I was like, I have no idea. And that was like a trigger, one of the triggers saying, well, we're doing something wrong. If we're doing research and we're looking at variants, and we get this valuable data, we believe they are valuable, but when there is a question coming from the guy who is actually translating that to the forecast, I'm saying I have no slightest idea, then we are doing something wrong. And actually, that was not only me, because more people started to think that we should get more focused on how this may translate to, if we are working on emerging viruses, how our work actually translates to the safety of the of population, and we started to think how we can improve that. And then, if you think about translating the basic research to real life, you realize that there is so many things that are consequences of small genetic changes in the virus. It's not about identification of the virus, but it's also about detecting the virus, about diagnostics, about tracking this virus in terms of the prevalence, but also in terms of diversity, genetic diversity, how it's changing, how this affects us, how the beta virus variant is different from delta, and why Omicron is actually not that deadly, but is spreading like a wildfire. Then you think about drugs, you think about prevention, you think about the advice concerning the non-pharmaceutical interventions, like the mask, like distancing, lockdowns, all the controversial things, because we shouldn't do educated guesses, we should actually do the research and base it on the data. So to do that, we realize we have to understand what is happening. And that actually is something that we and others started to do. We started to think whether we can understand in a translational manner what is happening. And several initiatives have been started. This is one of the initiatives that we are taking part with. It's called Durable. It's a European network of research, not diagnostic, but research teams that are teaming up to actually prepare Europe for the next pandemic, because we are just after one, but we have to realize that this will happen sooner than later, considering the climate change, the urbanization, number of people, globalization, and several other factors. So we don't want this to happen, so can we prevent that? So we teamed up trying to understand what is happening and how we can translate that to the high-level advice. And this is just a slide from this project. And if you look at this high-level, these are these high-level questions: how to forecast, how to do scenarios, how to validate things. But actually, the project itself is a hardcore science. So we are trying to understand on a very basic single-cell, single-tissue level, single-virus level, what is happening to be able to answer these questions. And actually, this is very in line with what we do in everyday life. And I'm going now to show you a piece of our research that we do, what we focus on during the everyday science life. And we focus fully on understanding of the viral infection in the context of the human cell, human tissue, and human body. I will try to explain why I named these three levels in this one sentence. So, understanding, it means understanding a lot of processes. If you're talking about understanding the virus, you actually don't care about the virus. Virus is like, corona is like 30K of RNA, not so much. Some are 5K RNA, some are even not coding a single protein. Understanding the viral infection is actually understanding the human cell, because what the virus is doing is simply hijacking and redirecting natural processes. So you have to understand the human cell to be able to understand the virus. And that's actually what we do. We try to understand what processes and how the virus is using to infect a human. And we actually focus on a very, for me, important part, early stages of virus infection. Why I'm saying it's very important? This is because early stages of viral infection is the recognition of the target cell. So on this recognition depends whether you will get a pneumonia or you will get a liver damage, whether you will get a sick cat or a sick bat, or you will have a pandemic. What about pandemic? Because this recognition predetermines largely the species, the cell, the tissue, the specificity. So if you would look on the infection with the virus, what happens when somebody coughs on you and you simply inhale it? I'm talking about the respiratory viruses. The virus is getting into your nose, into your lungs, into your bronchi, and then it looks for the place to attach. So first, what it meets when it gets closer to the tissue is not actually a cell surface, it is a glycocalyx, so a thick layer that is designed to prevent the viral infection and prevent the virus from getting to your to your cell, amongst other things. And actually, the virus already hijacks that, so it's using this glycocalyx as the adhesion receptors, binding to the surface, and actually, viruses develop a way to go through this glycocalyx. Some even are encoding proteins that are structural proteins on the surface of the virus that are cleaving sugars, and then by cleaving and binding to different sugars, they are simply surfing to the cell surface. And once they reach the cell surface, they find their actual entry receptor, so the protein that becomes a portal to the cell. We work a lot of that. We describe several adhesion receptors for coronaviruses, like NR63 virus. We described that it binds to special sugars on the glycocalyx, and then it travels to binds to ACE2 protein, the same as SARS-CoV-2, but it was like 20 years before. For HKU1 virus, we described that it binds to sialic acid with special conformation. A very nice follow-up for this work has been published recently, where it was shown that this binding to this adhesion receptor is not only an anchor on the cell, but it also induces the structural change in the spike, resulting in the exposition of the receptor binding domain, opening of this domain. So it enables binding to the entry receptor. And also for many animal viruses, we describe this process in detail. But that's actually not all. Receptors and co-receptors, adhesion receptors, entry receptors are not the only determinants of the infection. If you look closer, you will see that there is a whole complex of proteins on the cell surface that are responsible for the entry of the virus. And then one of these factors are activation factors, because the virus, when it's in the extracellular space, it is in its inactive form and it cannot fuse. Once it binds to the entry receptor, there is an activation factor required, and this activation factor frequently is a protease that is cutting the spike protein. Everybody knows now the spike protein, but just very shortly, it's a protein, structural protein on coronaviruses that extrudes like 20 or more nanometers from the surface of the virus. It's constructed from the S2 rod-like domain and the globular domain on top that is responsible for receptor recognition. So once this receptor is bound by S2 and the protease comes it cleaves between the S1 and S2, and this triggers actually the structural switch that in S2 that brings the viral and cellular membrane to proximity, and by exposition of hydrophobic regions, it induces fusion forming a so-called portal through which the ribonucleoprotein is inserted to the cell. And actually what we found, for instance, is that some proteases are so unique that their so presence on certain tissue will predetermine whether the tissue is susceptible to infection or not. So we described it like three or 4 years ago for calicranes. But there is much more actually coming in this topic. Once the virus actually goes, so once all the requirements are fulfilled, so there is a co-receptor, adhesion receptor, entry receptor, activating factors, platforms like tetraspanins forming this complex, it enters to the cell, but then you have to realize the cell is pretty big and the virus is some nanometer scale. So it has a long way to travel. And we also did a lot of mapping for this, actually for flaviviruses, how they enter the cell, how they reach the point where they set up their replication factory, how they cross the actin complex, actin cortex, and how they travel through the cytoplasm. This is all regulated very, very nicely. So these are the parts that we focus on mostly, but then we are facing a small problem. I told you that viruses are intracellular parasites. They are so dependent on the cell. Actually, you study the cell, not the virus. And then for some reason, virologists think that it's a perfect idea to use HeLa cells to study that, or monkey kidney cells to study that. And for years, we were using these models to actually study viral infection. So you can imagine if you want to understand the cell and you want to understand what is happening with these complex things, with glycocalyx, with receptors, proteases, and then you're using HeLa cells, you will get pretty crappy results. And then to solve that, already Oh, sorry. And then the next thing we go for animal models, which is great because at some point you have to go to animal models. But if you look on the viral infection, you will see that, you start with mice, for instance. To make mice susceptible to the infection. Mice don't get infected with SARS naturally. To get mice susceptible, you have to change the mice, so you have to humanize it. But still, you will not get the same disease. You will not get the same response. You will not get the same immune system in the mice. So the data you get are not consistent with what you see in clinic. And then, of course, you can change the virus. You can adapt it. You can do the directed evolution. It sounds bad, but it's pretty simple. And then you end up with the mouse virus. But still, if you're trying to understand why Omicron is milder than Delta, and then you make a mice virus, you again end up with pretty crappy results. So we try to solve this problem by going somewhere else. And then how important it is, actually, this is an example I showed already on multiple lectures, so some of you already probably showed this slide, saw this slide. This is a real life example how important the problem is. One of the biggest failures of the pandemic. 2020, chloroquine approved for use in humans in Poland and many other countries. Chloroquine is anti-malaria drug. They made research and then they end up with the conclusion that chloroquine is inhibiting SARS-CoV-2. And I can tell you, if you take viral cells and you add chloroquine, you get perfect data, laboratory data. You get nice IC50, low cytotoxicity, looks good. So the problem is that they use this as without the second consideration and translated this to the clinic. Chloroquine was approved for use for another indications, so pretty good idea. And we said in the beginning, well, it will not work. So they were a lot of people was like pretty irritated with this statement, and they asked why. And there is a very simple reason. If you would ask me in 2015 how viruses are entering the cell, coronaviruses are entering the cell, I will tell you they bind to the surface, they bind to the ACE2 receptor or another receptor, and then this triggers endocytosis. They go inside into the endosomal compartment, they travel in the endosomes, and that once they travel, endosomes get more and more acidic. And once they get more and more acidic, the catepsins, the proteolytic enzymes, gets activated, and they actually slash this S1, S2 the junction, and they activate the spike, and diffusion occurs. Boom, you have infection. But then chloroquine comes, and what it does, it blocks this acidification. So very simple. The virus ends up in the endosome, but the catepsins are not being activated because the pH stays low, and then they simply go into proteasome, they get degraded, okay, they just get recycled. Nothing happens. You don't have infection. A few years ago, before the pandemic, we discovered, however, that viral cells are not that good to mimic a human body. And we discovered that if you take a natural tissue, if you take a human respiratory tract, a very different mechanism occurs because you don't need catepsins. You have a lot of other proteases on the surface of the respiratory tract that actually allows diffusion on the cell surface. So in your body, the virus is actually completely bypassing the endocytosis pathway. And then it's entering the cell directly on the cell surface. So if you block endocytosis, it's beautiful. It works in viral cells, but it has no effect in humans. And a lot of this kind of artifacts actually popped up during the pandemic. We also had to struggle with a lot of collaborators who were actually willing us to test a certain inhibitor because it works, and some of them got irritated, but not because I didn't want to collaborate, but because I knew that what they see is an artifact, and then there is no reason to follow that. And then how we try to address this problem is actually to change the way we are working. For sure, for many of you, the ex vivo cultures is an everyday life. For virologists, it's not. So I started actually working with 3D ex vivo cultures already in 2006. Now, models like this one, like human airway epithelium cultures, is a standard model. At this time, it was pretty new. Nobody in Europe had this. So we started to implement this for virological research, and we got really nice results. The model is pretty beautiful. What you do, you take human lungs, You isolate the cells lining the conductive airways, you de-differentiate them, and then you differentiate them on the liquid interface. So we feed them from the bottom and leave air on the top. And they start to differentiate. They start to differentiate to form a fully decidiated tissue with the variety of cell types. Producing snot is really, you have to really take care of them, washing the snot off, because otherwise they get suffocated. What is the most beautiful, they actually reflect the natural microenvironment of the infection. And beautifully, you can do things that you cannot do with a real human, because you can simply observe how the single viruses are entering the cell. So you can track really single virus infection. You can also modify the cells genetically, even though they are primary with lentiviruses or CRISPR-Cas, you can modify them to study single factors that may constitute, for instance, the risk factors for the severe disease. And you may actually interactively try to understand what is happening in the tissue. And then we realized, well, lungs, bronchi, that's nice, but for instance, for SARS, we need more, because the SARS is not only a respiratory disease, it gets spread to other parts of the body. And so we did develop the gut epithelial model, and we implemented that. And it's also the very same beautiful model where you get this fully differentiated tissue that gets infected with human enteric viruses, but also with SARS-CoV-2. So you can study why you get diarrhea, once you have SARS-CoV-2 infection. You can try to understand what is happening there again. And we developed also some other models. This one was actually with Małgosia Borowiak from Poznań for pancreas. With Professor Majka from Krakow, we studied the brain infection, and many others. And at some point we realized, well, it's nice, but we have to stop at some point. You can recreate 20 further tissues, but then it's actually at me too. Well, it gets infected. This also gets infected, but it doesn't bring the relevance. It doesn't bring the value, because you first should understand the interconnection between these tissues, and then only it brings a new value to the research. And actually, that's what we are trying to do now. That's actually the future. I was telling you what we were doing in the past. That's where we are looking in the future. What is our ambition? To make it complete. To be able to use the in vitro or ex vivo model to understand phenomena like the cytokine storm, which is still not fully understood after SARS-CoV-2. Why people are dying a week or two after they clear the virus. Well, maybe a week after they clear the virus almost completely. And whether you can use certain drugs instead of just stuffing them into the patient in clinical trials to block this interplay between different tissues. So a few examples of where we are now. We are already doing the co-cultures of different tissue. This is a very simple example of co-cultures with the immune cells, where we're trying to identify whether immune cells may act as Trojan horses, whether they can take the virus from single tissue to bring it to another, because macrophages gets infected with SARS-CoV-2, but the infection is non-productive. This is another example how you can use it to study the interplay, how the immune cells are actually taming down the infection in the tissue, and how these cells are actually reacting to the infection. So we can study also migration. So we do quite a lot of that. This is just simple examples. Most of this is not yet published, but the problem that we faced is actually a very technical problem. A very technical problem because the basic plastics that we have are not adequate for the research. So we are now collaborating with material groups from Denmark, from Warsaw, from many other places, and trying to actually translate what we have into a body-on-a-chip for virological research. While body-on-a-chip is well developed, if you realize you have to work with that on the BSF-3 or BSF-4 conditions, this becomes really a challenge. And we would like to have such a system to be able to understand what is happening and to apply it, as I said, to understand the infection in terms of the communication between tissues and the pathology of the infection in humans, but also to develop drugs. This is just an example of the study we did recently, where we developed an inhibitor for SARS-CoV-2 and validated it in natural tissue. But as I said, this is just an example. We are part of another initiative where we are actually looking on in a multinational team on the possibility to develop drugs for the future pandemics. Nobody's looking for SARS-CoV-2 treatment anymore. It doesn't really make sense, right? So we are now looking for things that can be developed for the future, and we are trying to look on the broad spectrum in context of the human tissues, and for that we need more and more accurate models. So with that, I'm almost finished. As I said, we are aiming to translate the basic research that we do, because we are a basic research lab. We are trying to translate that into practice. We are trying to bring it to the population to be able to provide advice and informed advice, and to you, to the decision makers, to the society in the future. For that we have to still work a lot to be prepared for the next thing that is coming. And with that, I would like to finish and thank you so much for being here. And if you have any question, you can ask them now, or you can contact me by mail or any other way. So thank you so much.
Thank you very much. And due to the discipline of the speaker, we have much time for discussion. So we are open. Jacek Kolanowski.
Thanks so much for this very, very exciting presentation and the great results there. I wanted to ask you about particularly a part of certain personal susceptibility towards the infection. I think you hinted towards that a little bit through your presentation, and you've been talking about the requirement for the expression of different proteins, for example, on the surface of the cells and so on, which are involved in the process of internalization and then proteolysis and so on. Have you guys been able, or have you looked into these heterogeneity in the levels of expressions, for example, in different tissues or in different between different patients, of these critical proteins involved in the internalization process? And is there anything in that, or you think there are other reasons for those differences in this?
Yeah, so we tried to look into that, and not only we, a lot of research groups looked into the risk factors, as they were named, right? The genetic background that is directing the cause of the disease. The main problem is that while several of these factors were identified, and also there are differences in the levels of receptors, for instance, that are affecting the cause of disease. The main problem is that the main risk factors are not really genetic. So at least for SARS-CoV-2. So you may find these markers that will show you the 1-2% higher risk of severe infection, for instance. But to be honest, if you look for age, you look for sex, if you look for underlying diseases, this is simply overlying completely the overall risk. So I would say there are differences, but they are not that important. For SARS-CoV-2, that's not necessarily applicable for others. And then you may also ask for a question whether some people are completely immune to the infection, for instance, because that is what is happening, for instance, for HIV, that there are deletions in the receptor gene that are blocking the infection. And to answer that, I would say we didn't find such a marker.
Thanks for that. But I think you hinted into something which also I think is very important to distinguish. You mentioned about the genetic markers. What about the phenotypic markers of the not only expression of each of the proteins, but also relative levels of those proteins which need to collaborate together for the cells, for the virus to go inside.
So
On the level of the host, I would say. Yeah?
So I would say on the level of single host, you will see that differences. You will see that presence or absence of certain receptors or activation factors will guide the infection. But very often that's not that simple, because you have also ways that viruses bypass this process. So if you're asking whether the TMPRSS2, for instance, levels will direct the infection in humans, in lungs, yes. But then if you look on some other tissues, we did discover that, for instance, in pancreas, a very different pathway is being used. For instance, the catepsin pathway is being used in the pancreas, but not in the lungs. So yes, these factors, the pattern of expression of certain proteins will affect the cause of the disease and will affect the susceptibility of a single tissue. But I don't have simple answers to you, like if you don't have it, you don't have infection. It's as simple as that. And also probably due to that, efforts like using camostat, which is actually something like chloroquine, but it inhibits the TMPRSS2, it inhibits the serine proteases. It actually failed in clinical trials, most likely because there are ways to bypass this pathway.
Thank you.
Yeah.
Professor Filerowicz.
Very interesting presentation, and I would like to go a little bit to the lower level of life organization. What about single cell level? Are you able to analyze how virus behave in different type of cells? So, but I think that, for example, genetic variability, for example, in one type of cell and another type, whether it is able to produce functional virions in some type of cell, whereas in other not. So is it possible to observe, and do you see some really important differences?
Yes, we do observe the pretty important differences. For now, we were limited to the cell lines, and we do see that the infectivity of the viruses produced in one cell line is lower than in others. And we associated also with the changes in the glycocalyx, for instance, in the glycosylation pattern. For natural tissues, because that's what we are aiming for, we are now in process of that, because we are going into single cell level. This was not possible before for a very simple reason. You need to have viable cells to do the sorting. Now this problem is solved. So we are now preparing for going into single cell level. I'm saying why the problem is solved, because if you have a Bilza-free virus and you have infected cells, you don't really want to put them in a normal sorter, because then you may affect some of your coworkers, like kill them. So yes, we do see these differences. These differences are striking, and then we studied these differences in cell lines. We don't fully understand them, except for the fact that the translational modifications do play a role there, but we want to focus on them on the tissue level using the models I have described.
Are there any questions? Paweł Zmora.
Thank you for a very interesting talk. You mentioned that there's different pathways to the entry of the cells, but the virus is evolving all the time. So the Omicron is more dependent on TMPRSS2. Do you think that the next virus variants will be depending still on TMPRSS2 or other cellular factors?
Are you sure that Omicron is dependent on TMPRSS2?
Yeah, I was talking with Stefan Pullman, and his last publication is really showing that.
Yes, well, we do see it a bit differently than Stefan. So we do see that this dependence is more limited. And then if you look on the serine protease inhibitors, if you use serine protease inhibitors, you will see that the effect is milder for the Omicron variant. If you look for natural tissue and knockdowns or knockouts in natural tissue, you'll see that this dependence is lower. So we do believe that either the accessibility of the site is lower and the virus is already starting to bypass this pathway, still partially being dependent, or it has already switched to another protease, also serine protease, which is actually masking the the effect. So answering your question, yes, I do believe that this is happening, and the viruses will probably switch to other proteases like HKU1 virus, which is using kallikrein instead of TMPRSS2. And I do believe that it's of utmost importance to study that in the proper models, because you may miss this fragment and you may easily get tissue adaptation of the isolates, which is also a big problem, because very rapidly after you bring them to lab from humans, they change the protease specificity. And that's also one of the problems for studying some of these viruses that we do use in the labs. They don't infect humans anymore, because they switch, for instance, to the caltepsin pathway, which is inactive in human lungs. So we do see that a lot. Yes, answering your question, I do believe that the future variants should be monitored for the protease dependence, and then I do believe there are many unanswered questions already for Omicron, despite the fact that there is a lot of people believing that Omicron is using 100% TMPRSS2. I do think it's more complex.
Okay, thank you.
Are there any other questions, comments? I don't see any, so thank you once again.
Thank you so much.
And now there is time for our next speaker, who is a plant geneticist. So he will tell us about the population studied on the plant model, but I think it will be quite universal, because as far as I know, Professor Rykowski studies recombination, so general process of recombination, but also focused on mechanisms of gene expression regulation with the use of chromatin modification, yeah?
Yeah, that's true, but I won't be talking about chromatin modification.
So not today.
These aspects.
Okay.
Yeah, and also, thank you very much for invitation, for having me here. I'm really happy to be here and participate in this session and the whole conference. I'm probably the first speaker and the only speaker during this session talking about plants. So I hope that would be also something interesting for you. I'm not population geneticist, so I will be trying to show a little bit perspective, population perspective on this, on our research, but there will be a lot of molecular work as well. Right, so I will be talking mostly about the interplay between meiotic recombination, crossover, and DNA polymorphism, how those two things interplay in living organisms. But first I want you to acknowledge the fact that actually the huge variation that we observe in sexually reproducing organisms is that we can see in, for instance, this picture of random people from one institute is a consequence of huge variation that is mostly due to variation at the DNA level. So this is not environmental impact. These changes are not due to environmental factors, but rather to the genetics, to the DNA material. And of course, this is the result of accumulation of mutations over hundreds of thousands of years of evolution. But at a scale of short-time scale, the major results of those differences between phenotypes that we observe in living populations are due to two processes that are happening during sexual reproduction, and especially during meiosis, during gametes production, because this is a process that leads to gamete formation. That is random segregation of chromosomes. So during meiosis, we end up with reduction of chromosome number, and only one chromosome from a pair is segregated to each gamete. And the second thing is recombination, crossover recombination that is happening specifically during meiosis, and it's actually crucial for generating new combinations of alleles. And this is a scheme showing how it is happening. So chromosomes from both parents pair and exchange reciprocally their fragments, and in the consequence we generate a new combinations of alleles along the chromosome. And of course this model is quite universal, but from the human perspective we always think that we've got two individuals that generate new progeny. So because we've got mother and father, and they of course differ at the level of genetic information. They have differences at the genome level. But this is not always the case. So actually, many plant species are predominantly self-pollinating. It is estimated that about more than 20% of plant species are self-pollinating. You can see here some of important examples of that, like wheat, barley, rice, cucumber, pea, also our model plant Arabidopsis thaliana. So this is quite important from our perspective, as these are major crops. But the self-pollination is a very specific process in which the same individual is producing male and female gamete and then fertilize itself. So in consequence, this has very dramatic consequences at the population level, because during selfing, each generation is drastically reducing the heterozygosity level. Yeah? So you can see that each generation, the heterozygosity, so differences between two chromosomes in pair are decreasing, are dropping down. And in consequence, many of those species that I showed here, they are completely homozygous. When you take a random population of, for instance, barley in the field, you will see that they are exactly the same. All of them are completely the same. They have exactly the same genetic information. There's no variation within the population of those individuals. That's quite interesting, because of course it is against the variation that you need to keep in the population to be able to adapt to changing environment. And in such a situation, the only case in which you introduce a new variation is via events of migration from some distant populations, because even the most predominant self-pollinating species, they always have some level of outcrossing that is allowing to introduce new variation in the population. So after migration, you've got a hybridization, so outcrossing, and then you've got a hybrid individual. But then again, the population is developing through many generations of inbreeding. And in the end, you've got mostly homozygous population with some chunks of DNA that will be heterozygous. And that was something quite interesting for me. I wanted to see how this would affect crossover distribution along the chromosome. So, so actually, regarding DNA polymorphism, how polymorphism can contribute to crossover frequency and distribution on the chromosome, we can ask this question, answer this in two different ways. So first of all, we've got kind of cis-acting factors. Cis-acting factors, like asking a question: what is happening when the whole chromosome is homozygous, with small chunks are heterozygous? How it affects the distribution of crossovers? And there's also another aspect. Because variation at the DNA level can result in a variation in genes that are producing proteins involved directly in the recombination. These are trans-actin modifiers of recombination. So in my previous work, I identified a few of them, like HI10 or SNI1. Very recently in my lab, we identified this factor that are contributing to the distribution of crossovers at the genome-wide scale, shaping then the population, the crossover in the population and variation within the population. But I will not talk about this part. I will focus mostly on the part with cis-acting factors. This has quite significant consequences for population, especially focusing on the last recent papers from my lab that were published this year. That's okay. Yeah, but first I need to very briefly introduce a simplified scheme of how crossover are formed. So all crossover are first initiated by double-strand breaks that are programmed during initial phases of meiosis. And then we've got two pathways leading to crossover formation. ZMM pathway, that is the major pathway, and it generates crossover, class 1 crossover. And also the second minor pathway that is generating class 2 crossover. I will not go into details how different are they from the molecular level. I just need to, just want to remember that Actually, this scheme is quite universal. This is exactly the same in human, this is exactly the same in plants, because those processes are very well conserved. So actually, also in human and in plants, 90% of crossover comes from class 1 and only 10% comes from class 2. And of course, because we are talking about polymorphism in the cis aspect, so we need some molecules that sense, that detect the polymorphism between the recombining the chromosomes. And it is well known that there are mismatch repair proteins in all eukaryotes, and the major component of this complex is that can detect and sense polymorphisms is MASH2 heterodimer, so MASH2 with some other proteins that can detect mismatches between in heteroduplex. So I will now show the method that we use to measure the combination. So one of the methods that we use is based on a reporter system, fluorescent reporter system. So we've got two fluorescent reporters on one chromosome, genetically linked, so close to each other on the chromosome. And then when we cross this reporter lines with any other Arabidopsis lines, then we've got the hybrid, and the progeny of the hybrid is producing the gametes that will actually, dependently where crossover happens, we will have either parental combinations with two reporters and non-reporters, so this is parental combinations, or recombinant when crossover happens between them. Then we've got a single reporter or a single red reporter or single green reporter in the transfer to the gamete. So because those reporters are expressed exclusively in seeds, we end up with color seeds, and under the microscope you can single Arabidopsis seeds. Arabidopsis plants can produce thousands of seeds, so we can simply calculate the number of recombinant single color seeds and the total number of seeds, and then calculate recombination frequency within this interval very precisely. And that was something that I did during my postdoc. I actually created different combinations of this heterozygosity along the chromosome. So completely homozygous combinations, completely heterozygous when the two chromosomes come from two different parents. And then, as in the situation when only a chunk of chromosome, a fragment of chromosome is heterozygous, and the rest of the chromosome is completely homozygous and opposite. So we call them hom-hom, het-het, het-hom, hom-het, and in this system, I can measure recombination only in this region defined by those two reporters, so I always measure in this part. And when I measure recombination for those plants, I observe that actually there's not much difference between hom-hom and het-het. You can see that there is some difference, but not huge. But very surprisingly, when this region in which we were measuring recombination was heterozygous, then recombination was dramatically elevated. And opposite effect was observed when this region was homozygous. So then we lose recombination. So the interpretation of this result is that crossover is somehow attracted or stimulated in heterozygous regions where it is juxtaposed to homozygous regions on the same chromosome. That was quite unusual because the paradigm in genetics was a little bit different. But of course, we can ask what is the biological meaning of this. And this is, we can find quite nice explanation for that. So imagine if we've got a fragment that is homozygous and crossover is happening in this somewhere in the homozygous region. Wherever we place the crossover, we always end up with the gametes that will be exactly parental combinations. So we will not generate any new variation. However, when we place crossover in the heterozygous region, wherever we put it, we always generate new combinations of alleles that will be different from parental. So this will generate new variation in the population, and that's probably the reason why this effect is actually achieved. And we know also that it exists also in other plants, for instance maize. So it's quite interesting. But of course, we wanted to ask the question how this polymorphism is sensed. How is this polymorphism detected? What is the mechanism? And actually, Julia in my lab, a very talented PhD student, she generated a mutation in MASH2 group gene, so this gene that is responsible for detecting polymorphism, in all those different genetic contexts using CRISPR techniques. And then she measured recombination. So yeah, the question is, because this is in mostly, of course, will be affected class 1 crossover, so this is mostly this pathway that will be affected. And when she measured recombination in the MASH2 background, so when mismatch detection is not working, we observed that this effect completely disappeared. We couldn't see any further increase in recombination. We see some other differences, but we know that they have completely different origin. So that was something showing that actually MASH2 and mismatch repair system has a pro-crossover function for class 1 crossovers. That was quite unexpected. And then we wanted to see what is happening for the second crossover pathway. So the problem is that actually this pathway has, this is a minor pathway, so only a few crossovers are formed. We can increase it artificially by knocking out one of the DNA helicase, FANCAM or RECU4. Those are very conserved helicases that work also in human. And then we can boost class 2 crossover. And at the same time, with knocking out some of the genes from this pathway, we can block completely class 1 crossover. So then in this situation, in the FANCAM ZIP4 mutant, we have only crossover from class 2. And then we have observed that there's completely different behavior. Now, crossover are stimulated only when the region is homozygous. Yeah, you can see here home. This region is homozygous, a road of crossover, similar situation in home headline. While anytime this region is heterozygous, recombination was very, very low, showing that actually the two crossover pathways, have different sensitivity towards polymorphism. Okay, so is it also triggered by MASH2? That's another question that we ask. So for this, we combined MASH2 mutation with FANCAM Z4 mutation to study how MASH2 affects class 2 crossovers, and this is the result. So now recombination can also go to those heterozygous regions. So showing that for class 2 crossover, the MASH2 has anti-recombinational role, quite unexpected for us. This was, of course, made by studying the just short intervals on the chromosome, but we wanted to see it on the population scale. So we used a technique that we call GBS, gene typing by sequencing, in which we generate big F2 population between two different accessions of Arabidopsis to have a polymorphism that will be used to map where crossover happened, because we need to have some markers, so this is SNPs in this case, and generated hundreds of individuals, F2 individuals, and then generated libraries for each of them separately and pulled them together and sequence. And then based on this, based on combined data from all those individuals, we can see how crossover is distributed along chromosomes. And this is, we generated this for different mutant combinations. I won't go through all of them. It's probably a little bit boring. I just want to show you how these crossovers are distributed along the average chromosome arm. So in wild type, so also this shaded region here is showing the SNP density. So you can see that the wild type that is shown here in green is quite active in polymorphic region. However, so this is mostly class 1 crossover. While those class 2 crossover, FANCAM Z4, MASH2 FANCAM Z4, they are going mostly to non-polymorphic region. Yeah, so this is in line with what we showed before. Also, interestingly, we tested the other helicase, RECU4. So those mutants of RECU4 has very high level of crossover, but knocking out MASH2 is also leading to a further increase of crossover frequency, proving that it has this anti-recombinational role for class 2 crossovers. Right, yeah, this is the model that we constructed. So, okay, so we've got the double-strand breaks, then strand invasion during initial stages of recombination. Then MASH2 is detecting polymorphism in these heteroduplexes that are formed and either stimulates the ZMM pathway to do class 1 crossover or recruits RECQ4, FANCAM, helicases to dismount the joint molecules and to block class 2 crossover. So in consequence, it has anti-recombination role in this pathway. Right. But I didn't tell you that actually this what we found about this redistribution of crossover that is stimulated by heterozygosity, by polymorphism, is something quite unexpected, because there were quite a few data from other organisms, for instance from budding yeast, mostly from budding yeast, but also we had data from Arabidopsis that at the hotspot level, crossover is always happening only in non-polymorphic regions. Yeah, so this is so hotspots are short fragments on the short regions on the chromosome of the few kb in length, in which crossover is actually taking place, the recombination size. So you can see here that so those tick marks are indicating the SNP density. So we can see that. And then this is crossover. So we can see that always crossover is going to non-polymorphic regions at the hotspot scale. So yeah, there is a kind of paradox, because on the scale of whole chromosome or chromosomal regions, recombination is stimulated by polymorphism, while on the hotspot scale, it's actually inhibited by polymorphism. Something is not wrong. Is not okay here. Another very talented student in my lab, Maja Szymańska-Lejman, wanted to test this at the hotspots level, and she developed a very cool system to study hotspots by constructing very short intervals, these reported intervals, but with the size of just up to 50 kb, so very short in comparison to the intervals that we use for chromosome mapping. And then under the microscope, she can select recombinant seed. You can see red alone or green alone seed. We know that those seeds experience crossover within this interval. And then with long-range PCRs and then Illumina sequencing, we could identify very precisely where crossover happened with the resolution of single SNP for this interval. And then when we do it for hundreds of recombinants, we can do the profile of crossover along the interval. And she did this for several intervals, but I will show you only one of them, chili pepper. We call it chili pepper. It's located on one chromosome in the quite polymorphic region, because it is quite close to the centromere. And this interval after sequencing has three very well distinct, very well separated hotspots. You can see the first hotspot, Arrow, Coco, and Nala. We call them after our dogs that we have in the lab. So actually, I must say that Coco, the strongest hotspot, of course, this is my dog. And this is a benefit of being a boss in the group. And of course, here you can see also the SNPs distribution. And also in red are indels, small indels. So you can see again that where there is a lot of polymorphism, there is no hotspot. Hotspots are always in regions that have some regions devoid of polymorphism. So this is in line what we knew about the hotspot at the about recombination at the hotspot scale. So how polymorphism affects recombination? So to test this, we decided to use again the same mutant, MASH2 mutant. That is, like MASH2 mutant is, let's say, blind for polymorphism, cannot see polymorphism anymore. And indeed, when we compare inbred, MASH2 mutant is not different than wild type, because there's, of course, in inbred you don't. This is completely homozygous. You don't have polymorphism, so there's no difference between mutant and the wild type. However, in the hybrid, where we cross two different accessions that has a lot of polymorphism, then when the mutant it has very different recombination frequency than the wild type is lower in this case, and this is when we did the seed typing, the method that Maya developed. We observed here in red that recombination is decreased in all whole spots. We were a little bit disappointed, because actually you can see that all crossover hotspots are preserved in MASH2 background. So crossover is always going to the same locations. However, one thing that we notice is that this hotspot that is very, how to say, there's not much polymorphism in this hotspot. It loses only slightly recombinational activity, while the hotspot that is very polymorphic, it loses most of the recombinational activity. And we tested this for many different hotspots and plotted this, and you can see on this plot that there is a very strong correlation between the polymorphism density around the hotspot and relative crossover frequency in wild type compared to MASH2, showing again that hotspots that are polymorphic, that has a lot of polymorphism around, are stimulated in wild type. So this is still in the line and probably explains a little bit how those two things are really happening at the same time. And Maya also did very cool stuff, so very cool things. So she generated a lines in which only chili pepper, this very short 26 kb chili pepper interval is heterozygous, and the rest of the whole genome is completely homozygous. She backcrossed several times to one of the parent. So this is she achieved a line that we call R square. R square is only heterozygous in the region, and the rest of the genome is completely homozygous. So this is just to remove all the effects from other regions. And you can see that this is actually the data. It shows that recombination frequency is boosted in this R square lines compared to both inbred and hybrid, showing that this juxtaposition effect is always also working at the level of single hotspot, because we tested this also for single hotspots, and opposite is for the lines in which everything is heterozygous, only chili pepper is homozygous. And in MASH2 this effect disappears, proving that again MASH2 is playing a major role in detection of this polymorphism and then stimulating crossover. And yeah, this is a model. So okay, we've got the two hotspots. Recombination is always taking place in regions that are devoid by polymorphism. But if this is surrounded by polymorphism, it will stimulate recombination in this region. And with this slide, I want to acknowledge people that were involved in the project. So once again, Julia and Maya, they defended this year, and they also published their major papers this year. And also I want to acknowledge Wojtek that did a lot of bioinformatical analysis for all those data, and our collaborators, Ian Henderson from Cambridge University and Scott Pethick from University of Pennsylvania. And yeah, thank you for your attention. I'm happy to take questions.
Thank you very much. And I open the discussion. Professor Kamińska-Kaczmarek.
Thank you very much. Very interesting presentation and data. And I wonder if there is any sequence or structural peculiarity about the hotspot, that recombination takes place in hotspot. Why?
Yeah, very good question. So for a long time we hope that this is actually polymorphism, and we know that it is contributing, but definitely the major factors are related to the chromatin stage. So first of all, formation of double-strand breaks. This is done by a protein complex called SPO11, and SPO11 is only acting in the open chromatin state. So for instance, all hotspots in Arabidopsis and in plants, they are always located in the promoter region, sometimes at the 3' ends of the genes, so where the chromatin is opened, when there is no much
abundance of
yeah, I mean, this is also contributing somehow, but the major factor probably is just opening of the chromatin, and active chromatin marks like histone acetylation, H3K4 trimethylation, H3K4 dimethylation. Those factors are contributing, but we are still testing. On the other hand, like in human, this is a very different story, because in human we've got a major determinant of crossover formation is PRDM9 protein, that is the fastest evolving protein in human genome, that is actually defining the location of hotspots. It's a little bit different story. So yeah,
Just a quick question, because you knocked down RECL4, but what about there are other helicases in plants and BLM or some other?
Yeah, I mean, RECQ4, I didn't mention this. I should say it because I was a little bit trying to make very fast over this. I was afraid that I will not meet the time. But RECQ4 is actually homologue of BLM. This is a perfect ortholog of BLM. Pardon?
I was wondering if there is redundancy of the function, because in human cells, to some extent, they do the same. If you knock out RECL, the BLM is doing.
Yeah. I mean, that was tested in plants, and we know that those two, FUNCAM and BLM, RECQ4, there are two copies of RECQ4 in Arabidopsis. They are contributing to controlling recombination in meiosis. There are other homologs, of course, there's plenty of them, as in human, but they don't have function in meiosis. So these are meiosis-specific, let's say.
Okay, are there any questions? Piotr Kozłowski.
Thank you very much. Great talk. I learned a lot from today's talk. And do I understand correctly that recombination is, let's say, triggered by some SNPs or heterozygosity in specific, but generally is more likely to occur in a region, let's say, less dense in SNPs?
Yeah, I mean, I don't have a slide for that. We did some kind of correlations between them, and it seems that there is a U-shape correlation. So to some extent, the SNP density is stimulating recombination to something about 15 SNPs per 100 kb, or less. I don't know, maybe 5. I don't remember at the moment what is the threshold. But after that, it's just drastically dropping down. So at some point, so SNPs are stimulating to some extent, but if the density of SNP is too high, this will be inhibiting. But of course, everything depends what is the scale that we are testing.
Yeah, so it's what I understood, so I understood it correctly. But my question is actually, do you think that distribution of recombination points or recombination hotspot has some evolutional advantage, or it is just a reflection of mechanisms how they occur?
Yeah, I mean, if you can turn on the presentation, I don't think that this is possible. I've got some, okay. Yes, so I've got some additional slide. Yeah, so this is a slide, this is unfortunately showing only the pattern of crossover for plants. But you can see that there is a very strong correlation between the genome size and the distribution. So if the genome is very big, like for instance in species like, for instance, wheat, maize, that has huge genome in comparison to Arabidopsis, then crossover is always shifted, biased to the end of chromosome. So you've got this U-shape distribution of crossovers. And the major reason is that there is probably a lot of DNA methylation that is inhibiting, DNA methylation is inhibiting recombination. And also transposons that of course make it impossible to make crossovers. You ask about, I mean, I think that it's difficult to say for plants because there's not much you can, sometimes at some point, this is something that also breeders are struggling, that the recombination is happening only at the end of chromosomes and what to do. I mean, if you've got some useful variation inside the chromosome, you can't break the linkage because there's no crossover happening. And this is also one of the topics that we are working on, to target recombination to specific sites by DCAS9, by DEDCAS9, to modify chromatin inside the chromosome to stimulate recombination. Yeah, that's, I mean...
Yeah, thank you. It was just by the curiosity. Thank you.
Answer the question.
I'm afraid we have no more time for discussion, so thank you once again. And the last speaker in our session is Dr. Katarzyna Klonowska from our institute. She completed her PhD under the supervision of Professor Piotr Kozłowski, but she just came back from her 5-year postdoc in Harvard Medical School, and I think she will tell us more about the results she got during her postdoc research. Kasiu, the floor is yours.
Thank you very much for inviting me to this conference. As Dr. Henschel just mentioned, I will present today the main project that I was working on over the recent years during my postdoc in Harvard Medical School and Brigham and Women's Hospital, and also some new results that I was working on recently here in Poznań in the Institute of Bioorganic Chemistry. The title of my talk is Hidden Landscape of Tumorigenesis Driving Mutations in Tuberous Sclerosis Complex. Tuberous Sclerosis Complex, in short TSC, is a genetic autosomal dominant disorder which occurs due to pathogenic mutations in one of the two genes, TSC1 or TSC2. Together, TSC1 and TSC2 inhibit the mechanistic target of rapamycin mTORC1 signaling pathway. According to the Knudson 2-hit model, germline TSC1 or TSC2 mutation first hit is followed by a somatic mutation in the second allele of the same gene, which we call second hit, that results in aberrant activation of mTORC1 signaling and development of tumors in different body sites and organs. A comprehensive analysis of the genetic changes in TSC tumors is essential to improve our understanding of how they develop and to explore some new therapies for TSC. In my research, I have mostly focused on the investigation of somatic changes underlying development of skin tumors in TSC. Among the hallmark skin tumors observed in TSC patients, there are hypomelanotic macules, which are white-colored spots that can occur anywhere throughout the body. The second type of skin manifestation characteristic for TSC is the chagrin patch, which is thickened, elevated skin, mostly occurring on the lower back. The third one is angular fibroma, which may occur on the finger or toenails. And the last one is facial angiofibroma. So these are reddish bumps that occur mainly on the center part of the face, on the nose, or on the cheek, and they occur in most of TSC patients. And here I would like to introduce abbreviation for facial angiofibroma, which is FAF. So please remember, because I will be using it often throughout the presentation. In 2014, our lab in Boston, in collaboration with Thomas Darling, dermatologist from Bethesda, performed a massively paired sequencing analysis of fibroblast-like tumor cell cultures from a total of 29 TSC skin tumors, including facial angiofibromas. And this study has been performed to investigate somatic mutations underlying development of these skin tumors. And 90% of the second hit mutations were small indels or single nucleotide variants, in short SNVs, or dinucleotide variants, in short DNVs. What is interesting, almost half of these mutations were CC to TT substitutions, which are known to be generated by UV radiation, and they were seen exclusively in facial angiofibroma, which occur on sunlight-exposed facial region. And at this point, we knew that UV-induced mutation is a consistent second hit in TSC facial angiofibroma. However, the extent and prevalence of UV-induced mutation, as well as contribution to the FAF development, remained to be explored. And therefore, to investigate the extent and range of somatic mutations in facial angiofibroma and TSC patients, I have developed a new method which is an ultra-sensitive strategy for characterization of UV-induced mutations in TSC facial skin. We call this method multiplex high-sensitivity PCR assay, in short MHPA. This method employs barcoding of single DNA molecules, which enables compression of the data to consensus reads what leads to error suppression, and this enables reliable detection of variant allele frequencies of extremely low frequency, up to 0.01%. And this leads to tenfold improvement in sensitivity in comparison with previous MPS strategies that were used for characterization of low allele frequency mutations in TSC. And this method has a universal application, and it can be applied also beyond the study for TSC patients, for example, for investigation of residual cancer disease or forensic genetics. For this project, I have developed two different MHPA assays. The first one's for the analysis of TSC2 gene, and this assay contains 40 amplicons that cover most 75% of reported pathogenic mutations in TSC2, including all hotspots. The second assay was designed for TP53 gene, which is a gene most often mutated in cancer, including skin cancer. And this assay includes 12 amplicons with coverage of 93% of all reported mutations and mutation hotspots previously reported in normal keratinocytes as well as skin cancer. This is the study design. So in the first step, I've performed thorough validation of this new strategy to make sure that it delivers reliable results. And then I have applied MHPA analysis to a total of 81 samples, including 66 skin biopsies. And among these samples were both samples from TSC patients and samples, so-called non-TSC samples, which are samples from healthy individuals as controls. And among the TSC samples, we had a set of 24 TSC angiofibromas, 10 TSC normal skin from different body regions and subject to different extent of UV exposure, TSC angual fibroma, 3 nipple angiofibromas, as well as control samples from blood, semen, and buccal swab. Looking at the non-TSC samples, we had skin from different regions, also subject to a variable extent of UV exposure. And in this project, I have performed characterization of somatic mutations in TSC2 and TP53, including analysis of UV mutation signature in skin, which was followed by the analysis of the association of the somatic mutation prevalence and clinical characteristics of the skin biopsies and the patients. The MHPA analysis led of the set of 24 TSC angiofibromas led to the identification of over 100 low variant allele frequency somatic point mutations. And in the top plot, I have visualized the distribution of the allele frequencies of all the identified variants, where CC to TT substitutions are marked in orange, while other mutations in blue. And what is really interesting and unexpected is the very high fraction of CC to TT mutations, which was close to 30%. And in the plot below, I have summarized the total number of somatic mutations identified per each of the analyzed lesions with the same color code. And here we can observe that 2 or more somatic TSC2 mutations were identified in most angiofibromas, what reveals that these small biopsies contained at least 2 different FAF clones. We think that these mutations are specific to facial skin and have a functional meaning based on the comparison of their frequency in facial angiofibroma and other sample subgroups from TSC patients analyzed in this study. And in this plot, each dot corresponds to the number of somatic mutations identified per each of the analyzed lesions or samples. Based on this visualization, we can conclude that the frequency of somatic mutations was significantly higher in facial angiofibroma in comparison with other samples, including normal skin. We also did an analysis of TP53 gene using MHPA method and identified an average of 10 TP53 mutations per each whole skin biopsies from facial angiofibromas. And the number of the identified TP53 mutations correlated very well with TSC2 mutations in the same sample, what reflects the role of UV in the generation of both genes mutations. We are also interested in the investigation of a single nucleotide variant signature in TSC facial angiofibromas. And in this plot, we have compared The reference canonical mutation signature, SNV signature, observed previously in skin, which is marked with black bars. And this is compared to the SNV signature observed using our MHPA method. And this signature is marked with colorful bars with colors corresponding to the panels above the plot, representing different types of single nucleotide substitutions. The 3 nucleotide context from 5' and 3' sites is presented along the x-axis, and the y-axis corresponds to the fraction of different substitutions at variable sequence context. And based on this comparison, we can observe that these 2 profiles, the reference canonical UV-related profile and our profile observed by MHPA, are very similar. And dominated with C to T substitutions at variable sequence context, with moderate enrichment of C to A substitution, which occurred likely due to reactive oxygen species generated by sunlight. In the second step, we are also interested in DNV signatures in facial angiofibroma. And again, it is organized in a similar way where the x-axis corresponds to different types of dinucleotide substitutions with the fractions corresponding to each of those marked on the y-axis. And here, looking at the black bar, which is a reference signature in skin, and red bar, which is the signature observed in our study, we can see that they are highly similar with more than 90% of CC to TT substitutions, which are known to be related to UV radiation. And in this study, I have also identified a recurrent novel UV-related TP53 mutation pattern in skin. And there were a total of 18 complex adjacent indel and SNV or DNV that occurred in Cs on the same allele. And each of these events is presented as a separate row in this plot, where the location of single nucleotide or dinucleotide variant is presented in relation to the deletion. And 13 out of 18 events contained either C to T or CC to TT substitutions, which are known to be related to UV radiation. Next, we are also interested how the frequency compares looking at the TSC1 versus TSC2 gene in facial angiofibroma. And to investigate somatic mutations in TSC1 in FAF, I have developed a new MHPA assay which covers more than 90% of previously reported TSC1 mutations and all the mutation hotspots as visualized on this plot. And based on recent analysis of a total of 7 facial angiofibromas from patients with germline heterozygous TSC1 mutation or mosaic TSC1 mutation, it can be observed that UV mutations are much less common in TSC1 than in TSC2, which is consistent with the overall lower severity of FAF involvement in TSC patients with TSC1 mutations in comparison with patients with mutations in TSC2. In summary, in this study, we discovered that unique signatures of TSC2 UV-induced point mutations are highly prevalent in facial skin of TSC subjects, and the prevalence of somatic mutations in TSC facial skin likely represents multiple individual fibroblast clones, which we call microfafs, that were present in these tiny FAF biopsies. And we estimate that hundreds of thousands of independent clonal fibroblast proliferations occur due to the second hit TSC2 mutations in the skin of TSC patients, and only a small proportion of these precursor lesions eventually develops into clinically observable FAF lesions. And these observations highlight the importance of sunblock and other measures to limit facial UV exposures of TSC individuals of all ages. And I would like to thank all people involved in this study, my colleagues from lab in Boston, Krinia and Basia, our collaborator dermatologists, Johannes Greveling and Thomas Darling. And I would like to give special thanks to Dr. David Kwiatkowski, who was my mentor during my postdoc fellowship, and now one very close collaborator in the ongoing projects that I'm performing now here in the Institute of Bioorganic Chemistry. And this study was funded by TSC Alliance Postdoctoral Fellowship Award that I was awarded with during my stay in Boston, as well as Engels Family Fund. Thank you very much.
Thank you very much, Kasia. Because it's late, we have time for maybe one short question. Paweł Świtoński, but short question.
All right. Thanks for your talk. It was really inspiring. So do I understand correctly that TSC2 is a driver gene for... So based on different mutations that you observe, can you speculate about the mechanism on which these cancer cells grow and proliferate?
Yes, I think this is a very interesting question, and actually I think that I can refer to the comment of one of the reviewers of our paper that was published in JCI, and he was really intrigued with the amount or the proportion of the CC to TT substitutions that we observe to occur in TSC, because usually in skin we can observe 2 to 3% fraction of such substitutions, while here we observed up to 30% or even more in some samples. And he suggested that similar level of those dinucleotide substitutions occurs in Xeroderma pigmentosum, which is another skin disorder. And there we can observe the defective nucleotide excision repair mechanism and also defective translesion polymerase at activity. So he was intrigued if this may be also the case in TSC. So I think this is an interesting thing to explore further. And I think this could be our focus. Now we have many projects focused strictly on the genetic aspects, but some mechanistic study of this aspect could be some of interesting projects in the future, I think.
Okay, thank you again, Kasia, and thank you all the speakers and all the people who are here. And now we have coffee break.
Okay, let's start. Good morning, everyone. Welcome all of you on this session dedicated to synthetic life. And it's my great pleasure to introduce our first speaker, Professor Andrzej Dziembowski from the International Institute of Molecular and Cell Biology in Warsaw. The research of Professor Dziembowski lab focuses on post-transcriptional regulation of gene expression, and today we will have the opportunity to see how the results of his basic studies are important for modern medicine to generate modern mRNA drugs. So, Professor Dziembowski, the floor is yours.
Thank you very much for inviting me to this fantastic meeting. And today I will talk about mRNA therapeutics. It's actually the project which started slightly more than 2 years ago in the lab. And it is connected to the fact that first mRNA-based therapeutics were introduced, meaning the mRNA vaccines. So mRNA therapeutics are actually very similar to endogenous mRNAs, and we were studying mechanisms of mRNA degradation for many years. So we found it's a great opportunity to look actually how are degraded mRNA-based therapeutic mRNA vaccines. Because indeed, at least many people believe that mRNA therapeutics are a future of biological drugs, because they do not have to be used as a prophylactic vaccines, but they can also be used for replacement therapies when the target is liver, or, for example, expression of therapeutic antibodies. And at least a year ago, these two mRNA-based vaccines against the COVID were actually the best-selling drugs worldwide. But it is really astonishing how little we know about the metabolism of mRNA-based therapeutics. This is simply because this was very recently introduced, and actually our knowledge about mRNA degradation is based on mostly reporter mRNAs or some individual endogenous transcript. Also now and more we know about genome-wide degradation, but actually mRNA degradation of mRNA-based therapeutic was never really studied directly before. So just to tell a few words how they are built. So I don't know if I should, maybe I will use the pointer here. So they are very, very similar to the normal endogenous mRNA. They are just in vitro transcribed. This is actually the sequence of the anti-COVID vaccines. They have a cap. There are slightly two different types how the cap is made in case of Moderna and Pfizer vaccines. We have a poly tail and the body of the messenger. The main real difference between the endogenous and therapeutic mRNAs is the presence of methyl pseudouridine, which replaces the normal uridine. This replacement is to have a less unwanted innate immune response, because the status of uridine residues very potently induce innate immune response, which is very toxic, especially, for example, for macrophages. If you transfect macrophage with mRNA with a simple uridine instead of methyl pseudouridine, actually this will be the cell will most probably die. And mRNA-based therapeutics have to somehow enter the cell, so all of them are now packed by lipid nanoparticles. It's a chaotic mixing when we have a lipids, various types of the lipids, a very experimental strategy to check which actually LNP is the best for the transfection. So we chaotically mix several lipids, and then we have this kind of like nanoparticles composed of mRNA together with those lipids. And these lipid nanoparticles enter the cell by active process micropinocytosis. So it is, for example, this is not that every cell type will be transfected by lipid nanoparticle encapsulated mRNAs. And if you look for existing mRNA vaccines, there was one study based on single cell sequencing. The vaccines uptaken mainly by macrophages and dendritic cells. immune cells, for example, even after intramuscular injection, it actually does not really enter muscles, which have very weak properties to uptake any cargo through the active micropinocytotic process. So this is also a very important aspect when you think about mRNA-based therapeutics, that depending, for example, on the composition of this lipid, some different cell type may uptake this mRNA more or less efficiently, but actually people believe till now that after intramuscular delivery, either intermuscle immune cells uptake the vaccine and then they go into the draining lymph nodes, or simply the vaccine itself is also drained to the draining lymph nodes, and there it's uptaken by those immune cells. So, talking about mRNA degradation, so we know very well what is the pathway of mRNA degradation for endogenous transcript. So we have an mRNA with a cap and poly-A tail, and to degrade mRNA, you need to remove the poly-A tail. After the poly-A tail removal, there is also the cap removed, and then we have an active, fast process of mRNA degradation. And what people believe is that actually the speed of deadenylation, removal of the poly-A tail, by two different complexes, two different deadenylases, PAN2, PAN3, and CCR4 node, will dictate the stability of mRNA molecule. So, in the reality, not always poly-A tail is just shortened and then can be degraded. There are specific situations that also this deadenylation, removal of poly-A tail, can be counteracted by, for example, cytoplasmic polymerases. But those enzymes, in majority of cases, were rather studied in very peculiar, specific systems. It was never really considered in case of mRNA-based therapeutics. And what is important, also, that actually to study the poly-A tail is also a difficult task. I will go back to that later. So I will start majority of my talk will focus on one particular mRNA-based therapeutic. It is a Moderna. And I will actually focus on the poly-A tail of Moderna, so it would be a tail of Moderna tail. And there were two people from the lab which actually were the main responsible for this project. It was Severin Mroczek for experimental part, and also Paweł Krawczyk, who essentially performed all the bioinformatics connected to this project. So studying poly-A tails is a difficult task because every single polymerase stutters on homopolymeric track. So every PCR reaction which actually changed the length of the poly-A tail. Therefore, when there are new sequencing methodology introduced, which are single molecule methodologies based on nanopores, when there is a single-stranded nucleic acid is passing through the pore and you record the current, so you directly measure mRNA. And here's a single read of the nanopore-based direct RNA sequencing of the active mRNA, when you can see here the body of the messenger. Through the very sophisticated neural network-based algorithms, you can base call the sequence with not very high accuracy, and you can also localize the poly-A tail and very accurately measure the length of the poly-A tail. So this is a single read of single read of active mRNA, endogenous one, and this is the poly-A tail. You can actually study also very long mRNA. This is a single read of the murine coronavirus, entire single molecule read of the coronavirus. So with the methodology we're introducing in the lab to study normal endogenous mRNA to measure the length of the poly-A tail, we thought, why not look also on the mRNA therapeutics? We had this opportunity because we also got an access to mRNA-based therapeutics. The problem is that because this mRNA is actually modified with the metal pseudouridine, you cannot use the normal base calling software because the current after modification is different, so you cannot just read this sequence. Actually, to fish out vaccine reads, we also introduced some specific algorithm which is based on the comparing time series, dynamic time wrapping, and this way we can fish out the mRNA of the vaccine which is modified. So this is the single read of the Moderna mRNA vaccine. At the beginning, everything is typical. We have a body of the messenger, we have a poly-A tail, but actually we also realized that before the poly-A tail, at the exact 3' end, there is some additional perturbation of the current, and in the reality, Moderna vaccine ends on the pentameric non-A sequence. It was actually not described in the publication, mainly probably because the preclinical data were on some slightly different sequence and they would not get the allowance by the medical agencies. This was a very fortunate thing for us because now we can have an algorithm which will separate Moderna vaccine with a poly-A tail and poly-A tail with this additional sequence. And then you can measure the length of the poly-A tail and also for the unprocessed, non-modified vaccine, which have this additional sequence, and the poly-A tail. And the majority, of course, reads from the just pure vaccine contain this pentameric sequence because everything which does not contain this sequence is probably partially degraded. Well, then, with this methodology, we ask how mRNA-based therapeutics are degraded. So first we started to study that we administered vaccine to the simple cell line like HEC293 or I549, and in the time series look how the poly-A tail look like. So you can see that the length of the poly-A tail is shortening, and mRNA is quite swiftly degraded. Of course, both those cell types uptake the vaccine, produce the antigen, the spike protein. And as I told you before, what is the main enzyme which is responsible for deadenylation? It's a CCR4 complex. So if in HEC293 cells you deplete CCR4, you will slow down the deadenylation, and mRNA-based therapeutics will be then more stable. What is also interesting is that actually this pentameric non-A sequence is removed by some other enzyme, not really the deadenylating enzyme. So this was, I would say, rather expected. That's what we thought. We swiftly remove the poly-A tail, we degrade mRNA-based therapeutics. So then there is a question, what actually happens in vivo? So we administered the vaccine into the mice and isolated RNA from the injection sites in a time course. And what we can see actually, that 24 hours post-administration, you don't see just a deadenylation, there is something other happening because you can see that also poly-A tail can be extended, meaning that in vivo metabolism of mRNA-based therapeutic is really different than what we would expect from studying in a simple model cell lines. So then, of course, there is a question where actually this vaccine goes. And majority of people were always focusing on draining lymph nodes. So we isolated RNA from a draining lymph node and also directly from the injection site, and actually appears, at least for a Moderna vaccine, there is like 1,000-fold more this vaccine at the injection site than in a draining lymph node. So basically, this vaccine does not really efficiently enter the draining lymph node. This is a suggestion that everything really takes place locally. And then, if you saw the macrophages and dendritic cells, those two main cell types people believe are uptaking the vaccine, everyone was really focusing on the dendritic cells because those are the primary antigen-presenting cells. But if you sort these two cell types, it will appear that actually there is far more vaccine uptaken by macrophages rather than dendritic cells. So macrophages are primary cell type uptaking the vaccine. And what is also interesting, that this is a massive process. So actually there is more molecules of the vaccine mRNA per cell than vaccine, which means that there is probably 1,000 molecules per cell. It also shows how efficient this micropinocytotic uptake of lipid nanoparticle by this particular cell type. So by this we know which cell type uptake the vaccine, mainly macrophages, but also dendritic cells. You can also then employ in vitro model system for these two cell types. So maybe I will skip this. So when you administer vaccine to culture dendritic cells, either bone marrow derived or monocyte derived, you will see that actually the metabolism of vaccines there is quite similar to HEC293 cells. They are slightly more stable, but you will see the deadenylation, nothing like extension of poly-A tail. So this is not the cell type responsible for this potential elongation of poly-A tail we have seen in vivo. But if you administer vaccine to macrophages cultured in vitro, you can see the massive readenylation. So actually, from the 100 nucleotide long poly A tail, 24 hours after administration, we'll have nearly sometimes 200 nucleotide long poly A tail. That was absolutely not really considered by any people working on development of mRNA-based therapeutics. So, and this also can be recapitulated in human monocyte-derived macrophages. So, but possibly actually, if you put whatever type of RNA into macrophages, then they will readenylate the mRNA in the cytoplasm and stabilize. Possibly macrophages stabilize everything. But it's not really the case. So you can transfect macrophages with Moderna mRNA, you will see this massive polyadenylation. But if you put a free-fly luciferase reporter mRNA, it will be swiftly degraded. But there is something specific in the Moderna vaccine. So another question is actually, what is the enzyme responsible for this process? So we have the major polymerases are in the nucleus, but there are also several other enzymes present in the cytoplasm or in mitochondria, which also can have some functions like polymerases. But it's also important to know that administration of vaccine actually leads to a very strong transcriptional responses, because although this mRNA is transcribed with methyl pseudouridine, it's still recognized like a viral mRNA. And you have a very similar response to innate immune response after viral activation. So you activate a lot of transcript connected to antigen presentation, innate immune response. But among them, there is one particular cytoplasmic polypolymerase, this is 10-5A. And indeed, you can see that after administration of the vaccine, you will see overexpression of 10-5A polypolymerase. So this is the indication that possibly this is the enzyme which is responsible for the stabilization, at least extension of poly A tail of Moderna mRNA vaccine. So we got very much interested in that. And indeed 10-5A is also expressed in vivo after addition of vaccines. So for example, if you look on the single-cell RNA-seq from others which were looking for draining lymph nodes, but we also can see that 10-5A is expressed in macrophages, but not really in dendritic cells. This also can suggest that this is really the enzyme responsible for this process. So what is a 10-5A? So actually, that was a very fortunate for us that 10-5 is a family of cytoplasmic polymerases we have been studying for last 8 or 9 years. Therefore, we had a lot of models. We actually were studying those 10-5 in several different biological contexts. But 10-5s, to sum up, are enzymes which are present in the cytoplasm and are associated with endoplasmic reticulum. And this is important because also vaccine encodes spike protein, which have a leader to endoplasmic reticulum, and is then enters the endoplasmic reticulum, go to the cell membrane, and then is stripped off from the membrane. So 10-5A and 5C polyadenylate various different in vivo mRNAs which encode secreted proteins. So for example, 10-5C is expressed in B cells where it polyadenylates and extends poly tails of immunoglobulin mRNAs. If you don't have a 10-5C in B cells, then the immunoglobulin mRNAs have a shorter poly tails, then the stability of immunoglobulin mRNA is, they are less stable, and also there is some slightly defective humoral immune response. But if we talk about 10-5A, this is the enzyme, which is expressed in several different places, but there are patients with a mutation of 10-5A which have the congenital bone disease. There is a frequent bone fractures in the mutant of 10-5A. Normally, this disease is caused by mutations in collagens because actually bones are formed on the collagen chain. Collagens are, because of that, one of the most abundant proteins in nature, and the collagen chain is the place when the hydroxyapatite crystallizes and generates the bone. And all this collagen is produced by osteoblasts. And indeed, 10-5A is an enzyme which in the osteoblast is associated with endoplasmic reticulum and polyadenylate mRNA encoding collagen, fibril collagen proteins, but also other proteins essential for a bond formation. And as the result of that, there is a not proper structure of the collagen, which you can see in the KO mice, which recapitulate the disease I was talking about. And also, what is also important, actually more important for my talk today, is that the 10-5A and slightly also 10-5C are expressed in bone marrow-derived macrophages. We also know already that if you knock out 10-5A and C in macrophages, the length of the poly tails of several effector proteins involved in innate immune response have a shorter poly tail, they are less stable, and for example, there is less produced of the lysozyme or catapsins. Those are really effector protein essential for the function of the macrophages during the eating or clearing. So then, that was really our prime candidate for the enzyme responsible for a polyadenylation and potential stabilization of mRNA-based therapeutic. So, and it's also interesting that actually mRNA vaccine not only induce expression 10-5A, but also this induction leads also to extension of polyetanes of many mRNAs which we already have known are polyadenylated by 10-5A and C. So there is this relation that vaccine induced expression of this polymerase also changing the physiology of the or metabolism of mRNA. So what happens if you administer Moderna mRNA vaccine to the macrophages devoid of 10-5A and C? Then there is no cytoplasmic polyadenylation and mRNA vaccine is swiftly degraded and removed. And it really have a huge effect on the stability, so you can see that the stability of mRNA-based therapeutic here is much, so they are much less stable, and there is less antigen produced, at least in culture. So you can also show it on the human macrophages. If you simply do the siRNA-mediated depletion, you will also see that the non-treated by control human macrophages will readenylate mRNA therapeutics, but depletion of 10-5A will lead to the shortening of poly tail. You can also recapitulate that with the other model system. So this is transgenic mice with a rapid depletion system. So you have a tag on the 10-5A, you add the chemical, and the 10-5A is fastly degraded. You also will see that this will lead to the very fast deadenylation. So indeed, 10-5A is responsible for readenylation of Moderna mRNA vaccine and stabilization. So you can also ask another question, isn't the situation that actually 10-5A has some global effect of translation? So whatever mRNA is delivered to cells devoid of 10-5A, the expression will be lower. But if you deliver mRNA encoding GFP with various flavors to bone marrow-derived macrophages with or without 10-5A, there will be absolutely no effect on the expression and the stability. So there are these two undistinguishable curves of the expression in cells of GFP fluorescence. You will see that the mRNA have a stability of about 20 hours, and exactly the same stability and exactly the same expression with and without 10-5A. So indeed, there is this specificity to some specific mRNA like Moderna vaccine or some endogenous substrates on 10-5A. And also, really 10-5s are the only cytoplasmic poly or poly-U polymerases which affect the stability of mRNA-based therapy, at least Moderna vaccine, because if you deplete other enzymes, you will see no effect on readenylation or deadenylation. So indeed, this is this enzyme which affects the stability of mRNA, Moderna mRNA vaccine. So now there is an important question, does it matter? So we see all those in vitro models, we see that we can readenylate, stabilize mRNA-based therapeutic, but does it have any effect on real immune response? So we took a 10-5A KO mice and we immunized mice and look on the spike protein and also on the amount of anti-spike antibodies 2 weeks after immunization. As you will see, actually, for both Moderna and the Pfizer vaccine, that 1058 KO, some typo here. So spike protein expression declines faster in the knockout mice. So if you divide the amount of spike protein in the serum 4 and 2 days post-immunization, you will see that in the knockout, the longitude of the expression of an antigen is much shorter. Not really, the level is not as much changed like a longitude of the expression, but this indeed has an effect because in 1058 KO mice, there is far less antibodies against spike protein 2 weeks after the immunization. But, 1058 KO mice actually are rather thick also because of the problem with the bonds. So there is another possibility that there is some systemic problem with building new immune response. So we also immunized mice wild type and the knockout with a protein-based vaccine, the Novavax vaccine. And there's absolutely no problem. Actually, if something happened, there is more antibodies in the knockout rather than the wild type. So indeed, the function of 1058 is really specific to mRNA-based therapeutics and stabilize them at least after intermuscular delivery, where the macrophages are primary targets. So our results, I think, have a quite broad implication on different aspects of mRNA-based therapeutics. So first of all, we provided this absolutely unexpected basis for the efficacy. So we have shown that the metabolism of mRNA-based therapeutic is much different than people thought before, and in macrophages, they are very stable because of this readenylation, which was never really before studied or even thought. Also that cell type and the model really matters. So the delivered mRNA or therapeutics, prototype or reporter may be very differently metabolized in different cell types. So you have to take into consideration, for example, only specific cell types express 10-5 polymerases. They are not expressed in HEX cells, HeLa cells, or mast cells, or also not so much in the liver. So it also says something about the design and optimization of the sequence. There's also one interesting twist. In addition to BioNTech and Moderna, there was a third company, actually the first company producing mRNA-based therapeutic, the CureVac, which also tried to introduce anti-COVID vaccine. So their first prototype had a very peculiar structure. It actually was not ending on poly A tail, but after the poly A tail, there was some poly C sequence and histone stem loop. And this sequence for sure cannot be readenylated or polyadenylated by 10-5. Then they produced another prototype ending on poly A tail, and this was behaving better. Anyway, they lost the battle, but this was better, and we believe, although we don't have a direct proof on that this is all because this is a substrate for 10-5 mediated polyadenylation, and this is not. So now I wanted to tell you something about the outlook, actually, how this knowledge also can be used in some other of our projects. So one thing is, which really interests us, is that actually the metabolism of poly A tails really vary between different cell types and the tissues. One thing you can see, for example, that average length of the poly A tails is very, very different in different tissues. So you can have a pancreas which have extremely short poly A tails, about 30 nucleotides, and some like macrophages have relatively long. Also, depending actually what is the coded protein or localization of protein product, the distribution of the length of the poly A tail may be very different. Well, this is something which really is interesting for us, and also, for example, we would like to study those endogenous transcript, but also therapeutics in vivo, for example, focusing on the liver, which is also an interesting target. So in the liver, the poly A are quite short, but if you deplete the main deadenylation using this in vivo DTAC system, then you can stabilize a lot mRNA-based therapeutics and also report them. And actually, indeed, so if you have a vaccine after intermuscular injection, then there is this readenylation. In the liver, you have no readenylation, so the main determinants of mRNA stability is just the shortening of poly A tail. So here you can really have to think about different ways how to stabilize mRNA. It's already known that actually stability of mRNA therapeutics in the liver is much, much shorter. So they are much less stable than, for example, in other cell types. I will skip this. And another interesting outlook is actually 10.5, we really are very much interested in 10.5a protein. One of the reasons is that it is also a polymorphic protein. Actually, on the N-terminus, there are unfolded repeats on with the sequence GGDFG, which very well correlate with the human height. So you can have in humans between 2 and 7 repeats. Every additional repeat is on average 0.5 centimeter more. So this is somehow one of the determinants of human height. So we also generated the mice, which normally have 2 repeats, but now we have a mice with 7 repeats. So this is some model system we would like to study. We actually also know that 10.5a alone really influenced the size of the mice. So 10.5a KO, at least, especially young mice, are very, very small. So why it is so? Why it's correlating with height? Because 10.5a is expressed in a hypothalamus. And the hypothalamus really governs a lot of different hormonal regulation programs, but one of them is the regulation of the expression of growth hormone, releasing hormone, or vasopressin. There, 10.5a really readenylate this mRNAs encoding this protein hormones. There are huge difference in the length and the stability of those mRNAs in the hypothalamus, which open a lot of interesting potential studies because there are so many things which can be regulated by those various hormones. Another interesting aspect of poly-A metabolism is actually that what I was saying to you all is about pure poly-A. We just measure the length of the poly-A. But actually the poly-A is not always a pure tract of adenosine. Sometimes you have non-A residues in the poly-A, which actually are difficult to study. And the nanopolymerase RNA sequencing is actually very good to look on such a non-adenosine residues in poly-A, because if, for example, if you can in vitro transcribe RNA with specific nucleotides, you can see that they really affect the current. So Natalia Gumińska from the lab, she trained the neural network actually to identify non-adenosine residues in the poly-A. And this allowed us, this was a quite sophisticated training, actually the composition of and we gain a very, very high accuracy, especially with determining G, distinguishing G from adenosine. So, and that can be used for many different purposes, and also one of them is also connected to mRNA-based therapeutics. So it's interesting actually that the contamination of poly-A with GCs and U's with a synthetic mRNA will really vary about if you use different procedure to generate mRNA. So you can generate mRNA using encoded poly-A, and there we'll have a primary uridine as a contamination, or you can use polymerases, and then there will have a main contamination will be a cytosine, also depending a bit on the composition of the sequence. But we were more interested to looking on that actually what happened with the Moderna vaccine, especially as I told you at the beginning, Moderna is ending on this pentameric sequence. And this pentameric sequence usually removed before this readenylation. But it's not exactly the case, and actually this Moderna vaccine in vivo may have a lot of different non-adenosine residues, because actually 10.5a, if 10.5a is also not highly accurate enzyme, and quite often add non-adenosine residues, so this is another interesting subject of our further studies. We don't yet know really how it affects really the stability, these non-A residues here. So this is what I wanted to tell you. I just wanted to acknowledge a lot of people who really contributed to this project. This project really started slightly more than 2 years ago, and many people contributed from the lab. The very important contributors are our facility in the Institute Genome Engineering facility, which generated all the transgenic animals we use here, with the head Olga Gebartowska, and also our collaborators with this particular project, which are from the grant VIB and of course the funding agencies. So thank you very much.
Thank you. Thank you for your great and inspiring talk. So it's time for questions and comments. I see one over there.
Thanks, that was great presentation. Thank you. So I got two questions actually. First one would be, do you know what exactly drives specificity of 10.5a to the vaccine? Is it like a structure?
So we know that this is a localization. Sorry. So it is. We know that one of the primary determinants is the localization of mRNA. So we need to be targeted to endoplasmic reticulum. We don't see any real sequence specificity. But there are other specificities we do not yet understand.
Like a structure, for example. Can it be explained by the structure of mRNA?
I would say, nothing easy to identify using computational methods. So we know a lot of substrates of all those 10.5 polymerases in different contexts, with different, because they generate a lot of interesting phenotypes we were studying. And we've never been able to find out really easily definable sequence motif, easily definable. They have a slightly shorter UTRs than the rest of mRNAs, but really this needs further investigation, but there is nothing obvious.
All right, thanks. The other question would be, is there anything you can do with 10.5a to boost its activity and,
Yes, we think so, but it's too early to say.
All right, thank you.
Yes, very nice talk. Yes, it is interesting how such a product provided by company can be used also in scientific research, really providing some new observation. But I have completely different question. Have you tried to use some viral UTRs? Because in viruses, they don't have often poly-A tail. So, and they can stabilize RNA, and probably they are processed in a different way. And viruses are known as a very stable molecule. So maybe this is a way how we can specifically target RNA molecules, RNA drugs to specific type of cells.
So regarding delivery, yes, for sure. There are a lot of other ideas how to target those specific cell types. One is to manipulate with the lipid. Another is to target them specifically. Regarding viruses, there's also viruses in theory are a great source to do something like UTR without poly-A tail, but actually no one really succeeded to do that. It's really interesting how important is this poly-A tail. So histone stem loop is another mRNA without poly-A tail, but this one is maturating within the nucleus with some additional proteins bound, which then go to the cytoplasm. So it's not easy. No one, as far as I know, no one was really able to generate really good mRNA without the poly-A tail, unless it's a circular mRNA with the iris. Yes, that's another. We never tried that.
Thank you, very exciting research. So I have maybe a little bit naive question concerning this polymorphism in 510A. So is it any chance that it can be linked with the response from the patients? Like some people generate more antibodies and some not?
Let's say, so to be honest, so we really would like to see that. We actually, especially for this thing, we got an access to UK Biobank, which actually have this enormous amount of data, but the structural variants are not yet deposited, and we were not really, if you have to reanalyze yourself this 250,000 genomes, that's something we really didn't want it to go. But I agree with that. But to be honest, we also obviously immunize mice with 2 and 7 repeats, and we don't see statistically significant differences. But the immunization is a problem that there is such a high variation. So possibly in a human population, when you will look for this, Moderna vaccine was administered to 500 million people. Yes, so this is a huge probe, and I guess it should have some effect, but we didn't have an access, or we haven't. So for sure, we would like to know that.
Okay, so maybe another follow-up question with this polymerase. So what about this local overexpression after administration of the vaccine? Because as I understand, maybe I missed something, there is like overexpression of 10.5a in...
induction.
Overexpression
is
okay,
induction.
Sure. So can there be any side effect of that? Like maybe some other RNAs will be more stabilized or destabilized by this, and that can have some different local effects.
This is another interesting aspect. Not every mRNA you will deliver to macrophages will induce 10.5a. This is another variable which really change the thing. And you also have to know that this induction is connected to innate immune response. If you have a vaccine, that's fine. But actually, if you would like to have a replacement therapy in the liver, you don't want to have a permanent inflammation in the liver. So this is quite complex relation, and we haven't really gone deeply into the transcriptional regulation of 10.5a. That's all the things we would like to go in, but
Okay, thank you. Unfortunately, okay, short question, okay.
Hello, really exciting results. I just wanted to have a question. Did you try any rescue experiments for your 10.5a knockout mice? Did it repolyadenate the mRNA when you were just supplementing your mice with external 10.5?
So, not yet.
Okay, okay.
Okay, thank you again. Okay, let's move to our next speaker. It's Małgorzata Borowiak from Adam Mickiewicz University in Poznan. Her research interests include understanding the molecular basis of diabetes and development of cell-based therapy for this disorder. Małgorzata, the floor is yours.
Thank you very much. Good morning, everyone. Thank you to organizers for the invitation to present some of our results here today. And we're interested in pancreatic endocrine cells, especially beta cells, that are the main source of insulin in the human body, a hormone that we need to maintain a glucose homeostasis. And we're actually interested in two main aspects. So how are these cells formed and what goes wrong with it during diabetes? Actually, yesterday was an International Diabetes Day, and diabetes reached the epidemic status, but the main cause of the disease are really the beta cells. So in order to be able to study these cells, we just need a good source of human cells. And what seems to be a best option at the moment is to use the pluripotent stem cells as a starting point. And here, what you can see here, it's a very simplifying cartoon. Let me see how it works. Okay. Of a journey you have to undertake to generate human beta cells in vitro, where each of the steps of the journey is designed to mimic the key developmental events. We can navigate our cells along this journey using just a panel of markers, usually transcription factors. So when we embark this journey, the first stages, like generating definitive endoderm or early multipotent pancreatic progenitors, was actually quite straightforward. And soon, collectively as a scientific team, we figure out a way how to very efficiently, almost with 100% efficiency, coax stem cells into becoming these early progenitors. Later on, things get way more complicated than we would like to. And you can see this by application of numerous signaling pathways that have to be either activated or inhibited to push cells forward. And also, if you look at the single cell level, at these different populations that you are generating at different stages of this differentiation, you can see that they are quite heterogeneous. And as a result, in the end, you are not just generating simply a beta cells, but a mixture of beta, alpha, delta, enterochromaffin, or other pancreatic cell types. So when I started my lab, one of the first questions we asked was quite simple. So there are 5 different endocrine islet cell types in human pancreas, and they are all coming from one common progenitor that is marked by the expression of master transcriptional regulator neurogenin 3. So we ask when and how these cells that have to be all neurogenin 3 positive make a decision to become either alpha, beta, and so on. So we use a single-cell transcriptomic, and we sequence the murine pancreas at 2 time points where most of the neurogenin 3 positive cells are born. We actually did it before the 10x. This was made with the homemade drop sequencing, so it was quite a different play. And out of this spectrum of pancreatic cells, we first selected neurogenin 3 positive cells, and we look at them closely. Of course, we found the different subpopulations. That's another big surprise. They were molecularly distinct, but rather than reflecting future alpha, beta, and so on cells, these cells reflect their maturation, their development. So you can see this is if you apply pseudotime algorithms, you can see this is the earliest population. This one is a bit outlier, but then the last population will be simply the most mature one. And what we believe is that this EP1, they are corresponding to the cells that are just starting to upregulate neurogenin 3. They are still closely resembling the neighboring multipotent cells, and then these cells need to leave the epithelium and to surrounding microenvironment to form islets. And then in the last subgroup, you have cells that already starts to express genes associated with hormone processing. Okay, so we're quite interested in this one quite weird population, the EP2. Ones that they are distinct, 2, because they share a mixture of mesenchymal and epithelial markers. So in the next series of projects, actually, that's done by different people in the lab, we selected a number of genes that were specifically expressed in this EP2, and they were quite random genes. There were ECM proteins, there were adhesome members, there were like gluteum peroxidase involved in the redox balance or classical transcription factors. And we knocked them out one by one in human pluripotent stem cells, and then we studied their role during pancreatic differentiation. Sometimes we did a combination of different knockouts. So what I wanted to show you today is just a snap-off of some of the results, which is quite interesting, that no matter what we tackle, we actually end up in the same spot, that all of these genes at different level of organization in the cells are involved in the mechanosignaling, and as a result, they change the cell adhesion, the adhesome, but it all comes to the several signaling pathways that are being changed, and this has a profound effect on the cell fate decision. Okay. So here's an example where you have the AMOT2 deletion, and you can see that the expression of E-cadherin is downregulated, but it's also its distribution in the cell colony, where the E-cadherin, from being more or less uniformly expressed, now it's concentrated at the edges. This is associated with a number of other cytoskeleton changes, and if you look at the F-actin fibers, you can see that they are much thinner, they're also kind of more fuzzy and disorganized, and this leads to a change in the Hippo pathway status. So now the YAP, the main effector of Hippo pathway, moves from the cytoplasm, as it is in the Y-type cells, to the nucleus, and as a result, this allows the transcription of another set of YAP and TET target genes. So here's another example. You have completely different gene, the GPX2, regulator of the redox balance. So what happens if you knock it out in human stem cells and then subject these cells to the pancreatic differentiation? So when you look at the early beta cells of Y-type and GPX2 knockout, you can see that there is, with single cell transcriptomics, you can see that there are now new clusters appeared only in the knockout population that are not present in the control. And what is quite interesting that these new clusters, they correspond to the liver progenitor or multilineage progenitor, which is a mixture of liver and intestine or mesenchyme. So if you mess up with the redox balance, despite that you are very actively trying to push these cells to adopt a pancreatic fate, some of them will quite spontaneously move to completely different lineage. So that's quite an unexpected role of the redox during development. Okay, so coming back to this endocrine progenitors at 2 different time points. We knew there are 4 different subpopulations at early and the late time point, but we were wondering, are they the same? So when we mix up these cells, you could see that they don't mingle, they don't cluster together, they're quite separate. We were puzzled by this result, especially given that we have quite a different number of cell sequence. So we did a number of secondary assays where we purify neurogenin 3 positive endocrine progenitors at 2 different stages. And we did single cell qPCRs or single cell MATQ sequencing, which is another version of smart sequencing. So a way higher depth than the DROP-seq or 10x genomics. But all the results were kind of pointing to the same conclusion, that these cells are quite different. And if you look more carefully at them, you can see that the EPs that are born early, they have upregulation of the cohort of genes associated with the alpha cells, while the EPs that are born just 2 days later, they seem to be more associated with the beta cells. Then the ATAC sequencing kind of confirmed the same observation. So the EPs born later are more prone to generate beta cells, while the EPs born earlier, they are more alpha cells. So basically, for the EP, it matters when you are born in sense of whether they will end up being alpha or beta cells. So why? What else is changing? Why does time matter so much? And if you would look at the pancreatic development, you could see that during these 2 time points, there is a massive morphogenetic events going on, that basically everything that surrounds this epithelial cords of pancreatic progenitors is changing. So all the microenvironment around them is changing. So we wanted to look a bit more closely what's really changing in this microenvironment during this time. So what we could observe, which I'm not going to talk today, is that there are different subpopulations of mesenchyma. And what's quite interesting that this different subpopulations of mesenchyma, they have a very specific localization in the pancreas. And even if you dissociate pancreas and let it reassemble in vitro, they will find the right spot. So this would suggest that they have completely different roles. So there is a mesenchyma that preferentially will cluster along incoming nerves. There is a subtype of mesenchyme that will preferentially surround forming beta cells. So in completely independent project kind of happening in parallel, we actually went back to this paradigm that stem cell people are often telling you, that all these in vitro differentiation efforts are designed to mimic the development in a dish. But if you think about this a bit closer, it's not really the true, right? Because we are just so much focused on generating our cells of interest, in this case beta cells, that this usually happens at the expense of all the other cells that are present in the pancreas, right? So we only have the beta cells, progenitors of beta cells. That's what we want to have in a dish. Forget about all the mesenchymal, endothelia, and so on. And this approach in beta cell field is quite in opposition to what's, for example, is happening when you are using stem cells to generate intestinal cells, where they include the mesenchymal and endothelia and so on, other cell type population. So we look what will happen if we combine mesenchymal endothelial cells from human pancreas at different stages of development, assuming that this is changing, right? The mesenchymal endothelia are changing in the time, so the time matters. And as a control, we also use other endodermal markers. So the mixing the primary mesenchymal endothelial cells with the stem cells-derived pancreatic progenitors, we look what's the effect on the generation of beta cells, but also on the function of beta cells. Okay, so we're able to identify that there's two specific mesenchymal endothelial cells from very particular time points that seems to have a beneficial effect on induction of C-peptide positive cells. C-peptide is a byproduct of insulin synthesis, so basically insulin positive cells. And it's not only about making more cells, but by including this mesenchymal endothelial into differentiation regimen, you could also make these cells function. And you can see this by looking at the C-peptide insulin secretion in response to the increasing glucose. So you can see they secrete insulin only when it's needed, so when the glucose is high. Okay, but if you think about mixing primary human cells into your stem cell differentiation protocol, it's never going to be feasible in the long term. So we look about what is so special about this mesenchymal endothelial cells at these two different time points. And using transcriptomic and interactome analysis, we focus on the secreted proteins and ECM proteins that have the receptors expressed on the pancreatic progenitors. And we identified several of such pairs, and then one by one, we tested them in vitro using a recombinant protein. And the thing that was the most exciting was a Wnt5a that in dose-dependent manner induced the insulin-positive cells, even if you don't give them any extra inducers of cells, any other signaling pathways. It was quite interesting because I mentioned to you about these different subtypes of mesenchyme, and one of the subtypes that was always closely associated with the beta cells was the subtype that also produced a lot of Wnt5a. So what you can see here, these mesenchymal endothelial cells are expressing Wnt5a. When you mix it up with the stem cells-derived pancreatic progenitors, you spontaneously get a massive induction of insulin. And when you delete this Wnt5a gene from this cell line, then this effect is gone. This decrease in the insulin induction or the lack of the insulin induction can be partially rescued if the knockout cells you supplement with the recombinant Wnt5a, which we suggest that the Wnt5a has a main role here. How does it do it? We did a series of experiments and just showing you some examples. We think that two events have to happen. So first of all, or maybe I will just walk you through this. So this is a double reporter luciferase assay where we can measure the activity of two pathways. Here it will be the JNK pathway and the BMP in the same cell or cell population. So what happens is when you expose pancreatic cells to the Wnt5a, first you have an activation of JNK, and this has to be followed by the inhibition of BMP. And these coordinated two events are necessary for the efficient insulin induction. And you can see here some more data on the BMP inhibition, where you look at the activator of the BMP signaling, phosphosmad1,5, where this transcriptional factor is in the nucleus, where it can do its job, then there is no insulin expression. Where it remains in the cytoplasm, then you can see the cells will start to upregulate insulin. So this is just one of the examples, like how we can learn something more about the signals that are regulating the beta cells generation. And I would like to use the next couple minutes just to tell you a bit more about the kind of second branch of our efforts, which is focused on disease modeling. We are particularly interested in modeling of atypical diabetes. And what I mean by atypical, these are the patients that usually do not respond well to the conventional treatment. They often share a bit of features of type 1 and type 2 diabetes. They have frequent episodes of ketoacidosis that leads to the frequent hospitalization. So basically, they have a very severe beta cell dysfunction. And we started initially as a very small crowd, just with two clinicians, one at Baylor College Medicine in Houston, one at UCLA, where we identify a set of these patients and we biochemically characterize them carefully to divide them into 4 subgroups. And this division was based on the presence or absence of autoantibodies. So the patients that will have an autoantibodies against beta cells, which basically means that they are kind of like a type 1 diabetic patients where we have autoimmune destruction of beta cells, and then whether they have or don't have any beta cell mass left. So in this cohort of these patients, you could find those which would be like classical type 1 with autoantibodies present and no beta cells. There will be some that perhaps they are on the way of becoming type 1. But also it's a cohort of patients that is quite interesting. They don't have any autoimmunity signs, but they also don't have any beta cell reserve. And in this patient, there's an enrichment of monogenic forms of diabetes, and this was our first group that we are interested in. These efforts grew quite substantially, so up to date we have identified 1,100 of such patients. All of them has been biochemically characterized, and for 800 of them we have a genome sequencing. I'm just showing you one of the example, and of course we cannot do it for all the 800, but for a selected number of patients, we are generating iPSCs from them, we are subjecting them to our differentiation protocol to generate beta cells, which are then extensively characterized functionally, molecularly, depending on the needs of the project. And this is now kind of grew into the consortium, which is called Rare and Atypical Diabetes Network, which is sponsored by NIH. So one of these patients has a single nucleotide variant, heterozygous, which was in the 5' UTR of a gene, a transcription factor, PDX1, which is a known factor for beta cell development and function. This was a novel mutation, so we are quite interested. What's the effect of it? So when we differentiated the control and the patient iPSCs, you could see that we hardly could get any beta cells, despite all the different efforts, and we always observed the kind of differentiation block around the PP stage, so around the progenitor stage. And we are wondering why. And because this patient also has abnormal branching acids, amino acids, along with the transcriptomics, we also performed the metabolomics, and we observe a high content of leucine and its metabolites, suggesting a hyperleucine catabolism in the patient definitive endoderm, so the stage is just before this differentiation block. So we were wondering if this has something to do with the leucine concentration. So in the standard differentiation media, the leucine is 1 millimolar. And you can see here, you can nicely differentiate the control cells in this media, but not so much the patient-specific cells. If you lower the leucine concentration, you can get a partial rescue of this phenotype, which would suggest that this leucine cannot be normally metabolized by patient cells, and this leads to this kind of toxic effect. So we're wondering, is this something very specific to a patient, or is this like a general phenomenon? So when you take the Y-type cells, if you expose them to way higher leucine, 4 or 10 millimolar, you can also see that then leucine starts to be negative, leucine negative effect on beta cell differentiation. Okay, so what we also observe that this patient has an increased activity of mTOR PA3 kinase signaling. And what we could also see that if you treat the Y-type cells with this high leucine, you could also see the same signaling defect. So we corrected the mutation, and we could prove that this is the cause of this differentiation block. And we reintroduced the mutation into Y-type line, and again could prove that this is all about this mutation. And the question is why? So what we see is that the patient-specific cells, despite that they upregulate the PDX at the right time point, they always do it at the low expression, either if we look at the population or the single cell level. And what we kind of put together is this proposed mechanism, where you can see that this mutation happens to be within NFYI binding motif, and this mutation disrupts the binding of this transcriptional regulator, and if you downregulate this NFYB, you can recapitulate the same phenotype. So what we think it happened is that this patient has two unfortunate events. One of them was this higher susceptibility to leucine and its metabolites, and the second was this 5' UTR SNP. Okay, and with this I would like to thank all the people involved in the project, and I would like to spend a few last seconds on thanking my wonderful team of lab members that we created here. It's really a crowd, I think, exceptional, of very smart, very positive people who are putting their heart in the projects. And they're also very creative, and you can see an example of their creativity where we started. This was our first office. Thank you very much.
Thank you. Unfortunately, we have time for only one question. I can see. Yeah? Sure.
Hi, thanks for this. I might have missed it, but you've been talking about the impact of the mechanical stress potentially on the process of differentiation. But I did not notice if there are any mechanoreceptors or other things which you've identified in your process which could be the mediators of the effects which you talked about a little bit at the beginning of the talk.
Yes. So yeah, that's kind of a really ongoing work. So one of the things that we see change in the cells is, for example, piezo, which could be a mechanosensor. Yeah.
Okay, thank you. I encourage you to ask questions during our lunch break. So now we have to move to another speaker. Thank you. Okay, it's my great pleasure to introduce our next speaker, Professor Marcin Drąg from Department of Chemical Biology and Bioimaging, Wrocław University of Science and Technology. And his research interests include the design and synthesis of substrates, inhibitors, and activity-based probes to decipher the mechanism of action and the function of proteases in health and disease.
Okay, thank you. First, I would like, of course, thank organizers for inviting me for this great, let's say, all-discipline meeting, basically. And I wish you all the best on the 35th anniversary and many, many more. And today I will tell you on unnatural amino acids. So unnatural amino acids, this is the specialty of my lab. And I will tell you how actually they impacted the development of my lab, but also some of the drugs in recent time. So basically, we focus in my lab mainly on proteases. So we work on natural amino acids and we try to apply them for the proteases. So just to remind you what proteases are doing, they just cleave the peptide bond. But they are so specific that we have around 650 of them only in human body. And of course, many viruses, bacteria, and other living organisms, they also have them. So on what we focus on the active site of all the proteases. So basically, active sites are all these packets on the surface of the protease where amino acids bind. So here you have the examples of natural amino acids which binds to different sides of the packets, and it more or less looks like a lock and key, but there is much more conformational changes than in the lock and key model. So in principle, we came up a couple of years ago with Guy Salvesen that there is proteomics, and proteomics focus mainly on all the proteins expressed at the time. But not all the proteins are active at the same time. So we came up with the idea that on the top of the proteomics, there should be something what we call activomics. And I will tell you why activomics is changed, which didn't play actually any function. So in activome, you are actually focused only on the active protein, but if you want to investigate this, you need selective probes. So what are the selective probes? We developed something what is called activity-based probes. So activity comes from the name that it's actually interacting only with the active protein, and it consists three elements: warhead, linker, and tag. So warhead, this is the element which binds covalently to the active site of the only active enzyme. Then you have a linker, I will tell you more about this, and then detection tag. So how they work, they have, of course, different chemical entities here. So we build them in my lab in different ways, depending on kind of the protease what you're interested in. But how you use them? You use them exactly the same way as antibodies. So basically, you can do enzyme labeling, and then with the detection tag, you can detect them. So what is the advantage of the activity-based probes over the antibodies? They interact only with the active enzyme, and they are cell membrane permeable because they are small molecules. While the antibodies, they actually label all protein amount usually, and they are not cell membrane permeable. So basically, you cannot use them in the studies we want to use them. And then we, of course, focus on the substrate specificity to get these small molecules as selective as almost monoclonal antibodies. So it seems to quite impossible to have just very few amino acids and get them super selective, but I will tell you how we do it. So basically we apply unnatural amino acids. So what are the unnatural amino acids? Are all the other amino acids basically which are not natural, and we have 20 proteinogenic amino acids which builds protein. So here you have phenylalanine. If you cut by one methylene group, you have the unnatural phenylglycine. If you add one more, you have the homophenylalanine. And of course you can have different oxidation steps. And in total now in my lab we have around 200 unnatural amino acids, and we apply them in our different projects. But what is the most important, because I was working for many years in States on different types of the molecules which were supposed to be, for example, types of the drugs or delivery system, what actually is the problem, that very often if you build the molecule built of the natural amino acids, then you apply it to biological system, there is a problem of stability. So we're hoping that actually we can increase stability, and you can imagine basically this giraffe being built only from natural amino acids, and then the lions as proteases. So basically if the lions, they attack the giraffe, they will eat it without any problem, and this is something what we don't want to have. We want to have the system, może pan kliknąć na film? We want to have the system with unnatural amino acids like this. So you can imagine this is the molecule built of the unnatural amino acids and proteases jumping on it in the cell wherever, and then the molecule is still stable. So this was like the ultimate goal of applying of unnatural amino acids in our case. And then it's like we built the library which help us to actually select the best unnatural amino acids. So this is called the hybrid library, because we took all the knowledge building this library from Big Pharma. Because Big Pharma was using this type of combinatorial library for a long time, and then in their projects, but we added our technology on unnatural amino acids, so this is why we call it hybrid. And how does it look in the numbers? So if you use natural amino acids in this approach, you can have around 130,000 combinations because you cannot use the cysteine here. But if you use unnatural amino acids, we easily get around 300 million combinations. And with our recent libraries, we go up to 1 billion combinations. So you can imagine we have 1 billion combinations, and we can test it within 3-4 hours, and then tell you it's like what is the specificity. And we published the Nature protocols. I'm not going to go through all the details a couple of years ago about this. And we develop for all major the protease families this type of the approach by Paulina, Marcin, and Viola. So they were working on this approach. Then we use it in many papers and actually approaches for the bioimaging, for building the selective probes and building the selective molecules. And of course we published several papers, got a couple of awards. And at that time we were looking into the new projects. So we wanted to actually do something what is quite unusual, but I'm not going to tell you about this today. But we wanted to use these small molecules actually to tackle these big proteins. And then COVID started. So COVID started, and we had this technology, and we immediately knew that actually the SARS-2 contains proteases. So I had this luck or unluck working on the SARS-1 in States, and actually we had bought proteases at that time for testing. So we had actually at that time some experience with it, but actually I was hired as a postdoc to work on this disease, smallpox. So I had some actually experience working with the protease from smallpox, and then the American government asked us at that time if we can develop any test for the detection of smallpox, because this is non-existing disease, but of course many people are not vaccinated, and of course we know that there are samples around, and if they get released, we will have a huge problem actually with smallpox, because there are still no drugs for it. And Oops. Przestał działać. Nie działa. Nie, to jest taki normalny na ten, akademicki. A, bateria jest słaba. Okay, and then it's like, then working on these viral proteases, we decided to continue to work on the viral proteases, and this is Rolf Hilgenfeldt. So during the Zika pandemic, we decided to start collaboration with him. So Rolf actually helped a lot with the SARS-1, and to memorize this, the Singapore government built this sculpture. So this is the sculpture of the active site of the protease. And this protease is the EMPRO, main protease from the SARS-1 virus. So actually this is dedicated to Rolf Hilgenfeldt. And Rolf actually was coming very often to Wrocław before the pandemic, and he convinced us to build the library only for the viral proteases, which makes no sense, basically, to build the library for coronaviruses, or had no sense to build it before the pandemic, but he convinced me to build this type of the library. And actually, so this is the picture from the beginning of March 2020, just 3 days before the lockdown. So this is the conference organized every year on proteases in Alps. And on this conference, we decided actually who will do what with the proteases of coronavirus 2. So this is the meeting, and that was the first time I was coming back in mask or wearing the mask during the pandemic. But how COVID is related to proteases? And I think it's totally run by the proteases, because if you look, you have two proteases, which is EMPRO and PLPRO, which are coded into the coronavirus, but the receptor ACE2 is also protease. It's angiotensin converting enzyme. TMPRSS2, which cleaves the spike to make it more conformationally susceptible for the binding to ACE2, it's also a protease. And then you have two cathepsins, B and L, which some people think are important in the development of the COVID, but some people actually say that they are not that much. And how actually we knew that they are important? So we knew that they are important basically from the beginning. On January 10th, when Chinese, they published the genome, we looked with Rolf into the genome, and we found both of these proteases. So working on them for SARS-1, actually we had a lot of experience, and then immediately many groups contacted me if we have the library or we can compare basically these two proteases from coronavirus. And why they are important, and we knew that they are a medical target, because if you have the SARS entering the cell, releasing the RNA, and then you have the RNA translated into big polyprotein, then this big polyprotein needs to be chopped by the EMPRO and PLPRO, these two viral proteases. If you stop functioning of any of them, basically you completely stop the replication of the virus. So we knew that if we have any molecule which can inhibit either EMPRO or PLPRO, we have the antiviral drug. And that was our strategy, which actually we decided to work with my library long before the COVID. And then if you look for the types of drugs, and of course you can have three major types of drugs for the antiviral drugs. So you have the biological drugs and chemical drugs. So biological drugs are antibodies. Chemical drugs, you can divide into two groups, allosteric and active site-directed. Antibody and allosteric are totally useless for the coronaviruses, we knew it. Because if the virus mutates, actually, so these types of drugs, they interact with the surface of the virus. So any mutation can kill the drug, and then it's like any new variant can be totally not reactive with this type of drugs, but you can have active site-directed drugs. So what are the active site-directed drugs? So if you have the protease in the active site, it cannot mutate. Because if it mutates, it kills itself because it cannot process this big polyprotein. So we knew that actually both of the proteases should have the same substrate specificity from EMPRO from SARS-1 and SARS-2 and PLPRO from SARS-1 and SARS-2. So we actually decided to go this way. And so I'm going to tell you today only about the EMPRO. So EMPRO looks like a heart, and some people say it's also a heart of the coronavirus. So it is a dimer with this blue inhibitor bond here. And then it's like immediately we knew that this is important, and then Chinese government immediately asked Rolf Hilgenfeld and asked Rolf to talk to people with who collaborated if they have any molecules which can be used to treat the disease, and that was already in October 2019. And then Rolf went to China for a couple of months working there, and we're corresponding on how this virus develops. And then Rolf, on that January 10, when we had the genome, and then Rolf decided to actually get the protein, and he told me, I crystallize MPRO, and all the rest of the protease will go to your lab, and I will bring it in person, so we can do substrate specificity, and we can compare all these proteases. So how this library is unique. So this library was developed actually in 2018, 2019, and Rolf was supposed to come with the MPRO from SARS-1 and MPRO from MERS, from 2 coronaviruses before Christmas 2019. But at that time he was in China, so we were super lucky at that time because we had the library ready and we could use it immediately for the project. So we actually, Rolf came, we screened the whole library in actually 3 days with analysis of the data. We wrote the paper and we published this paper, and we decided not to patent it because we're like, okay, so this is the gift from my and Rolf Hilgenfeld lab, so maybe someone can use it to develop the new type of drug. And then we actually pointed which amino acids should be used in the drug structure to get really selective molecule and which can be translated into the drug. So that paper was one of the most downloaded at that time from bioRxiv, had more than 100,000 downloads within just the very first few weeks. And then people started criticizing us that actually you cannot use unnatural amino acids in the drug because it's toxic, it's immunogenic, and it's dangerous, and there are no drugs with unnatural amino acids. So I was getting lots of emails or comments like this. But here you have drugs, actually existing drugs, and accepted by FDA, which have unnatural amino acids. I don't have a pointer, but in each of these drugs you can find one amino acid, unnatural amino acids. So at that time there were already drugs with unnatural amino acids, but there was not existing drug or not accepted drug by FDA with all unnatural amino acids in the structure. So I'm going to tell you, so we decided to do this, the direct comparison of MPRO, and you can see this is the MPRO on top line. So this is the heat map. So more red, then the more amino acids it's preferred. And then you have the leucine natural amino acids, which is recognized in P2 position. Pretty much none of the other amino acids is accepted in this position. And then it's like, if you look into the Pfizer drug, which they developed for the SARS-1, this drug actually had the leucine in P2 position, so we knew that actually this library is super selective, and we can get really nice the view of all preferred amino acids in each position. So, but this drug didn't go into the clinical trials. Pfizer stopped it because this is the intravenous. So they didn't want to have the intravenous drug after all the stories with the remdesivir because they wanted to have a pill. So they wanted to actually totally change the structure of the drug to make it a pill and not the intravenous drug. And now we're going into the P3 position. So in P3 position, you can see that there are many amino acids recognized actually by both of the proteases, and there is no mutation. So you can see that all amino acids are recognized pretty much equally by both of the proteases, but the most recognized was D-tyrosine and then the third leucine. So we pointed out actually in the paper that if you want to develop the super selective drug, which will not interact with the human proteins, but will actually give you the selectivity and stability, you have to use the third leucine. And to our surprise, actually, 1 year later, Pfizer came up with the idea of this drug. So this is the Paxlovid and nirmatrelvir, and you can see actually maybe better here that they use the third leucine from our screening. So you can see that actually third leucine in P3 of the nirmatrelvir Paxlovid drug, it's exactly the same, the third leucine, which we pointed. And this is actually the first ever accepted by FDA drug built totally from unnatural amino acids. So Pfizer this way actually made us a great present because he confirmed actually that our technology works, and then actually that you can use unnatural amino acid in the structure and have totally selective and specific molecule. And then of course we published this paper, we published another preprint, and then actually, I will give you a couple of really interesting data from these papers and our recent papers. So, for example, on the individual substrate kinetics. So these substrates you can see here, so they are built totally from unnatural amino acids. We released them in the paper. 3 days later, a couple of companies were already selling these substrates without our permission or without asking the university for the permission. And they were telling us that they are helping to fight with the pandemic. But actually, you can see that third leucine was pretty much in all of this substrate giving the selectivity. So after quite a fight with a couple of the companies and lawyers, we commercialized these substrates, and they are sold by this Peptide Institute in Japan. And actually, the Wrocław University of Science and Technology signed a 10 years contract on selling them, and then they produce them. But also, if you want it, I have in the contract that they should send me for free to Poland, and I have several samples. So if you want this type of the substrate, I will give you for free. So just contact me, and we actually share the substrate for free with any group in Poland. And then one thing was that actually we wanted to prove, because people were telling that the virus doesn't exist. And then how you can prove that the virus exists? So we were like, what about we prove that the virus exists using our technology and using activity-based probes? So if the MPRO exists in the human cells, what about we develop the probe and we use this probe on the human samples, and maybe we can see the activity from the MPRO. So we designed this molecule, which is on the bottom with the body P, with the strong fluorophore, and of course with the unnatural amino acid sequence. We tested it on the recombinant enzyme. It labeled beautifully. So we actually decided to prove that it binds in the active site. Because talking to many groups at that time, they were telling us that this amino acid, they don't bind specifically. They were telling us that they bind in the allosteric way, that we actually don't know if this is the real selectivity. So Rolf immediately crystallized the activity-based probe, so we could prove that it binds beautifully in the active site of the MPRO. And then with the Professor Wojtek Młynarski from Łódź, who was working with the COVID patients at that time, we decided to do the final experiment. And as you can see here, we actually labeled, so top panel are the DAPI, of course, the nucleus, angiotensin converting enzymes. And then the green one, it's actually active MPRO inside of the human. So sample taken by the swab, dipped in the activity-based probe, and then we did the confocal microscopy imaging. So this way, actually, not only on one, but on several samples, we could prove that the virus exists, that the MPRO is quite actually robust, expressed into the cells. And the bottom panel, you can actually see that there is nothing, because this is the healthy patient without the COVID. So this was like 100% also confirmation for us that this approach works. And then we asked Johan Nates. Johan Nates is, I think, one of the best virologists, and he runs huge laboratory in Belgium. So he's my good friend and Rolf's good friend. And at that time, he was doing pretty much all the most important screenings for big pharma on the antivirus candidates. So he decided to test also our compound on the virus-infected cells. So at that time, we could see that actually the inhibitor we developed was of the potency of the remdesivir, but with the less cytotoxicity. So we continued these studies, and we also commercialized this inhibitor, so you can buy it, and of course for everyone in Poland it's for free. But the major question now is, if Paxlovid, which is the only antiviral drug, and we know it works, many of my friends actually took it, and then it kills the viral infection immediately almost. So if there is a hope with Paxlovid for the future coronaviruses. So we did screening actually of pretty much 31 different mutants of the MPRO from all the major of concern variants of the coronavirus. And in all of them, actually we see pretty much same substance specificity, which means that Paxlovid will work. But also what we did, we tested several other MPROs from other coronaviruses known to exist in humans, and actually the profile of the substrate specificity is very similar. So there is hope actually that if we have the next pandemic, or basically maybe next coronavirus, the Paxlovid will work with good efficiency. So before we develop vaccine, which will take a couple of months with the existing technology, So we can use Paxlovid for the treatment. So in summary, I think that unnatural amino acids are an answer for perfection in activity and selectivity for drug design. So we have the first drug built of unnatural amino acids. It's FDA accepted. It actually helped to kill one of the most dramatic disease in the history in the last years. So, and then we need this type of drugs for the coronaviruses because, as I already mentioned, vaccine will be not available immediately. And then, so this is the queen of the project, is Viola. So she did all the studies pretty much by herself, working for 24 hours per day for several weeks, actually, sleeping in the lab and working on this project. So we had the permission for her to work on the time. But she actually scored 4 first author papers in 1 year in top journals, actually, for this work. And of course, many other groups from Poland, from Germany, from States, from Belgium, and of course, our sponsors. And this year, actually, in June, I was invited to Singapore with this type of the lecture, and then they let me to go and take a picture inside of this, the emperor protease. So thank you very much for your attention.
Thank you. Thank you for your great talk. And questions, comments?
Hello, thanks for a great talk. So I have a question regarding the structure of MPRO protease and this probe. Do you know what concentrations were used to get this complex? Was it somewhere near the Km value, or were the crystals saturated with the probe?
Oh, you actually sort of saturated the complex to get the crystal structure, of course, with this probe. But you don't need very high concentrations of this probe to work. We use actually quite low concentrations, less than 1 micromolar, actually, which is quite standard for the application of this probe for this type of the studies.
Okay, thank you.
Thank you. Do you think there's a chance for using similar approach for fighting other viruses?
Oh yeah. Of course we use it now. So yeah, we in my lab now, I think we have the collection pretty much of all major proteases you can imagine from viruses. So we actually, that was like a side project always for Viola to work on these proteases, on viral proteases. But then after pandemic, actually pretty much all groups from the world, all scientific and also from pharma contacted us. So we have actually many viral proteases, and of course this approach can be also translated to bacteria, so to develop new types of the antibiotics. Because the problem always was that bacteria and viruses, no one wanted to have the small molecule built of the natural or unnatural amino acids as an antiviral drug. So Pfizer did us a great actually job on developing this molecule and then getting it accepted by FDA. But now you can imagine that you can develop any type of these molecules for, actually we work now on viruses, bacteria and parasites also. And then this is quite quick therapy, it's just a few days, so you can use it without any problem.
I have just a question about side effect, because you said that unnatural amino acids may be toxic. So
yeah.
Probably they made some research on animals, yeah? What about side effects?
Yeah, I mean, so the answer always is if you take for example, I don't know, any type of drug which is not related to protein or whatever you can find. So for example, if you take aspirin, it's also unnatural. So that is the answer always. So it's all about how it's getting metabolized, and unnatural amino acids are quite quickly released. So they give you higher stability of the molecule comparing to natural amino acids, but there is no problem with the toxicity. So that was more like a gossip or that someone didn't test it, but actually we know that they are quite safe. And you can use, so it's 300, 500 milligrams dose, one shot actually, of the Paxlovid you take, the nirmatrelvir, the active molecule. So it's quite actually high concentration, no side effects.
Okay, thank you.
Yeah, thank you.
Join me and thank our speaker. So now it's time to move to our selected talks. Today we'll have 2 selected talks from abstracts. Our first speaker is Katarzyna Tutak from Adam Mickiewicz University, and she will tell us about RAND translation and possible therapeutics.
Sorry. So there's no pointer in a way from this. So this is the one. Oh, sorry. Oh, can you see that? Oh, yeah. Yeah. So it's harder. Okay, so it's quite hard, but okay. So thank you for the invitation, and I represent Professor Sobczak lab from Adam Mickiewicz University, and today I would like to tell you about a newly characterized modifier of RAND translation of RNA harboring expanded CGG repeats in Fragile X-associated syndrome. I still struggle. Okay. So in our lab, we study molecular basis of Fragile X-associated tremor ataxia syndrome, and it's a pretty hard name, so we use a short form FAXTAS. It's a neurodegenerative disease, a late onset one, which affects mostly males. So what underlines this disease? It's a mutation in 5' UTR region of FMR1 gene, and the mutation consists of a repetition of a CGG triplet, and here the expansion varies between 55 to 200 repeats in that region. So the prevalence in the population among males is pretty high because it's estimated 1 in 4 or 850 males. But it has a relatively low penetrance because it's estimated that 1 to 5,000 to 10,000 males will actually develop the disease. So the mutation has several molecular consequences for the cell. First of all, it leads to the transcription of a toxic RNA that forms a hairpin structure within those expanded CGG repeats. So such RNA has a tendency to aggregate and is toxic. Also, it sequesters proteins leading to functional depletion. As an example, I can use a SAM68 protein as a splicing factor. So these toxic RNAs are sequestering SAM68, leading to spliceopathy, which is observed in FAXTAS patients. Also, this RNA serves as a template for a main product of the gene, which is FMRP protein. And this protein is responsible for proper neuronal function. But next to the canonical event of translation, this toxic RNA also serves as a template for non-canonical protein synthesis, which is called repeats-associated non-AUG translation. And that leads to the production of a toxic protein, which is called FMR poly-G. It's a polyglycine, glycine-rich protein that stems from expanded CGG repeats. Polyglycine has a tendency to aggregate and also form inclusions in the nucleus. Okay, so what I would like to highlight here that toxicity of polyglycine is a hallmark of the disease, and as you can see here on the picture, the aggregates of polyglycine are present in the samples from the brain tissues, and it's believed that it's highly pathogenic and contributes to disease progression. From a molecular perspective, RAND translation event utilizes a canonical cap scanning mechanism. So it's similar to canonical translation, but it does not start at the canonical start codon AUG. It is initiated in non-canonical start codons such as ACG or GUG codon, and that leads to the production of so-called RAND proteins. There are multiple proteins produced by RAND translation in FAXTAS. However, polyglycine is the most predominant and the most toxic one. Despite the fact that RAND translation was discovered more than a decade ago, we still don't know much about how this process is governed. So the main aim of our project was to take a closer insight into mechanistic component of this process, and we wanted to identify novel factors that are related to CGG-related RAND translation regulation using mass spectrometry-based approach. So in our research, we adapted well-known technique, which is called MS2 biotrap. And these techniques allow to capture RNA binding proteins that are natively binding to the RNA of interest. In our case, that was RNA with expanded CHG repeats and RNA that mimics the FACSAS condition. So we developed a pool of two constructs. First, it's RNA with expanded CHG repeats. It's in the full context of 5' UTR of FMR1 gene with so-called stem lobe aptamers that allow the procedure to work, basically. And as a control construct, we use guanosine and cytosine-rich RNA that mimics the load of GC nucleotides in investigated RNA. Also, it's important because we know that many RNA binding proteins have a tendency to bind to GC-rich transcripts, so later on in analysis, we wanted to be able to cut these proteins off. So if it comes to a comparative analysis of mass spectrometry data we obtain when we compare the two sample groups and we apply pretty stringent data stratification criteria, such as discarding all the proteins that are present in negative control, so GC-rich, and focusing only on those proteins which were present in free triplicates, we ended up with 16 proteins that with high probability we can say that they bind specifically to 5' UTR of FMR1 with expanded CHG repeats. Among those proteins, we decided to study further so-called alternative 40S small ribosomal subunit, RPS26, in the context of its potential run translation regulatory properties. Using orthogonal technique, we also confirmed its direct interaction with RNA with expanded CHG repeats. So a few words about RPS26, why we thought it's actually an interesting candidate. So first of all, I need to highlight that it's so-called alternative ribosomal subunit. So it means that the ribosome can easily go without it. It's not a core component of the ribosome. Also, RPS26 is localized next to mRNA E site of actively translating ribosome. Also, what's very important, RPS26 has a specificity to recognize upstream sequences, more or less 10 base per upstream of start codon. And also we know from the literature that RPS26 dictates the translation of selected pool of proteins, so we can say there is RPS26 dependent or independent translation, and we hear some examples, like upon silencing of RPS26, some proteins are down or up regulated. So this all led us to form a hypothesis that maybe RPS26 can act as sequence-specific run translation initiation modifier. So to test that hypothesis, we actually developed a model in our lab to express the polyglycine protein in a stable way, taking advantage of so-called flipping T-rex system. So in that model, we wanted to express in the cells expanded CGG repeats and also control cell line with normal repeats range. And in that case, we wanted to use also the full length of 5' UTR of FMR1 when polyglycine was initiated from ACG, a run translation specific codon, and the polyglycine was tagged with GFP. And also we wanted to verify the presence and the effect of RPS26 by Western blotting. So, using basic siRNA silencing of RPS26 in these two models, we show that upon sufficient silencing of that factor, we observe a decline of polyglycine, both derived from longer and the shorter repeats. So later on, we wanted to ask the question, what is the effect here? So does it stem from mRNA, so from transcriptional regulation, or maybe it's more translational regulation? So based on qPCR, we wanted to check what's the level of FMR1 upon silencing of RPS26, and it seemed that there was no effect on mRNA, therefore it suggested more translational regulation here. Okay, so we got an effect in cells. We saw the polyglycine was declined, that's nice, but what about the run translation related toxicity? So as I mentioned at the beginning of the presentation, that polyglycine form aggregates. So we wanted to check whether the aggregates might be formed less efficiently or they might be smaller when we silence RPS26. And indeed, actually, what we observed in the experiment that silencing of RPS26 resulted in a decline in the number of the aggregates. Also, what we saw that it slows down the apoptosis process evoked by run translation. So it's also a very nice, promising result. We wanted also to take more mechanistic insight of how RPS26 might regulate the run translation event, and we know from the yeast model that RPS26 has its own private chaperone protein, it's TSR2 and S-cortin. And we know that, this TSR2 protein, first of all, binds to RPS26, stabilizes its level in the cell, so balancing the level of RPS26, but also it's involved in incorporation into maturating 40S ribosome. So TSR2 binds to RPS26 and drags it into forming 40S ribosome. So we hypothesized what might happen when we silence TSR2, whether we will see the same effect or not. So when we silenced TSR2, we observed a decline of RPS26, which was not a surprise, but it was still very sufficient to significantly decline polyglycine. But here, as I mentioned at the beginning of the talk, also the same toxic transcript is a template for the formation of FMRP proteins, so the protein which is good, let's say, not toxic one. And we see that when we silence TSR2, that silences RPS26, there's no effect on FMRP. So it's very important because in a way we silence a toxic protein, but the proper protein is not affected. Okay, so later on we wanted to dig that deeper and ask more global questions. So how RPS26 depletion might affect the synthesis of other proteins? So what will happen with translation in general, and what proteins might be changed despite polyglycine? So to answer that question, we actually applied the SciLag experiment. Sorry, I still struggle with this pointer. So we applied the SciLag experiment, and for those of you who are not familiar with the technique, it's stable isotope labeling with amino acids in cell culture, and basically it's a method that allows to label amino acids and then compare it to non-labeled amino acids from a control group, and mix the lysates and analyze them by mass spectrometry, and then we get the information how certain protein was changed upon a given treatment. So in our experiment, we just basically silenced RPS26 as an investigated sample and compared it to the SI control and checked what proteins were changed. And having the results from this experiment, actually, they were not really surprising because it seems that RPS26-dependent translation affected very selected pool of proteins. So when we apply the cutoff of 50% up or down regulation of the given protein expression, we saw that roughly 1% of the proteins were actually changed. So specificity of the translation regulated by RPS26 is very high. Even though that we got very little proteins that were downregulated, we still were able to validate those results by Western blotting, by ILF3, for example, or RPS6. So we got the same results here. And here, what's the most important from that slide and from that chunk of the result is that both in SciLag data, but also on the Western blot, we did not see the effect of FMRP. Okay, so here I would like to sum up the presentation. So in our research, we conducted mass spectrometry-based screening that allows to identify the new pool of proteins that would natively bind to FMR1 with expanded CGG repeats. And among those proteins, we focused on RPS26 that seems to regulate run translation without no effect on toxic mRNA. And we dig that deeper, and we also show that silencing the chaperone TSR2 also reduces the level of RPS26 down-regulating polyglycine without no effect on FMRP. And globally, RPS depletion affects very selected pool of proteins. And a take-home message here is that ribosomal composition seems to play a role in CGG-related run translation. So that's it from myself. I would like to acknowledge the people who were involved in the project, especially Anya, Isa, and of course, brilliant Professor Sobczak. So thank you for your attention, and I'm happy to take questions.
Thank you very much. Okay, are there any questions, comments? I don't see. Okay, maybe I'll start. Do you think that your results of these basic studies are very interesting, but maybe they can help to design some therapies, yes? Because RAND translation is a common factor, common process in many neurodegenerative disorders, yeah?
Yes, sure. So first of all, I can't answer with full confidence because we didn't go into even animal model or something like that. But basically, we can either inhibit or degrade by RPS26 in the cell by small molecules. That's one approach. Or second, we are thinking about, for example, blocking the interaction site in toxic mRNA, so RPS26 cannot bind to it. And also in our lab, we developed antinucleotide assays, that binds to this toxic RNA, and we know it's effective and it blocks RAND translation and also the binding with other proteins that might be involved in the regulation of that process. So definitely it has a therapeutic potential, especially that we show that there's no effect on FMRP and also it's selective, so there is a potential.
Yeah, the selectivity is crucial here, yes?
Yes.
Okay, thank you. Okay, so thank you again. Thank you very much. So it's time for our last talk, that will be given by Emilia Iwoska from Department of Organic Chemistry of the University of Gdańsk. She will tell us more about self-assembly peptides.
Thank you very much. I would like to thank you organizers for the invitation. And as you can see, I would like to tell you something about the self-assembly peptides as a novel approach in antimicrobial and anti-cancer research. In the start of my presentation, I would like to tell you something about nanomedicine, nanoparticles, nanotechnology, because nanotechnology has been widely used in nanomedicine. Because we can use this technology in target therapy, gene delivery system, diagnostic tests, biomarker mapping, and the most important for me as a drug delivery system, because nanoparticles, nanomaterials have a few advantages, very important advantages. The first one is high potential of the modification. As you can see, I'm here. No? Oh, okay. As you can see, we can modify nanomaterials by fluorescence, small drugs, and other peptides. So, in my opinion, this is potential of nanomaterials. They also warranted effective targeting, controlling drug release, prolongated half-life, and in some cases, reduce systemic toxicity. If we're talking about nanomaterials, we must know what is nanomaterials. So, in generally, there are particles from 1 to 100 nanometers, but in details we can classify in many areas. In example, we can classify it based on dimensionality, depending on the origin, based on chemical composition, and based on the phase. For me, I would like to focus today on the first one classification, because, in my opinion, this is an area of the nanoparticle as drug delivery system, especially nanofibers or self-assembly structures. You can ask me why I decide of this topic, because I would like to tell you the answer in whole my presentation. I was focused on natural nanoparticles It's mean fibrils, exactly peptide fibrils. They are good nanomaterials because they have big potential. They adopt the beta structure. They are built from natural amino acids. They are highly organized, very stable. They have strength similar to the steel. They can be easy to modify. They can be used as carriers for another active substance. They have natural tendency to the self-association, and we can show these nanoparticles using microscopy techniques. The most important, in my opinion, is two of these things, because the one of them is highly organized, and the second is easy to modify. Because when we're talking about the peptide fibrils, we're talking about short sequence that can adopt a very stable beta structure. So we can add to them another peptide sequence. We can add to another small drugs. And this is the potential as an anti-cancer and antimicrobial research. In my project, I was focused on the novel self-assembly peptides with two activities. One of them is antimicrobial, and the second one is anti-cancer activity. So I decided to design new peptides, new sequence, combined with two different sequences with two different, completely different activity. One of them, blue, is activity to fibril formation, and it should be yellow. It's a sequence with biology activity. In example, anti-cancer, like I say, and antimicrobial activity. I divided my project into three parts. The first one was studies about nanofibrils. The second one about the active sequence, especially about antimicrobial peptides sequence. And the last one part was combine both of them, the new nanodrug with biological activity and self-assembly properties. For all of these steps, I do the same things, the same stages. So I design and synthesize peptide using MOC strategy, characterization of peptides using mass spectrometry and HPLC. I do fibrillization studies, identify the fibrils using atomic force microscopy, transmission electron microscopy, and thioflavin TSA. For all of them, I do stability tests, antibacterial tests, cytotoxicity assay, and in the future, I would like to membrane permeability assay. So today I would like to talking about only few peptides, exactly about 6. 4 of them should be fibrillogenic potential. The short name of this fibril is in the table. For all of the tests, like I say, I synthesized, characterized using 2 specific techniques, HPLC and mass spectrometry. After I do fibrillization process, during the incubation by 7 days in PBS buffer, pH 7.4, at 37 degrees. And after I identify the fibrils using transmission electron microscopy and atomic force microscopy. On the first picture, you can see very small fibrils of one of these peptides, and after they are observed after this immediately after dissolution. But after 7 days of the incubation, we observe really big fibers. So the best candidates for me for the next step of the studies was a peptide, namely 1K. And the second, I go to the next part. I would try to check short fragment of only one human catalytidine, because this peptide is really really important, because they on the one side they adopt on the one side they adopt alpha helical conformation. This is typical antimicrobial peptides because it is a cationic peptide, and this peptide is important in many areas of the medicine. It's mean about wound healing, wound closure, stimulate the hemotaxis, and on the other side, it has a really important role in breast cancer. For this peptide, of course, I checked antimicrobial activity. So the short fragment of this peptide has really nice antimicrobial activity. So I decided to combine one key peptides and this short fragment of the catalysidine and synthesize the combined peptides. For these combined peptides, I do, of course, on the one side, fibrillogenic studies, the same like in the short sequence. Identification using THTSA, transmission electron microscopy, and another spectral microscopic techniques. For all of them, I do in vitro antimicrobial studies against Escherichia coli and Staphylococcus aureus. And the important thing was stability, these peptides. So I do a stability test in human plasma. Why stability is important? Because antimicrobial peptides are not stable in humans. They degrade in few minutes to 1 to 2 hours. So this is a big problem of this peptide. So when I combine this peptide with fibrils, I expect that the stability of this combined peptides will be higher. On this slide, I presented a few results of my research. On the first graph, you can see thioflavin TSA, and as you can see, on the black line, we observed fluorescence about fibrillogenic peptides. And combined peptides named 3K has really nice fluorescence of THT, but the level of the basic of the thioflavin T is really low. In the stability test, as you can see, we observe really degradation of what? No. we observed degradation of the catalytidine peptides after 2 hours. But when we combine this peptide with fibrillogenic sequence, the stability of this peptide is really nice. Combined peptides form fibrils too, as you can see on the TEM micrographs. The new fibrils are not big like in the short sequence, but the fibrils is presented. And the last part of my studies was antimicrobial activity against on the top Escherichia coli and down against Staphylococcus aureus. And you can see the level of the concentration and the activity is almost the same when we used alone antimicrobial peptides, namely and the new hybrid peptide. On the summary, I would like to tell you that the best candidates for my research for the fibril sequence is 1K peptide. The short fragment of catalytidine, of course, have antimicrobial activity, and the combination of these two sequences, fibrillogenic sequence and antimicrobial sequence, short antimicrobial sequence, give us a new peptide. These peptides are more stable, it less fibrillogenic tendency, but still high, and show similar activity. On the end, I would like to thank you, my group, and you for your attention.
Thank you, Emilia. Are there any questions, comments?
I know that maybe it's not your purpose, but have you considered delivering just mRNA for expressing this peptide?
No, it's not my area.
Maybe in the future, yes?
Maybe, yes.
Okay. Okay, since I can't see any questions and comments, so with this I would like to thank all the speakers of this session and enjoy your lunch. Thank you.
Shimadzu's latest imaging mass spectrometer.
Good afternoon, everyone. I think it's time to start the last session of this conference. So at the end of the session, we will thank all the speakers, but now I would like to take the opportunity to thank the audience, because you stayed with us to attend the last session, so we really appreciate this. So thank you very much. My name is Tomasz Piątek. I'm from Poznań Supercomputing and Networking Center, where I lead the application division. This session will be a little bit different from the previous ones, because we are not going to focus on only one single level of complexity of life. During this session, we are going by examples to show you how all these layers, scientific working on research on all these layers, can take advantage of modern software and hardware IT solutions just to improve, extend their research, to make it more accurate, to do it more efficient. So I hope we will find the session interesting for you, because as it was mentioned many times here, the IT solutions are, I would say, are necessary. You cannot agree, so we can make some kind of compromise that are just help the scientists to achieve a higher level of scientific excellence. So let me introduce the first speaker of the session, who is Rosen Apostolov. So Rosen is a researcher at PDC Center for High Performance Computing at KTH, Royal Institute of Technology in Stockholm, and also director of BioExer. He came here with a keynote on HPC for life science research, advanced application for extreme scale biomolecular simulation. I'm a HPC person, so I know Rosen mostly from his contribution to the GROMACS software, very popular and sophisticated software package to perform molecular dynamics. But of course, his research area and expertise is much broader, so we will get to know it in a few seconds. So Rosen, the floor is yours.
Good afternoon, everyone. Thank you for the kind invitation to attend this meeting, and congratulations on the anniversary of the Institute. This is a major milestone, and I wish success of your research work. We've seen a lot of great presentations today. A very broad spectrum of domains were covered, and as Tomek said, we'll now focus more on the IT computational side of science. My name is Rosen Apostolov. I work at the Royal Institute of Technology, KTH, in Stockholm, in our supercomputing center, PDC. I've been working with molecular dynamics simulation software for over 15 years now, and in the last 8 years, I've been leading a center of excellence, BioExcel, where we developed some of the most widely used applications for biomolecular modeling and simulations. And I will give you a bit of an overview of the work we do there, and maybe some of these applications might be useful for you in your work. IT technologies have developed so much that they are no more used as a proof of concept, but they are a great complementary tool. So even if your work is completely experimental in its nature, all these new methodologies, not only of modeling simulations, but also big data analytics, AI, can be of tremendous help. So I think that we are at a point where experiments theory and modeling and simulations are working really great together. So for several decades now, across the world, countries have been investing heavily in the development of supercomputers, and Europe is putting a lot of effort in this direction as well. EuroHPC joint undertaking is, for those of you who don't know what it is an organization that was established several years ago to drive forward across Europe the development in the area of high performance computing. And there are already a number of supercomputers deployed in different countries across the continent. These machines have millions of cores. They have tremendous power. Also, very soon, Poznan will be also receiving one of these machines for quantum computing next year or 25. So researchers now have access to this tremendous compute power, but how does it translate? What's the use for it for, in our case, for life science research? Well, we can show that for all these different scales of modeling of life science systems and all these domains, computing is fundamental to the research work there. Starting from simulations of small molecules, macromolecular complexes, cells, up to the organ, individuals level, and also the population. In all of these areas, computing is adding a lot. But how about HPC? What is the why do we need HPC for life science research? Here I'm giving a few examples of the domains where we are working on biomolecular simulations. For example, if you want to understand in depth the structure and the function of all the proteins that are the building blocks of our bodies, there are thousands and thousands of those, and all the interactions there that you could look into. You can model this straightforward on big supercomputers, but you need enormous amount of compute power to do that. Similarly, is this showing? Yep. So another application area is if you would like to do large-scale screening of potential drug candidates. This is also something that methodologies are being developed and is readily done. We are reaching major milestones, such as there is work on modeling of a whole cell in near atomistic resolution. So all of these bacteria and viruses, we are able now to very soon to model them in almost atomistic details. So it's not changing. Oops, sorry, oops. Went too far. Okay. So, in addition to all the hardware infrastructure that Europe has invested heavily in it, there was understanding of the need that we also need to build the software and the expertise for that, which is why at the moment there are 19, there's 2 more that were still approved, but there's like 20 centers of excellence for high-performance computing applications. And those centers of excellence cover all different domains of science, including life sciences. One of them is BioXcel, the one that I will focus on in this talk. It's a center which we established in 2015 with 3 main goals: to develop core important applications for life science communities, increase the expertise of the communities, provide training and support, and also strengthen the community. I will now present you several of the applications that we are developing, and maybe in some ways you can find them that they might be useful in your research as well. So the first one is called GROMACS, that Tomasz is also familiar with. It's one of the most popular applications for molecular dynamic simulations, and it's able to show petascale, sustained petascale performance on large supercomputers. It's able to efficiently use all modern types of CPUs or accelerators, special cards, and it's readily available on all supercomputing systems. It's a classical molecular dynamic simulation code, which means we model the interactions between atoms using Newtonian dynamics. There's a lot of algorithms that are implemented in it, and you can do all different kinds of fancy modeling that might be needed in your work. It's open source and it has probably 10,000 users worldwide. One very important aspect of the application is the focus on its performance and scalability, because it's not trivial to run an application, let's say, 100,000 times faster just because you have a supercomputer with 100,000 CPUs. And if you have one supercomputer with 2 million cores, it's really, really difficult to make best use of this, unless you put a lot of effort in how you structure the application, how it's working. And a lot of the work in Grombox has been on ensuring that all of these machines are very efficiently used and you get the best out of the supercomputing capabilities. So a few examples of what is Grombox is often used for. It's routinely used for simulations of membrane proteins, of different, let's say, cellulose or macro systems. I already mentioned there is work on simulating already whole cells, viruses or bacteria. It's also widely used in materials science for soft matter physics applications. And as you can see, we are getting at a level that we are able to simulate systems of 500 million, even a billion particles. This is something unthinkable a decade ago. But in order to achieve that type of scalability and performance, one needs to put a lot of effort in the software engineering of the code. The way, once you have a simulation system like the one in the picture here, you need to cut this box, your simulation box, in many small boxes that you need to send to all these processors on the supercomputer. And to do this in a very efficient and straightforward way is not so easy. So a lot of the work is being done on implementing algorithms and methods of achieving that and being able to scale to hundreds of thousands of cores. This is a bit technical here. I will skip over that. One aspect I want to highlight also is that for the development of these applications, it's very important that we work very closely with the hardware vendors. And in Grombox, for example, we are working with all the major makers, NVIDIA, Intel, ARM, AMD, to ensure that the code runs fast on all the architectures. The Grombox development team is largely based in the Stockholm area, but there's a lot of contributors worldwide. One of the methods implemented in Grombox that is very useful and finds a lot of application is free energy calculations. And in BioXcel, we are developing a tool called PMX, which helps with the automation of the calculation of these free energy calculations. And what this tool allows you to do is you can do all sorts of alchemical transformations of mutations of amino acids or nucleotides, and also to simulate in very, very big detail the stability of protein-protein complexes or protein-ligand complexes which are needed for example in the development of new drugs. And efficient development of these code has allowed us to run very large-scale drug screening that in a matter of few days we were able to run over hundreds of different types of poses between ligands and the receptor proteins. With PMX, we have also done testing to see to what extent can we also scale in a high throughput fashion on Amazon cloud services. So even if you don't have access to HPC or for other reasons you might want to use cloud resources, you'll be able to make very efficient use and use it in a very straightforward, automated way. Another thing worth highlighting is that this software is open source, it's freely available, and what you can see from these plots here is that the accuracy that you get with our applications is on par, and in some cases even better than very expensive commercial software that is being used by a lot of pharmaceutical companies. And all of these applications that I will show later also are adopted by many of the pharma companies and biotech SMEs as well. PMX is developed mainly in Göttingen in the group of Bert de Groot. Another application also very popular is called Haddock, and it's used for integrative modeling. Haddock also has thousands of users worldwide. Initially was offered as a web server, and even now you can register a free account and test it. You can also deploy it locally, or we also have ready solutions to deploy on HPC, and you can run it in HPC fashion. So what does Haddock do? The main strength of Haddock is to work on protein-protein docking, and also ligands as well, where one needs to find what is the optimal configuration of two proteins interacting. And when you're searching for the optimal pose, one needs to explore a very wide energy conformational landscape. And this is often very time-consuming. It takes a lot of compute power if you want to do an extensive search. But what can speed up this tremendously is if you have additional information which you can incorporate in your conformational landscape search. And this is the information-driven search, which minimizes these walks in the conformational space that you are doing. And this is where we get to the integrative modeling aspects, where one can integrate information from experimental data, could be electron microscopy, could be NMR, many different kinds of data can be included in the application to speed up the process. You can also incorporate information derived from different computational modeling or simulations. And Haddock is offering this platform that allows you to use all of that in a very straightforward way. One can use it, as I said, through a web interface or also installed on supercomputers. Haddock is also one of the applications that consistently performs at the top of various blind screening competitions. For example, a recent one, the GPCR doc experiments. It's one of the best performing codes. As I mentioned, if you deploy Hadoop on a supercomputer, it's very efficient. You can also do a very large-scale, high throughput docking experiments. What you see here on this graph down in the left is each of these lines is a different node and how much is being used during the whole experiment. And this is what you typically see when you have a high throughput experiment, where most of the nodes run simultaneously and finish approximately at the not at the same time, but they don't have sustained performance during the whole duration. At the end of the run, you have various additional computation which only few of the nodes are doing. But most of the work is done in the first half of the session. And we show that you can use thousands of cores in an automated way. There is a new version, Hadoop 3, that is much more modular and much faster than previous versions. Hadoop is developed in the group of Alexander Bonven in Utrecht University. Now, typically when you do any computational modeling or simulations work for every scientific case, it's rare that one uses a single application or just a single tool. Usually you need to get the data from somewhere, maybe to convert it in a different format. You want to run some simulations, then you want to analyze the results, potentially go back, redo some of the calculations or do additional analysis. So there's a lot of steps that one needs to do. And some of the workflows that we typically do on a regular basis may look something similar to that. And you have different tools being incorporated there, or maybe the same tool being invoked in different setups. And usually in the biomolecular modeling simulation work, we need maybe 10 different tools to solve a single case. And traditionally, we tend to do a lot of the work manually, like the pre-processing. Then we would send the job to a large supercomputer, which is the bulk of the calculation, and it saves a lot of time. But then you need to get the data back, do analysis, and so forth. And this takes a lot of time. What would be very beneficial is if we can automate all of that, package all the applications into a single solution, a single workflow that can be readily deployed on the machine. For that, we have developed a framework called BioXL Building Blocks, where each application is part of a small module. And similar to Lego blocks, one is able to build their own workflow using these building blocks, so that in one go you can send the case to the supercomputer and just wait for the result to come back. As of the release from spring this year, we have a very large number of modules supporting a lot of the most commonly used applications in the area. BioBB is also, if you're interested, you can have a look at the website, and you can see a lot of different pre-made workflows that maybe they will not solve your case directly, but they are a good starting point to use as a template and build on top of them. The software is also open source. It's freely available, and there are templates which you can use to write your own modules if you are interested in expanding it. Maybe you have your own tools that you would like to use, and it's very easy to plug them into the framework. Now what we are showing here is how with BioXL Building Blocks one can build a workflow and run it in a parallel fashion on a supercomputer. What you see in the upper right is it shows how in the duration of the run, in the initial peak of the computing of the nodes, they have a sustained performance throughout the whole simulation, and they finish pretty much simultaneously at the end of the run, which ensures that the machine has been very well utilized during this simulation. BioBB is using additional tools called, in this case, it's called PyComps, application developed in Barcelona, to distribute the job across the different nodes in the cluster. And we've done several different showcases with looking at binding affinity and also running molecular dynamic simulations with BioBB, and we can show that we can readily run on thousands of course, this software. And the developer team is based in the Institute for Research in Biomedicine in Barcelona, the group of Modesto Orozco. So these are the main applications that we are developing in BioXcel. As I mentioned at the beginning of the presentation, this Center of Excellence, similar to the other COEs in the program, we're not only developing the codes, but also doing a lot in increasing the expertise of the community, helping novice users get on board with applications or training existing users to become better users, become power users, and make good use of these supercomputing resources that we have at hand. In BioXcel, we provide a number of services, support forums. We run a regular webinar series where we invite developers of popular applications or method developers to showcase their work. We provide extensive documentation, tutorials, hands-on workshops to the user communities. We organize a lot of training online, in person as well. And from this year, we are also starting an ambassador program where we are extending our outreach through the communities in Europe. We're slowly building up. Currently, we have around 20 countries in the Ambassador Council that we would like to engage closely with all these local communities. It's very difficult for a small team to be able to know and to get in contact with everybody. So we're looking for representatives in all of these countries, and I hope soon we will have a Polish representative as well. So thank you. This was my overview of BioXcel, and if you find any of these applications applicable to your research work, don't hesitate, please get in contact with us, and we'll see in what way we can be useful for your research. Thank you.
So thank you, Rosen, for your interesting speech. You are perfectly on time, so you left 10 minutes for questions. So let's open the discussion. Questions? So as a warm-up, I will ask two general questions. So first, in your personal opinion, subjectively, what was the biggest, the most important simulation you have ever performed? You can choose the context, the biggest resource consumption, the most important subject. Choose one.
For me, the most important simulation was of monoamine oxidase, a membrane protein, because this was the simulation I needed for my PhD. This was a crucial one. Other simulations, it's a very good question because different types of simulations have completely different problems associated with them. For example, for the large scale simulations, you end up with problems that you didn't prepare for, such as you run out of memory, or so you need to go back into the deep into the algorithm to see what's happening. Like I can simulate with 100 million atoms, but when I go to 150 million, it doesn't work. And then you realize that it's not a very simple fix. You need to go many steps behind and to redo the algorithms completely. It was something that I had one of the slides that I wanted to talk about, but I thought there won't be time for that. So when we want to go to exascale or yotta-scale eventually, we need to redo the algorithms in depth. Like it's not simple adding extra nodes or extra power. It doesn't work like that. Sometimes we need to completely redesign everything from scratch, especially with new processors, with new architectures. That's often the case. Yeah.
So you partially answered my second question, but I will ask it. Oh. So you work for the computing center. Mm-hmm. So, but you are I don't think that you consider yourself as a resource provider, rather as a software or solution provider, or maybe even as an end user. So from your perspective, what is the biggest difficulty in accessing nowadays HPC system, looking from the perspective of end user?
So there are two types of users, I would say. We have the novice users who are often a bit afraid of getting on HPC because they're not used to how the whole system works. When they're faced with the black screen, they need to submit jobs, there are schedulers. Some of these terminologies, it's a bit frustrating for some of them initially. Later, when you have very good utilization of the resource, you have a lot of users who submit their jobs, they're waiting in queue, and they sometimes they're asking like, oh, we have this big computer, but I can't use it because I have to wait, or they're not very familiar with how to properly set up their jobs to make the best use of the machine. It's not as simple as you would do it on a desktop when you do it on the supercomputer. You need to understand a bit more how the machine works and what are the best ways to set it up. And for that we have the experts who are helping in this, the level 2, level 3, the advanced user support. So the users at different stages of their expertise are facing different problems. So we, as a resource provider, or well, the HPC Center is a resource provider. BioXL is the software developing initiative. We need to cater to all of those people, build internal capacity and competences to support this broad range of users.
Thank you very much. We have a first question from the audience. So, Jacek Kolanowski, please go ahead.
Hi, thanks so much for this very exciting talk. I'm not a computational scientist, but I am very excited and eager user of those sort of solutions. I actually had initially a different question, but because Tomek provoked some sort of the discussion about the issues with accessing HPCs and so on, I wanted to maybe start off with asking you, you know, one way of going around it is what you guys do, so train people to know how to kind of, you know, submit the job properly, think about the job and so on and so on. But the another way of thinking about it could be to make it even simpler. So with the graphic, you know, interfaces, or for people who are scared of the black screens, but they are not scared of buttons and tiles and things like this. So how much work with your software and with your approaches goes into this simplification of the usage of those ones, even beyond, you know, towards the graphic interface-like or very more intuitive usage by people who are completely kind of like in the blind when they approach this?
Yeah, thank you. That's a very good question. So let's start from there. Gromox, the first code that I showed, it's probably the most popular one. It's been developed for over 20 years now, probably. And ever since I've started using Gromox developing, every year we get a question, oh, we want a graphical user interface. When is the GUI coming? Every single time. And there have been many attempts to build some GUIs around it, not very advanced ones. So it's a tricky topic because if you it's not very difficult to build a GUI, right? It's straightforward. But if you want to make efficient use on HPC, the GUI will not save you because you will still need to know what settings to put to submit the job. So you can write it by hand in a configuration file, or you can use a drop-down menu to select a value. What we are trying to do now, for example, with the BioXL building blocks, is to build workflows, to build this automation, and the way we see it as most beneficial is the following. We make the platform, we make these building blocks. We also make example template workflows. And there are no two projects that are exactly the same. I simulate my monoming, you simulate something else, there will still a small tweak that you need to change. But this template should be easy enough to modify and adapt. Also, if you get a large user community on board that can share openly their templates for their workflows, once you build a lot of this big library, then we'll make it much easier for new users to come on board. I think this is a better way of doing it than just a single GUI for one application, because you always need additional tools. Just for one code is not enough.
Thank you so much. And just as a comment, I think the BioXL integrative tools which you showed are just absolutely exciting and amazing. And I will be happy to talk about them maybe later on, but I don't want to monopolize the discussion. I know there are other people wanting to ask questions. Thank you.
So we still have time for last quick question. Luisa.
The first question, is it free? Yeah? It's free? Free?
Yes, yes. All of those applications are free, open source.
Okay.
Yeah.
And do you consider including user feedback into developing of the software, or you rather think it's a complete solution, which is not developing anymore? So you're still developing the software and
yes. The software is
count the feedback from the users.
Yes, indeed. And if you go, so what we did in BioXL just before summer, we did the following. All these applications did surveys with the users, and we asked them, what do you think is a high priority feature that you would like to see implemented? And we got, say, for Gromox and Hadoop, for the bigger user bases, we got hundreds of responses, maybe 300-400 per code. Out of these feature requests, we selected, we prioritized them, and you can find on the website, if you go BioXL software, let's say Gromox, software development roadmap, you can see there the features that we are going to implement based on user feedback. And those will get implemented for sure. There's maybe like 15, 16. Out of this, we have a batch of 4 or 5 that we work on first. And we are doing these user surveys regularly so that we keep a pulse on what the community needs. We will not implement everything the community wants. We don't have the manpower. And also in some cases, somebody wants something that is valuable for. We want to implement features that will have a wider impact. The codes are open source, so we encourage also contributions from the community. They're on GitHub, so pull requests we very much welcome that. But yes, this user-driven development is a very fundamental part of our work.
Okay, thank you.
Okay, Rosen, thank you very much for your interesting presentation, inspiring one. It's my role to keep the time regime, so thank you very much. We have to switch to the second speaker, who is Aleksandra Pękowska. Aleksandra is the head of Discovery Center for Chromatin Biology and Epigenomics at Nęcki Institute of Experimental Biology, Polish Academy of Science in Warsaw. Her lab combines omics approaches to better understand the genomic basis of astrocyte functions and evaluation. And Aleksandra will give a presentation entitled Molecular Signature of Primate Astrocytes Reveals Pathways and Regulatory Changes Contributing to the Brain Evolution. Aleksandra, the floor is yours.
Thank you very much. This talk will be much more biological than bioinformatical or computational. However, we do also actually publish extensively all our code. So I invite you, whenever the paper is out, to visit our webpage, where we have everything that you can use to reproduce every single panel, every single figure of our papers. So let's switch gears to evolution. And we will be talking about a project that is within the framework of this Dioscuri program. Many people ask me what this is, and this is a collaborative initiative by the Max Planck Society, and it's financed mutually by the Polish and German Ministry of Science and Education. So it's essentially a very nice grant to start your team and fulfill the needs of challenging experimental workflow. So upfront, I would like to thank the people implicated in this work: Katarzyna, Ola, Bondita, Debdip, and Derek, who tackled the problem of astrocyte evolution. So we are in the lab interested in essentially how we came to be as species. So we are interested in brain evolution, and we address it from a slightly different perspective than typically people do, because most of the research is focusing on disentangling how it happened that our brain is so big and produces so many neurons. We address this question from a different angle, brain evolution. We are interested in the biology of astrocytes, and these are particular cells of the glial lineage. This is a cell type that has long been considered as purely homeostatic cell type. So in fact, it is a homeostatic cell type. It supports neurons in their function. It provides all the nutrients and basically precursors for neurotransmitters. But this sort of homeostatic functional view of astrocytes has changed over the past years, since we have discovered that astrocytes are active players shaping the formation, function, and pruning of synapses. So all the functions that are essential for higher level brain activity. And here's just to show you how deeply entwined astrocytes are with neurons. They basically touch and then shelf synapses and basically regulate what is going on in synapses. And there is a really load of papers that show not only a role in synapse biology, but also the possibility that astrocytes actively impact synapse activity. So in this project, we ask ourselves whether there is a link between astrocyte evolution and brain evolution. So do we know anything about it? And the answer is that there has been quite a remarkable evolutionary expansion of astrocytes. So you can see a rodent astrocyte is typically way smaller than a human astrocyte. But as you can also agree that, this is expected. Mouse or rodent in general is way smaller. However, you can appreciate how much more complex human astrocytes are. Here's an example of an astrocyte from brain cortex. So in the rodent brain, such astrocyte, which is called protoplasmic astrocyte, contacts roughly 200,000 synapses, which is pretty remarkable already. But in the human brain, this number goes up to 2 million. So a single astrocyte can contribute to functions of many synapses and potentially many circuits. And these morphological changes are translated into changes in activity pattern of genes. And a couple of years ago, people have published the first account of transcriptomic changes of astrocytes in humans, chimpanzees, and macaques, because this is essentially how we model evolution. So we can't model evolution in the dish in a sense that we can observe it. What we can do is we can compare us to our closest living relatives and infer the changes that might have been driving evolution of cells. So here the authors used post-mortem adult cortical tissue and identified the genes differentially expressed between species. And what was quite striking and was actually validated by others is that astrocytes and glial cells in general have changed even more than neurons, suggesting that the changes in gene expression pattern in these cells might be important for brain evolution. So when we started this project, we asked ourselves, but what about fetal astrocytes? And why is it interesting? Actually, genes that are important, that we know of, that are contributing to brain evolution, and neuropsychiatric and neurological disorders in general, are very often highly expressed in the fetal brain. So we thought that it would be interesting to address the fetal astrocyte evolution. So you can imagine that it's impossible to obtain a fetus from a chimp. This cannot happen. So we essentially have to model fetal astrocytes, and we do it by using induced pluripotent stem cells. So I have chimpanzee iPS cells, rhesus macaque, and of course human. So just to give you an example, here we have a relationship, evolutionary tree of humans. So our last common ancestor lived around 6 million years ago with chimpanzees, and around 26 million years ago with the rhesus macaque. So this is the evolutionary span that we are modeling with these iPS cells. And what I want to mention is that we have established this very nice collaboration with the Warsaw Zoo, and we have access to some leftover tissues from chimpanzee individuals, and thanks to this collaboration, we established dozens of lines, of iPS lines. So here I'm going to show you lines from this individual called Mandy. We have also other individuals. And we also collaborate broadly in Europe. We obtained chimp iPS cells from Svante Babo or from Rudiger Bär. So we implemented a protocol that is multi-step and pretty tedious, and we are obtaining routinely astrocytes in the dish. And we also accommodated this protocol, which was also not obvious to non-human primates. And I'm happy to discuss why we had to do it. Maybe for the sake of this presentation, let's move on. And just to show you that the astrocytes that we obtain are genuine, so they express the markers they should be expressing. They react to extracellular stimuli by mounting calcium waste. They take up glutamate, which is the reminiscence of their function in taking care of synapses in the brain. And they actually promote neuronal survival and synapse formation in vitro. So these are genuine astrocytes that we obtain in the dish now from these 3 species. Transcriptionally, these cells express markers that they should be expressing. So here's a panel of genes. And in order to obtain this plot, actually, we needed to construct what is called a consensus genome, which is a genome in which we basically align 3 species with each other and obtain information for gene expression using gene models in humans. And thanks to this, we are actually addressing here 60,000 genes in general. Here is just a couple of tens of genes that are astrocytic markers. So as you can see, these genes are expressed in the iPS-derived astrocytes in a very similar level as in primary brain, fetal brain astrocytes from humans, which validates our system. So we also looked at gene expression patterns genome-wide, and our cells are expressing fetal type of transcriptome. So now I've showed you that we have a tremendous expansion of astrocyte size and complexity in evolution when we take a human or rodent astrocyte from the brain. But can we recapitulate that in vitro? And that was a major question, because this would tell us whether our system is meaningful. So we compared human iPS-derived astrocyte with chimpanzee and macaque. I'm just showing you here human and chimpanzee. So what you can appreciate is that human cells are way bigger and more complex than chimp cells. And here's just a calculation of the area of cells, astrocytes, in 3 species. You can see that we produce larger astrocytes. And this is the case only for astrocytes because they're precursors, the so-called induced neural progenitor cells. They don't really differ in size. So it's something that is emerging upon the differentiation to astrocytes. Also, the astrocytic complexity is much enhanced, so we produce more primary branches, which can also be expressed in this analysis, which is called Scholl analysis. So we can recapitulate the salient features of astrocyte evolution, so now we can dwell on molecular signature of these cells and identify genes that have changed in evolution. And also, in the second part of the presentation, I would like to show you how we use this system to derive information as to how regulatory systems or elements or networks actually evolve. So first I'll give you just a couple of highlights in what changes in gene expression occurred in our system. So first of all, we see largely congruent changes in gene expression between human and chimp and human and macaque. This is something that we were expecting. This also shows us that we are comparing apples to apples. And most of the genes that are differentially expressed in a congruent fashion in both comparisons, they are also active in acutely purified brain astrocytes, which means that evolution occurs in a stepwise manner. And if we identify those genes that are expressed in brain-derived astrocytes and are differentially expressed between humans, chimps, and humans and macaques, we have a list of around 1,000 loci, which is twofold increased with respect to what is known in the adult brain. So one of the questions that is interesting is what genes might be driving the extended complexity and size of astrocytes in evolution. And we have identified a couple of interesting candidates. And one of these is TED3. TED3 is a transcriptional factor that is activated by the Hippo pathway that we heard of during this conference. And we find that TED3 is particularly upregulated in the human lineage, and that's in fetal astrocytes. Now, actually, this very same thing is also present in adult astrocytes in the brain, so this is something that is retained in development. And now, if we inhibit TED transcription factors in astrocytes, what we see is that astrocytes shrink and lose complexity, and these cells actually do not die. They survive this treatment. So we believe that TED3 might be implicated in expansion of size of astrocytes and their complexity. On the other hand, I told you that we were interested in the link between evolution and possible link between evolution and neurological disorders. So that's why we screened in our evolutionarily affected genes for genes that are related, have been related in the literature to neurological disorders. And we found quite an interesting observation that genes that are related to brain disorders, brain diseases, are particularly often downregulated in evolution. And this is true, for instance, for intellectual disability genes. These genes, in our data set, they are barely ever activated in evolution. They are typically downregulated transcriptionally. And we used a larger database of this intellectual disability-related genes. And out of 738 genes that have been related to ID, 36 are downregulated in evolution, only 1 is upregulated. So here's just a heat map showing those that are downregulated. And we, of course, wanted to confirm that using brain tissue. And we took advantage of this data that was very recently published, like 2 weeks ago, of transcriptomic profiling of middle temporal gyrus, which is this area here in the brain that is responsible in the human brain for language comprehension and integration of multimodal sensory information. And we compared so we identified genes that are over and underexpressed in human as compared to chimpanzee, gorilla, rhesus, or marmoset. And now what we see is, again, the overrepresentation of transcriptionally downregulated genes. So it seems that intellectual disability genes are vulnerable to transcriptional downregulation in brain evolution. So what you've seen so far is that the iAstrocyte system recapitulates the salient features of astrocyte evolution, that TED activity is, we believe, most likely essential to maintain astrocyte size and complexity, and that genes related to intellectual disability are frequently evolutionarily downregulated. And I think it sort of sparks interesting questions pertaining to the possible trade-offs between brain evolution and our mental capacities. So in the second part, I would like to show you how we can use this system to address more broad questions related to how regulatory systems evolve, how to produce, for instance, an enhancer, that is a sequence that activates gene expression in evolution. And just a little introduction. So genomes in mammalian systems are organized into functional domains. These are called topologically associating domains. And typically genes are regulated by regulatory elements, promoters that sit right in the beginning of the gene, and enhancers that can be found anywhere. And it's a big task for molecular biology to understand which enhancers regulate which promoters, because there is a specificity. However, we simply don't know how the system works. So what we know is that perhaps the organization of genomes into these topologically associating domains at least restrains enhancers from activating whatever gene, and basically channels their activity towards a particular set of genes. So essentially, we can use molecular biology technologies to map this kind of topologically associating domains. So here I'm showing you the HiC, which we use in the lab. And HiC is a method based on high-throughput sequencing, where we basically count the number of times sequences are close to each other in the space. And what you see is that the color reflects how frequently sequences are very close to each other. So from this, you can actually infer the three-dimensional structure of a genome and particular loci. So here's the locus ALDH1A3. It's an astrocytic gene. And here it's promoter. And there's plenty of elements that we can also annotate using ATAC-seq or ChIP-seq. And these elements most likely activate ALDH1A3. So we mapped genome topology in human astrocytes derived from iPS cells and in primary human astrocytes. So what you can appreciate is just randomly chosen chunk of human genome. What you can see here is that iAstrocytes recapitulate remarkably well what is happening in primary astrocytes. Sorry. So the system is really faithful. And what was really striking to us is how conserved the structure is. So you can basically align species looking at those topologically associating domains. So we have this data since a couple of months already, and very recent paper just published that chromatin structure is by and large conserved in primates, and we can only say that we see very similar thing. So we can focus on TAD annotation from humans. And what is important now also as corollary to this result that we obtained is that essentially regulation of transcription and evolution would rely on switching on and off enhancers. That wouldn't be so much dependent on potential changes in three-dimensional structure of chromatin. So in order to map enhancers, we used mapping of chromatin openness, ATAC-seq, and ChIP-seq. And here I'm showing you a snapshot of a piece of genome that encodes LMOD1. This is a gene that is transcriptionally upregulated. You can see it through this RNA-seq track. I'm sorry, I'm trying to get accustomed with this pointer and somehow doesn't work very well. Anyway, you can appreciate that we have really stronger signal in RNA-seq data in human than in chimp than in macaque. And we have this emergence of a regulatory element decorating LMOD1 locus in the human lineage. And all this is possible because we are aligning the data to this so-called consensus genome that I was mentioning. And we do that only for visualization purposes, because essentially when we look at evolution of regulatory elements, we then go to respective genomes. So what was really nice to see is that for most of genes, we can actually identify enhancers that are switched on and off in their vicinity that would regulate their expression. And what is really cool is that enhancers, the numbers of enhancers scale with the evolutionary distance, so the net gains of enhancers scale with how many enhancers we gained already in the past. So basically, we add enhancers or remove them sequentially in evolution. And the numbers, so net gains of enhancers, can explain the net expression increase or decrease. So here I'm showing you only the increase. So essentially, it's a very simple model system where you just add blocks to enhance, like a rheostat expression. So we can identify now these kind of cognate, let's call it, promoter-enhancer pairs. So elements that are switched on in the vicinity of transcriptionally upregulated genes. So here's another example, ROBTB3, actually a gene that might be related to cytoskeletal and the cell structure. And it's nicely upregulated in the human lineage, and you can see there's a couple of human-specific enhancers that all map to this domain. So we call these kind of events linked events. So we can link an enhancer to a possibly upregulated gene.
Time for conclusions. Just to leave time for at least one question.
Okay, sorry, because I can't see the time. Okay, so let me just quickly tell you that by comparing sequence of the linked and non-linked enhancers, we found a pattern of transcription factor changes that occur at regulatory elements that need to be switched on in order to activate enhancers in evolution. There's by far the only study like that, and we found that these factors are the so-called striped transcription factors that are implicated in regulating the strength of binding of tissue-specific regulators. And we used machine learning to actually prove that striped transcription factors are important for transcriptional onset of enhancers. So in conclusion, we can use modern tools to validate evolutionary changes that occur within regulatory elements that might be driving gene expression in evolution. And with this, I would like to acknowledge people that were implicated in the study, and collaborators from the Institute and from outside, and you for your attention.
Thank you very much. We still have 1 minute for questions. I fully agree with the first statement that this presentation was much more focused on biology than on IT, so please rescue me and ask a question.
First of all, congratulations on the results. I'm very impressed. I'm not sure whether I understand everything correctly, but I have one question. When you, regarding this differential expression analysis, in this analysis you compare genes differentially expressed in human versus macaque and in human versus chimp. And did you try perform the analysis in the way, let's say, from the perspective of rhesus versus chimp and rhesus versus human, because it would reflect the, higher distance of rhesus both from chimp and human, both in terms of brain size and evolutionary distance here, but because from rhesus we and chimps are cousins, so maybe...
Yes, of course. So you see many more differentially expressed genes when you compare human to rhesus and chimp to rhesus than when you compare human to chimp. So you capture this kind of evolutionary trend. Yes, but in this, so in this particular design that we follow, we are interested in the changes between human and non-human primates. So we are not that much interested in species-specific events, because it's very hard to disentangle them, because all the species will have their particularities, right?
Okay, but because I'm thinking from the perspective of the first question you asked, what drive the, let's say, evolution of brain size? Did I correctly understand?
So some people address that question. Yeah, perhaps we can also somehow contribute to that.
Okay.
But that wasn't our.
Yeah, of course, I understand. Thank you very much.
Thank you.
Okay, so let's thank the speaker. Thank you very much. So next two presentations will be given by scientists, and what is worth stretching out, clinicians from the Cardinal Stefan Wyszyński Institute of Cardiology in Warsaw. The institution will collaborate with, among others, in Mosaic projects. So the first speaker will be Professor Mariusz Kruk, and will tell us about advanced coronary CT image processing, benefits for patients and healthcare professionals. And then we will hear Jacek Kwieciński, AI support in diagnosis and treatment of patients with cardiovascular complications. Both presentations will show us the practical side of using modern IT technologies in today's medicine, and how the whole process of diagnosis and treatment of patients can benefit from broadly understood IT solutions. So, gentlemen, please go ahead.
Can we switch to my presentation? This is Jacek's. If we can just switch the last one, because we switched our positions. Oh yeah, that's it.
Trzecią z czwartą, jakbyście mogli zamienić miejscami. Tylko.
Hello, it's a great pleasure to be here and present our clinical approach to IT and AI solutions. I was feeling a little bit uneasy listening to the previous lectures. This is really very other level of abstraction for me. We are more concrete. We treat patients and just want some solutions from IT specialists. So let me present you the diagnostics capacity and benefits of using coronary CTA enhanced with modern post-processing using AI. As an introduction, I would underscore a role of CCTA in diagnostics of coronary disease. Coronary disease have remained the leading single cause of death worldwide and substantially contribute to loss of health and excess health system costs, despite the advances in treatment and diagnostics. Compelling clinical data pinpoint a dominant role of cardiac CT in diagnostics of patients with suspected coronary disease. According to current guidelines, actually all the patients that have any chest pain should be diagnosed with CCTA first. And indeed, over the next few years, and it's already happening, the request for CCTA will increase exponentially, and therefore the tools that can improve CCTA workflow pipeline, increase its diagnostic accuracy, or provide additional clinically relevant information should be embraced and introduced to clinical practice. CCTA uniquely provides images of both coronary artery lumen and its wall, which both are relevant. However, basically in coronary disease, it is the lumen that defines clinical symptoms and myocardial ischemia. High diagnostic accuracy for diagnosis of SAT, of coronary disease, is the main attribute of CCTA. It is not only the highest among the traditional non-invasive tests, but also brings some problems, especially with increasing volumes of the examination, because when anatomic testing is applied, like CCTA, the borderline stenoses, which do not define whether the patient should be further treated with invasive treatment, either PCI, stenting, or CABG, become a growing problem. The problem is that less than 20% of stenosis between 50 and 90% cause ischemia and should be treated invasively. And this means that around even 20 or 30% of patients diagnosed with CCTA have intermediate stenosis and should be diagnosed with other tests. The routine reference method for evaluation of ischemia of those borderline stenosis is an invasive test used in cath labs, which is called FFR. It is an invasive measurement of pressure gradient through the stenosis and involves both relatively high costs and a risk of rare but very serious complications. So how AI can provide an added value over the traditional CCTA to evaluation of a coronary stenosis? The latest technologies allow prediction of translesional gradients, so-called virtual FFR, based on CCTA images. The technology is currently recommended by American guidelines, not yet European, with very high level of recommendation in patients with chest pain undergoing CCTA and diagnosed with intermediate between 40 to 90% stenosis. The analysis is based on routine CCTA dataset and it does not require any additional acquisition or radiation or contrast or modification of drugs or patient participation at all. CT-FFR has proven to be an effective test in several trials comparing it to the reference invasive FFR test. First, FFR-CT solutions were based on computational fluid dynamics models, and it involved mathematical presumptions and models concerning coronary blood flow. Then required recalculation of these models with regard to the individual actual anatomics. Such approach costed multiple hours of computational time on then supercomputers. The first machine learning method of FFR-CT calculation was brought by Siemens a couple of years ago. The algorithm was trained using a database of 12,000 synthetically generated three-dimensional coronary anatomies, for which the CFD-based CT-FFR had been calculated. And then the CFD-based results formed training data for the machine learning CT-FFR models. This approach demonstrated very high diagnostic accuracy compared to the reference invasive FFR. The average computation time was down from several hours to 2.4 seconds for the entire coronary tree, thus providing near real-time evaluation of FFR, and this was of course a breakthrough. Comparative images for both traditional CFD-based and machine learning methods show how much the actual results were similar also on the individual lesion levels. Currently, AI-based CT-FFR solutions seem to be the standard. They are less computationally demanding. Across the literature, several providers of CT-FFR service with certified or otherwise validated AI products can be identified. Knowledge of CT-FFR result improves diagnostic workflows. As was shown, CT-FFR dramatically decreases the number of patients with normal coronary arteries referred to invasive diagnostics, which means that we actually get rid of invasive diagnostics only. And it translates into maintaining or increasing the proportion of patients undergoing invasive treatment. So it improves cath lab performance because the patients are not going through diagnostic tests but already go for intervention. CT-FFR is cost-effective just because it can prevent unnecessary invasive angiographies and thus reduce the costs of medical care, because invasive diagnostics is relatively expensive. CT-FFR also was shown to predict clinical outcomes. Recent meta-analysis of several trials showed that CT-FFR results correlate with the 12 months clinical events, which is always the ultimate validator of a diagnostic test clinical value. Very fresh data presented around two months ago strongly suggests that when applied in real-life settings, CT-FFR may even reduce mortality, including cardiovascular mortality, decrease the need for further testing, and improve cath lab efficiency by reduction of invasive diagnostics by around 5% and simultaneous increase of coronary interventions by around 8%. CT-FFR allows to simulate PCI outcomes, which is a very hot topic among cardiologists today.
2 minutes. 2 minutes.
Okay. FFR-CT adds another dimension to PCI planning, where we can see here the images of serial lesion functional evaluation and the proximal and more distal intermediate stenosis and corresponding pressure gradients. CT-FFR allows to evaluate strategies of selected either proximal or distal stenting, only to show that both lesions need to be stented in this case. Moreover, CCTA allows for risk evaluation. Crucially, what happens within the lumen is only secondary to what is going within the wall. Most of stenosis detected by functional methods, those causing ischemia, exceed 70%. These earlier stages of coronary atherosclerosis remain invisible for perfusion or invasive imaging, and CCTA allows imaging of vascular wall, which translates into the ability to detect atherosclerosis as well as spotting features related to high risk, like lipid core, like positive remodeling, like napkin ring. The presence of high-risk plaque on CCTA, irrespective of the stenosis grade, increases the risk of cardiovascular events in long-term follow-up, and therefore should qualify the patient for aggressive treatment even without significant stenosis. However, the reason we like CCTA is that its diagnostic use was shown also to improve outcomes. Patients with chest pain diagnosed with CT versus other functional tests has been shown to have dramatically improved prognosis simply due to more appropriate therapies applied following CCTA testing. How AI can help in cardiovascular risk determination? Plaque quantification and differentiation with traditional manual tools takes a lot of time, and AI-based automatic segmentation demonstrate a very high agreement with reference intravascular imaging. They are also able to automatically spot high-risk plaques. A recent study showed that AI QCT plaque staging may have improved prognostic value over traditional risk factors, calcium scoring, and manual CCTA evaluation. AI also makes feasible incorporation of additional multiple factors into analysis for improved diagnostic outcomes. In another recent study, machine learning models integrating all available clinical information, CT-FFR and plaque structure, showed that those data add to diagnostic value that can predict clinical event. So, in conclusion, the addition of AI-based tools to routine analysis of CCTA improves imaging workflow, decision-making in patient management, therapeutic guidance to effectively apply revascularization strategies, intervention outcome prediction, and clinical outcomes. Thank you very much.
Thank you very much. I was told that both presentations complement each other, and the second one is a logical continuation of the first one, so my proposition is to collect all the questions and ask them after the second presentation, so please go ahead.
Thank you so much, and it's really a great pleasure to be a part of such an event. And I've learned more about biology and proteins and simulation over the past hour than I did over my entire studies, and that was really something. So in modern medicine, what really happens is that we've got all these fancy tests, and it's more and more data. And I don't understand most of these results, but I know that I should consider all of them. And then at the point of care, it is really not trivial to integrate all this data and come up with a solution for a given patient, especially when I only have a quarter or so to do that decision making. So in reality, although modern medicine is fantastic, it can be overwhelming. And although initially we have a patient who actually has a problem, in clinical practice it's actually often the doctor who has issues. So we've heard about quantitative plaque analysis, how looking into the plaque in patients with coronary artery disease, the main killer, can really improve the management and ultimately the outcomes. But the way we were doing this back in 2017 up to 2020 really was moving all these dots, as you see on the screen, those tiny red dots, and you stick to a mouse and it takes an hour. You can get a PhD out of it, but you're not going to take it to the clinical setting. The way it works now is a bit simpler. So thanks to AI, we only need to define the beginning and end of a lesion and then allow some time for computing, and we will get the output within 20-40 seconds. And this is how actually AI can help us at the point of care to save some time and really make our discoveries translatable into the clinical setting. Then what happens when you're dealing with multimodality data? I spend a better part of my scientific life teasing out plaque activity from nuclear imaging from PET. And early on, we've noticed that all our findings are very much correlated with the morphology of the disease. That does make sense. If something looks ugly, it probably is active. But then, how to make sense out of it all? If you use conventional statistics and try to build a Cox proportional hazard model, at some point it will stop working because everything's so correlated. So this is where actually AI with decision tree algorithms can be very useful. So what we've seen is that although both methods can have prognostic implications and provide independent information, it is actually when you merge the data, combine both these approaches, that you can really make the most out of it. And what is nice about AI nowadays is that although people perceive it as a black box, we now have methods that give us some insights into what the variables were really driving the prognosis. And this is both true for these kind of easy and palatable decision tree algorithms that I work with on an everyday basis, but also for image-based learning, deep learning, where we look at images. We have the GradCam, which will actually highlight the area within a given patient which was driving the diagnosis. Now, the other thing that happens in medicine, which is fantastic, is that we have breakthrough therapies that change the landscape. But this translates into new populations. And then these new patients are completely different from what we are used to. So we need to have tools to better understand how to proceed with them. One such disease which is around for as long as people are on Earth is aortic stenosis, a disease which is all about progressive narrowing of the aortic valve, which then leads to an increased strain on the left ventricle. So what we need to do is simply replace the valve, easy peasy. But actually it's not that easy, and for decades it was only feasible with surgery. And obviously surgery has its own risk associated with the procedure. Fortunately, for now almost 2 decades, there's a new approach. We can do it with a transcatheter approach, simply going through the arteries and implanting a valve. And this allowed us actually to tackle patients who are very elderly, in their mid-80s. But such a population requires a different strategy for risk stratification. And we knew early on that in such patients, age is not a predictor of outcomes. You know, it's easy to stratify a population of a city just looking at the age. Or there can be tools, as in this case, which were very robust when looking at surgical patients. But when you moved into a new field of patients with your new device, which then translates into a new population, you need new tools. And as we're dealing with very elderly patients, which are now in the thousands in Poland, and globally over 300,000 procedures are performed annually, all these patients have a unique biology. And unfortunately, all the conventional clinical predictors will be, again, very much correlated. If someone is frail, probably their kidney function is going to be poor. So here, yet again, AI is actually lending us a hand and helping us to actually distinguish those patients who should be brought into the clinic earlier on for follow-up, so that we don't expose them to the risk of an adverse event early after the procedure. Because we know that actually roughly 12 to 13% of our patients are going to die within the first year after the procedure. And then the final thing. In medicine, we love evidence-based medicine. We want randomized clinical trials. We need to have convincing data. It's not just a single center study that we're going to accept. Even if we have a registry that spans across the country, people will still say that it has its limitations and inherited biases. And fortunately, a few months ago, and it was presented at a major cardiology conference a year ago, we have the first randomized clinical trial of AI, which was published in Nature by the team of David Uyang from Cedars-Sinai Medical Center. A very simple trial. But what happens is that when we do ultrasonography of the heart, which we refer to as echocardiography, in Western countries, it is not necessarily performed by a clinician, and typically at the first stage of analysis is done by a technician. And then these segmentations are then reviewed by a clinician. So what the team of researchers done here, they randomized over 3,000 patients to be either segmented by an AI tool or a tech, and then a clinician would make adjustments. And what they observed is that actually with AI, you're more robust with segmenting the heart than when it is done by human beings. Does all that mean that humans become irrelevant? Do we no longer need doctors? Well, definitely that's not going to be the case, as someone has to go to prison every now and then. And you're not going to lock in jail someone who created the algorithm. People say that with AI, it's all about the data that you use for training and validation. But then who is the person to blame? The people who provided the data, or the people who established the model? It will always be the clinician. But what AI can give you is a bit of a consultation. It can really help you re-figure out a given individual at the point of care. And we like the idea of the heart team in cardiology, which is just a consultation of clinicians. But people are in short supply. So I think it can be beneficial to have a tool that will alert you to some findings that might be relevant, but ultimately it is going to be your own decision. So undoubtedly, AI gives us an opportunity to take full advantage of the available data, because otherwise, when we have such an abundance of clinical tests, inevitably what's going to happen is we're going to focus on a few findings that we believe relevant, but we can miss a lot. And it can enhance patient management. And its implementation into the clinical practice is already underway, so we definitely need to get used to it. And with that, I would like to conclude. Thank you, and we're happy to take your questions.
Thank you very much. So we still have time for 1 or 2 questions. Who is going to start?
Sorry for my trivial question. I am not a clinician. Maybe I have a question to the first speaker. What does it mean that we have a significant stenosis? Is there any threshold between non-significant and significant?
Well, significant stenosis should be understood as functionally significant, which means that such a stenosis produces ischemia and should be treated. Looking at the anatomy, solely on the anatomy, lesions which make stenosis more than 90%, so really tight, most of them, nearly all, maybe all, are significant. They all produce ischemia. But the borderline stenosis or gray zone of stenosis anatomically is between 40 up to 90% of stenosis, which we don't know just looking at the stenosis grade whether they produce ischemia. So then we need another test that will tell whether the patient has ischemia, because if he has ischemia, he qualifies for invasive treatment, which is either stenting or CABG, bypass surgery. But the advantage of CCTA is that around 70 up to 80% of patients do not have stenosis exceeding 40%. Therefore, we can save those patients from any further testing. And additionally, we know whether they have atherosclerosis, which should be treated medically. So this is the advantage, and that's why we like CCTA, because it's just one test allows to effectively diagnose and treat very many patients and to reach those conclusions that we can reach with single CCTA, we had to use several tests before CCTA.
So it is not that you only calculate the percent of stenosis, but symptoms like ischemia is also important.
Actually, ischemia is the ultimate reason that we provide, that we know how we should treat the patient. So we need to confirm ischemia before putting a stent or directing patient for a CABG. We have to have confirmed ischemia, and the CTFFR just does it.
Okay, so I will ask the second question. Let me remind the audience that you are both clinicians, and I like asking controversial questions. So the question is, if the answer is positive, when you will be completely replaced by artificial intelligence? I'm asking the question because the same end is anticipated for my group, it means programmers. So the answer can be very short in one word, or you can elaborate longer on it.
The short will be, we are just plumbers, and they will not be replaced by AI so soon. AI will just help plumbers to perform their job more adequately, and in those patients that really need the plumbing job. So I just can say sorry to programists.
Thank you very much.
Maybe to the previous speaker, the second one. Yes. Okay, so I can continue talking. So I was thinking, of course, AI is a powerful tool, and I have no doubt about it, but I was thinking, how is it applicable on your regular practice? Because I can't really imagine in Polish system that, you'll laugh, so you know what I mean, that, you probably have to collect the data about the patient from multiple diagnosis tests and so on, ideally from the whole lifespan of the patient, and you put it into some sort of software, and it gives you some answer. So is it really applicable anyhow?
So in clinical practice, in particular situations, we are already forced to use some calculators that are on the website simply, where you input some key variables that are supposed to help you decide whether to proceed in one way or the other. So we already have to use such tools every now and then. So then the only question is what's behind that calculator or estimator, is it AI or not? And you can actually have AI models running on websites. It is all possible. So it's not the most convenient thing on earth. It would be if our software within hospitals would allow such data management, let's say, which would easily incorporate particular variables into models. But at the point where you have a trained model, it actually does work like a calculator. So we have such tools that are on the website. And for years now, we use such calculators in other scenarios which are not empowered with AI. So this is all doable.
Okay, so a more private question in a way. Yeah. So would you trust the AI outcome to give a treatment to the patient, or you would trust yourself more?
So this...
So go ahead, answer it, and we have to
okay.
go to the next presentation.
No, so I talked about in my presentation. For me, it's like another doctor talking to me, like it's a suggestion. But I trust my clinical judgment because it's my responsibility. So this is the way I would use it.
I believe otherwise. I believe that in cardiology and in medicine, evidence-based is the king. So if we show that AI is better than doctors, then we should even put in jail a doctor which didn't use AI, because we knew that AI would be better. So it goes into this way, and I believe it will be this way. I don't know in how many years, but ultimately evidence will prevail, not the doctors, the evidence.
Okay, we are out of time, so we have to go further. So next questions after the session in the lobby. Okay, so going further, Rosen started with a presentation on molecular simulations. Now we are going to the opposite end of the scale, so we will be talking about aspect of predicting of behavior of the whole populations. So let me invite a person I know very well from many, many years, Derek Groen. And Derek is from Department of Computer Science, Brunel University, London, where he works on modeling and simulations. Controversially, I will say that fortunately for Derek's career and research, and unfortunately for many, many other people, the research of Derek is becoming more and more important, relevant, and topical, not only in Africa, Asia, but now even in Europe. And Derek will tell us about simulating conflicts-driven population displacement using large-scale computing. Derek, go ahead.
Thank you very much. So I've got a pointer here and one for the slides here. So yeah, good afternoon, everybody. So I'm talking about a simulation approach to model conflict-driven migration. It's not AI-based. There are AI techniques as well, but the technique I work on is actually not AI-based. So why would we want to model conflict-driven migration? Let me start with that. When I became a lecturer at Brunel in 2015, there was the Syria crisis, and a lot of Syrians came to Europe. And at least in the British and the Dutch newspapers, everybody was surprised that they arrived. Nobody could have predicted that so many people would come to Europe, and I thought, this is really strange. And then I looked in the academic literature, and I realized that nobody was really predicting it. There were no simulations predicting how many people would be displaced if a war breaks out. Even though it's enormously important and was dominating all the headlines in all the countries in Europe. So that didn't make a whole lot of sense to me. So I started developing a model for that. So yeah, the idea is basically the model predicts a conflict erupts, people flee, where do they go? And there are various reasons why that is useful. First of all, it can help the preparations of NGOs, of governments to make sure that supplies are there before the people arrive rather than after. It also can give us an idea about how damaging border closures could be, or camp closures, or what happens if you move a camp, if you change its location, or if you change other types of policies. And in many areas, especially in Africa, there are whole regions where people don't have estimates of how many people are displaced there. And you could also use models to make estimations of, okay, how many people are really displaced? And then you don't only include the ones that you're counting empirically, but also the missing parts, so to speak. So the model that I started developing, but now we're developing it as a team, is called FLEE, and it's a so-called agent-based model. So what does that mean? Well, basically it means that every person is a digital object in the code. It's an agent object. These agents are placed in conflict zones, so areas where violence has occurred. So for example, this is an example from South Sudan, and all the conflict areas at some point, all areas that were a conflict at some point are marked in red. You place the agents there, and then there are rules about how humans behave and the agents likewise behave, and they move from place to place, more or less until they reach a safe haven, like a camp or a major settlement, where they can find shelter, food, and healthcare. FLEE predicts where people go. It doesn't necessarily predict how many people are being displaced. There are actually ethical concerns around that. So that's still very much an open topic. And what makes FLEE quite interesting is it doesn't need training, because it's not AI. You put your knowledge about human behavior in the model, you take geographic data to create these location graphs, and then you make your forecast based on how you think the conflict will go. And as a result, we're able to validate the same model against a lot of different conflicts. And in fact, we've validated it across 13 different conflicts. The model is also open source, and it can be used by anyone. There have been discussions around that as well, because it's a migration model. But actually, in discussions with NGOs, a lot of NGOs actually think the governments have these kind of models or even more advanced ones. So this is actually an effort to level the playground to make sure that also NGOs have access to this kind of forecasting. Okay, so very basic. I'll be very short about this, but every agent is a displaced person. And every agent, on a given day, they decide, okay, do I stay put or do I move? And if I decide to move, where do I move? And that's the way that FLEE works. You have conflict zones where agents, of course, are very likely to move because there's violence occurring. There are towns where the chance is lower, and then camps where the chance is very low. It's not zero because people do return from camps, for example, back to their home location if it's safe again. Now, what affects these decisions? Many factors. And the first version of FLEE was quite prescriptive and made very rigid assumptions about these factors. But the latest version, FLEE 3, actually allows users to define what factors they think are important that drive the migration movements. This is a more technical graph for to just nerd out a little bit. But basically for every day there are attractiveness scores for locations, links, new agents are spawned as conflicts occur. Then we do that travel modeling, then we complete the traveling, and then we write the logs essentially. So what do the results look like? So here FLEE is quite different to all the other models out there. FLEE predicts arrivals on a daily basis in individual locations. It doesn't always do that accurately on the individual location level, but across whole conflicts it does a reasonably good job. So the kind of forecasts that you can get, or validations in this case, are basically what you can see on the x-axis, the number of days elapsed in the simulation, which map to real dates, and on the y-axis in the left graphs, you can see the number of arrivals in each camp. In this case, you can see, for example, in Nigeria and in Mauritania, 2 camps, and then the blue line is the UNHCR data, as it has been observed, and the red line is the forecast made by FLEE in that same setting without prior training data. Then on the right, you can see error plots, where we use the averaged relative difference. Essentially, if that value is higher than 1.0, then more than 50% of the forecast is wrong, and the simulation is definitely not useful. So you want to stay well below that value, and ideally below 0.5. So as I mentioned, actually, there are a lot of conflicts, and I don't have the time to cover them all, but in terms of validation, the error we get between what the UNHCR observes and what we forecast with our code is variable, but in general, it ranges between a couple of percent and about 30%. Now, there are also different ways you can use FLEE. So, for example, you can take settings that are pretty close to optimal in terms of accuracy, but not in terms of realism. So in this case, agents will move quite fast, about 600 kilometers per day, and also the capacities are very low. You basically take the highest point at any point in the data. Now, if you make those constraints a bit more realistic, so you reduce the movement speed and you set the capacities of the camps a little bit higher, then the errors go up, but you actually have a more realistic simulation, so you get a fairer validation score. It's just really difficult to accurately validate. Another issue is actually the validation data itself. UNHCR collects data, but the data is often supplied by governments, so it can be biased, it has missing values, and all sorts of other issues. So you should see these percentages as being approximate, but what you can see is that in many cases, the code is able to have an error of 20% or less. You see one clear exception, which is Ukraine. So the error in Ukraine is much higher, and there are a couple of reasons for that. One important reason is that Ukraine actually had an active train network during the whole conflict. FLEE doesn't normally resolve train networks, it tends to look at roads, and there's some support for walking routes. So actually, I think one way that we could improve the accuracy of the code is to add in the train network. But there's a second important reason, and the second important reason is that I suspect that a lot of people in Ukraine knew in advance where they wanted to go. It's not like they were displaced in the panic. They probably saw what was coming and already decided, okay, I'm going to this location or I'm going to that location. So then it's also a different kind of algorithm. You have a different kind of behavior, and that you can see back in the arrow. Another example of a situation with high error is South Sudan 2016, where there are very large climate and flooding effects mixed with the migration situation. So there too you see higher errors. So it spreads out, but you can also see that overall the code does a reasonably good job in sort of forecasting what is going to happen given that a conflict erupts. Now, to run these forecasts, Flea takes some computational time, so we also parallelize the code so you can run it on a supercomputer. And basically you can use up to about 1,000 cores for simulations like that of Ukraine, which has up to 10 million agents basically. And the efficiency does go down, but basically if you use about 100 or a few hundred cores, then you've got a perfectly efficient simulation. And when you make these forecasts, you never run one simulation because conflicts can develop in different ways. You want to quantify for uncertainties and incorporate what-if scenarios if certain governments or other organizations intervene. Let's see. Yes, and then one more thing that I just wanted to mention is that we're about to publish a paper about the newest version of Flea, Flea 3. What is quite different about that code is that it has support for age, gender, ethnicity, and religion. So you can stratify and make forecasts for what kind of people are actually arriving. Like I said, the movement rules and the behavior can be defined much more flexibly, so we can also start using it for internal displacement modeling and for disaster-driven displacement. So we're looking into that as well. And we're also trying to have sort of AI-based optimization codes, for example, that you can advise an NGO about where to place a new camp when a conflict erupts or which intervention is the most effective. Yes, and in particular, the disaster-driven displacement is quite a broad collaboration with UNHCR, Columbia University, and even the UK Sciences and Technology Facilities Council, who kindly borrowed us a PhD student or lent us a PhD student for 6 months. And that's basically all I have. I hope I'm within time.
Exactly on time. So time for a few questions.
Thanks for this presentation. I wanted to ask, for me to understand it a bit better, that the underlying principle behind that model is the fact that people are scared and they move away. Is that what's kind of like, there's a lot of parameters of how the movement goes.
Yes, yeah.
But the underlying kind of reason for the movement needs to be
Yeah, so the vast majority of simulations we've done are conflict-driven displacement. So that means there's violence, people are unsafe, they want to flee from the violence and they seek safety. So that is the use case where Flea has been validated. We're now broadening that, but that is the core situation. And yeah, so they tend to call that primary displacement. So it's the displacement from a country of conflict to the adjacent countries.
So I wanted to ask that because you already hinted into that you go into the natural catastrophe-driven, for example, displacement and other things like this. You can broaden that. And it's almost kind of inappropriate to say that, but could you also use the same model, changing a bit of the principles of movement, but using the same model, the same tool, to start into looking into a more leisurely-driven movements, or movements caused by the economic parameters or other things like that?
Yes.
I just don't understand how deep what's behind this so much, whether that's transferable or not.
Yes, so you can definitely do that with Flea 3. The only thing that you would need to do is figure out, okay, what are conceptually the rules for economic push and pull factors, and then someone in the team would code that in. But you can definitely do that. Why are we particularly interested in conflict? Of course, because of the original situation. But there's another thing worth considering, that when migration is very gradual, actually a lot of the AI-based approaches do a fairly good job. But when you have sudden events of migration, basically most of the AI-based migration models, they say, well, I get very good results unless something unusual happens, and then the model doesn't work anymore. But that's when you need the model. So we tend to focus on the unusual situations, but you can also do the usual situations.
Luisa?
My question is a little bit related to what Jacek asked, that because there are so many determinants of people migrations in history, so it's difficult to include all of them, yeah? So you just mainly focused on the determinants which are in the place of the conflict, yeah? Not all over the world, yeah?
Yeah, so the original scenario is conflict-driven displacement. So then, yes, there are still many factors at play, but there are only a couple of factors that are really important. And we showcase that by only putting in those factors and showing that we are validating reasonably well. Now, the latest version of the code can support other factors as well. We'll have to see how important they are, and that's still very much open research. So, yeah, when it's really primarily conflict-driven displacement, there are only a couple of factors that are truly important, with Ukraine being a notable exception because of all these other circumstances. But for example, if you're talking about internal displacement, economics-driven migration, you probably have to incorporate many more factors in the code. And the code does support that now, but we haven't constructed those simulations yet.
Have you included decisions of politicians? Or something like this?
Yes.
Okay.
Of course, the consequence of their decisions, and we don't model the Senate or something, but if they do a border closure, or if people are not allowed by car, or if they forward people from one location to another, all those things we can put directly in the simulation.
Could such model be used for example, location of refugees into better places, maybe? Not try to perceive where should they go, yeah? Where the best solutions.
Well, actually, there has been an EU call on that topic about sort of figuring out where to place migrants or refugees. There are huge ethical concerns around it. That hasn't stopped the European Commission from funding the research. So yes, computationally it can be done. Whether it's ethically feasible, I guess we'll find out in the next few years, because there is a project funded that's going to work on that.
So maybe I will go further, asking almost the same question. If you take into consideration in your research, in your approach, the fact that one conflict can trigger another one, and that one migration can influence another migration.
So you mean does the code have does the code support these chains, or am I afraid that there are consequences to the forecast I'm making? Which way?
The question is if you consider every conflict separately, or you can consider 2 or 3 conflicts together.
Okay. So in IT flows, the setup was originally that there would be a large-scale model about migration towards the EU that was an AI-based model, and then with a small-scale model that would model the individual conflicts separately. Now what happened is that large-scale and the small-scale model never worked together because the large-scale model doesn't have any notion of space. It doesn't have a location graph, it just does point-to-point interactions. It is possible to make a flea model on a larger scale that you also include the secondary migration and incorporate multiple conflicts. But secondary migration has very different push and pull factors than primary migration, because in secondary migration you're already in the adjacent country, so you're not being displaced by conflict anymore, but you're displaced by other factors. So there's some research underneath that we need to do to make those models. But mechanistically it's supported, but the simulations haven't been built on that topic. So it's a very good one, yeah.
Thank you very much. So joking at the end, I could ask a question. If you take into consideration the result of Brexit, of people movement, because Derek is from UK, many people moved or left UK after Brexit, so.
Yeah, when Brexit happened, I had a lot of EU-funded projects, so my attention was firmly directed towards modeling the EU. I think if those projects wouldn't have been funded, I probably would have written a paper about how people have been displaced by Brexit, and in particular academics, I think, have felt a clear push factor based on Brexit. On the upside, the UK is at least backing Horizon Europe next year, so that's a tiny step back, I guess.
Okay, Drake, thank you very much.
Okay.
So time for the last but not the least presentation that will be given by Alexander Martin from Orca Computing, a manufacturer of photonic quantum computer. For the last 2 years, he has been there as a researcher at the interface between classical machine learning and quantum mechanics. And this was announced yesterday or 2 days ago by Chris Kurowski. This year we are expecting to have to Orca Computing as a first data center in Poland. So please tell us what they can be used for. Go ahead.
Yeah, great question. Hi, I'm Alex. I'm going to try and answer a bit of that question today. I'm going to talk about what our near-term quantum computer looks like, so how these devices that are being delivered to the supercomputing center here soon actually work. I'm also going to talk about what they can actually be used for. And finally, I'm going to motivate some applications in life sciences, looking at some work that we've already done at Orca in our algorithms development team, but also looking at some potential future work that could be built on and what you could do with an even bigger and even better quantum computer. So starting off with the first one of those, what does a near-term quantum computer look like? It looks a little bit like this. So we have on the left some photon sources that are firing these red single photons into a circuit consisting of multiple channels. Each one of these channels can interact with each other, and we can tune the level of those interactions. What that enables us to do is encode information into the quantum state of light, and at the end, we measure it. So you can see a vector representation here as well of how we, sorry, let me just have a look at this pointer. There we go. You can see this vector representation as well of how we look at these states. So here, there's one photon in each one of these channels. So we have a vector of all ones. At the end, we could get multiple different state vectors out of any one input state. This is because of the fundamental probabilistic nature of quantum mechanical measurements. These different measurement outcomes will be distributed by a thing that we call the Born rule. An important detail is that working out these measurements at the end, given an input state, is hard to simulate classically. That makes the output distribution very complex, and it means that it can have correlations that cannot be obtained by classical sampling processes. So how can we use these classical sampling processes? Well, at Orca, we've already developed a few applications, and here you see kind of the three broad classes of them. One of them being combinatorial optimization for binary optimization problems, the next being image classification problems, and I'm not going to dwell too much on them today. I'm just going to focus on the last one, which is generative modeling. For our generative models, we focus on GANs, and we include the best of classical machine learning and quantum machine learning into one algorithm. And we can see performance improvements on some data sets, and I'm going to explain exactly which data sets we expect to see that on later on in the presentation. Okay, so let's start off with what is a GAN. So a GAN is a generative adversarial network. It's a generative model that doesn't necessarily need any quantum computer, right? They are already an existing technique in classical machine learning. It consists of two different neural networks, a generator and a discriminator, that are trained with opposite objectives. The generator's goal is to create fake data that the discriminator will think is real, and the discriminator's job is to tell apart the data that it's shown, and it's going to be shown both real and fake data. The way the generator does this is that we feed it a random noise vector, and it translates it into something. And in the training loop, it starts to learn exactly what it should be translating it into. Once we've finished this training loop, we can throw away the discriminator, and we just use the generator to create new data. This generator, every time we use it, still requires a random latent vector. Now, this random latent vector is where the quantum computer can come in and can be useful. We already know that these latent vectors in classical machine learning can encode important aspects of the data. So some results that we call style GAN showed this quite succinctly. Here you can show an example where if we vary some of that latent vector that we put in, we can see a smooth transition in the data that is created. So, for example, these faces created by a GAN use latent vectors that are kind of take a walk through latent space. So, for example, in the top one, as the hair moves across, maybe one part of the noise that was fed in, the latent vector noise that was fed into the model, is changed smoothly. On the second one, you can see it with, a similar change with glasses. The key takeaway from this is that the generative model is linked to the latent space that you seed it with. And in fact, GANs work better when they are given a latent space that matches the probability distribution of their data. This is where the quantum computer comes back in. We have that quantum computer that is creating a highly complicated distribution that can't be simulated classically. Well, what if there's already data out there that you would want to generate that shares those statistics, right? What if it's data from, for example, quantum processors that you want to be able to generate replicas of? Could we use this quantum computer to enhance the capabilities of that model? So what we do, you can see a very similar diagram to the one earlier. What we do is we sub out that latent vector, which normally is randomly generated Gaussian noise, with a quantum latent space, and that is those measurement results from the boson sampling device that I showed you earlier. This is a little diagram of exactly what that quantum computer will look like when it's delivered to the supercomputing center here. It's rack mounted and kind of fits into a normal data center environment. It's a relatively compact platform for a quantum computer. Some of you may be familiar with photos of kind of large devices the size of a room that look like an enormous golden chandelier. This device is built slightly differently to that. And so we use the measurements from this device in this hybrid GAN setup. Just to show an example of how this actually works, so here if we want to create a fake image of a fake handwritten digit image, this is a kind of standard task when playing around with new image machine learning tools. If we were to take a classical model, focusing on this latent space over here on the left-hand side, we would take that randomly generated Gaussian noise, feed it through fully collected layers, and then feed the resultant latent vector into a convolutional neural network to generate our image. The only change that we make for the quantum is that we sub in our PT series device here. And the results on this kind of toy data set, here on the y-axis you can see a kind of measure of generated image quality, and on the x-axis you can see as we go through the training. The blue line representing the quantum enhanced model, the orange one representing a kind of regular classical generative model. The quantum latent space yields some slightly better results on this toy data set. This is a good motivation for kind of taking it a bit further to something more complex, perhaps. And that's where these applications in life sciences come in. So far, we've looked at hybrid GANs for novel conformer generation. So we use a graph neural network to represent the structure of a given molecule, and we seed that with a latent space from a quantum computer, and we've seen minor improvements in the conformers that are generated from that model. We then want to expand this kind of work to look more generally at image generation for scans. In particular, on the left-hand side, you can see some work that's been done on expanding and improving data sets of medical image scans. We also want to add to this conditional GANs, where you could feed in as a condition to that generative network, for example, one of these images, and it could be used to extract the features out of it to help clinicians or researchers study them. And then on the right-hand side, inspired by this conformal work, we're interested in looking at larger molecules, looking at protein design by, again, encoding them in graph neural networks. Finally, I want to talk about something which those of you who may already know a bit about quantum computing may have been wondering about, which is kind of where are the qubits? This is the kind of fundamental unit of quantum information that we use in information processing. Our current devices don't use qubits because we're in I guess what we call the kind of NISQ era, the near intermediate scale quantum device era. And as we look forward, we will be building devices with logical qubits, although they are many years away, at the very least 5, potentially more. But such a device will be able to perform simulation on a much larger scale, and that will help in applications where exact simulation of quantum systems are desired, whether that's drug design or designing new nanotechnologies. On the right-hand side, you can see an example of the kind of structures that we're trying to build in order to coherently control the quantum information for these large devices. So that's kind of all from me. That's an overview of our hybrid quantum classical models. And I just leave you with a GIF of what it looks like to actually move through a quantum latent space. So as you vary the measurement outcomes from a given quantum state, what do the actual outputs of a neural network, how do the actual outputs of your classical neural network change? And here you can see it when you can see it for a generator that's been trained on that handwritten digit data set.
Thank you very much, Alexander. So, time for questions. Please ask them. Otherwise, I will have to force our quantum team to ask the questions they probably know answers to. So, any questions? If not, I will start. Okay, so I will start and then you will be the next one. So many times, many institutions, many companies announce the supremacy of quantum computers. I know and I'm fully aware that it depends on the scenario, let's say. But in that case, it's not a question if, but only when. So when do you predict that quantum computers will be generally faster, better than, let's say, conventional or analog computers?
Yeah, so I guess in my slides I said at least 5 years. I would expect that for a wide range of programs it will be even longer than that. However, I think one thing that often isn't communicated about quantum computing is that it will only ever be used for a specific set of tasks. To run a quantum computer, you need a quantum algorithm, and it's not a panacea to solve kind of any large computational problem you have. That is unlikely to ever be the case. And in the near term, before then, I think what we expect to see is just these specific cases of where you can use quantum computers to enhance already quite strong classical approaches.
Thanks. Thanks for this presentation. I wanted to ask about one of the aspects which you hinted on, and I think throughout the whole sessions we have heard that, for example, for the AI systems, but also here, we always talk about the here you call it inception rate or something like that. So the correctness of the system, of the recognition of whether something is false or not, and so on. And I think we often focus on, the true positives versus everything. But in many applications, for example, in clinics, but also in some other applications, actually it might be much more important to have zero false negatives, or false positives, or zero, or zero false negatives, or things like this. So this might be more important than the actual accuracy. So I wonder has such work been done to compare on this similar problem, quantum versus classical approaches, and what are you getting out of this in terms of not only the accuracy, let's say, but also rates of false negatives, false positives?
Okay, yeah, it's a good question, and it's an important question as well when you consider that these actually need to be deployed one day. On the specific case of image generation, scoring the quality of the images is kind of a bit of an active debate in the community of the best way to do it. And so if, for example, you wanted to feed it into, you wanted to judge the quality of something like a classifier, maybe that's a bit simpler than an image generator. I guess it's just a matter of how do you actually train the algorithm? What loss function do you put in? For example, in GANs, you can actually introduce quite a high penalty for undesirable characteristics or problems. A recent example that comes to mind is GANs, in particular, used to generate quite accurate but blurry images. So, they'd be quite hard to actually interpret, even if the underlying structures were correct. And you can just add a penalty term to your loss function that explicitly detects that explicitly penalizes any model that starts to generate images that are too blurry, for example. So techniques like that could help in tailoring models to kind of do more exactly what people want in that direction.
Is there any intrinsic feature of quantum versus classical that would be favoring one over the other, or no, it's more about rather putting the criteria?
Yeah, no, it's not necessarily a kind of quantum versus classical distinction, that kind of problem.
So last chance to Mateusz, go ahead.
So I have a question of my own. So we will have 2 ORCA quantum computers here arriving at Poznań. So did you test it? Are there any advantages to enlarging the feature space of the, like using more photons, basically?
Yeah, so in this explicit example of GANs here, increasing the number of photons in the quantum computer corresponds to increasing the dimension of the latent space that you're projecting your data into, which in all cases, given the small scales of these kind of devices, will always be helpful, given the current numbers. There are, of course, limits to that. But yeah, so for this specific example, yes, there are benefits, and that is one of them, is that you have a kind of more expressive model. More generally, are there benefits? Yes, in all of the algorithms I've mentioned, more photons, more modes are better.
Did you experiment with using 2 quantum computers, or are there any problems with running simultaneous calculations on both?
So with the 2 that we're delivering, there's kind of 2 ways you can use them. You could either use them in parallel to kind of speed up your computations, as is well-established practice for classical computing, and we've tested that. The other thing that you could do is you can connect them in such a way as you, not quite, but you effectively have one device that is kind of twice as big. And we definitely experimented with that, and we've experimented with what the performance degradation is compared to actually having a device that is twice as big. And it comes pretty close to that as well. So it can be used in both ways.
Thanks.
Okay. Great. Okay, if there are no more questions.
So if there are no more questions, let's thank again all the speakers.
Thank you.
And also the technical team directing light to our faces and providing the high quality sound and streaming. So Eric, thank you very much. And I'm passing the microphone to the organizer.
Okay, I have an unexpected pleasure to close the conference. And I see the audience migrated somewhere. I don't know why. Maybe we need a model to answer the question. But I think that those of you who decided to be here till the end of the conference are satisfied now, and that the very interesting lectures we could listen to in the last session compensated your time. And I hope it will be opportunity to come back and to meet again, because regarding the title of the conference, Understand and Describe Life, we discussed about life in different aspects. But I'm afraid that we still cannot understand everything. And it's very good and optimistic, because we still have much to do. So thanks a lot, and I think we will see soon. Thank you very much.